how's the spotted lanternfly situation in Pennsylvania?
It's still pretty bad.
Not good.
Oh, thank you. Oh my gosh.
It's killing grapevines.
Okay.
It's spreading.
Okay, wow.
Yeah, it's not good.
I believe the population dynamics in Korea is quite stable and low compared to, let's say, 10 years ago, something like that. So sometimes it's really harder to find the spotted lanternfly, even on the tree of heaven.
And we'd like to know why that. We'd like to know why that is.
We want to do some studies in Korea and find out why that is, so maybe we can replicate it.
I hope so.
So is there any scientist who study population genetics on spotted lanternfly? Is there any scientist?
I think there are a couple people at Penn State University who are doing something. I'm not sure exactly who they are. Do you know, Julie or Hannah?
I don't. We're doing genetics on the parasitoid, the egg parasitoid, but not on spotted lanternfly itself.
Okay.
Okay, probably it is almost time. Yes, it is almost time for starting our symposium. But still, not a big groups, big members participating. But anyway, we can start. We have probably you have the programs I released. And then we, in the morning session symposium for the name is the Quarantine Issues on Exotic Pests and the International Year of Plant Health 2020. We have 7 speakers, and then we will have 4 talks, and then having about 10 minutes break. And after then, we will have another 3 talks. And yes, and every speakers can have average 20 minutes, but depending on the question or the content. The first speaker by Dr. Kim Holmo and the second speaker by Julie Gold will have more time, or 5 minutes more or 10 minutes more. But it is almost 20 minutes. So maybe I ask you, the speakers, please make your presentation in not more than 15 minutes. Then maybe that's why we have that. In that case, we can have at least 5 minutes. The discussions. That's the first thing I have to ask every speakers. Okay, I think we have to start right now because almost time. So let me introduce first speaker, Dr. Kim Holmo from ARS USA. He will talk about the update on the promising agent for the biological control of spotted winged Drosophila. So Kim, are you ready?
I am. Yeah.
Yes. Oh, yeah.
Yes. Do you see my screen?
Yes, it's good. Yes.
Okay.
You can start. Yes.
Well, thank you for asking me to participate in the symposium, and I'm sorry that we can't be there in person and have to do it virtually. So I'm going to give a brief update on the status of biological control agents for spotted winged Drosophila. And you'll see this abbreviated on many of my slides as SWD. So this has definitely been a team effort. Spotted winged Drosophila was discovered in North America about 20 years ago, and particularly in the last 10 years or so, there's been a tremendous outpouring or tremendous interest in research on biological control of spotted winged Drosophila, and there have already been more than 100 published scientific studies on natural enemies of spotted-winged Drosophila, and I'll try to summarize the most important points today in this talk. A lot of this information is just coming out in two recent articles, one which was just published late last year in the Journal of Integrated Pest Management on current and pending tactics in biocontrol, and another review of biocontrol of spotted-winged Drosophila that is coming out shortly in CAB reviews. So there are several different biological control approaches, and they all depend to some degree on the fact that non-crop reservoirs are very important in spotted-winged Drosophila populations. So these serve as tremendous reservoirs for reinfestation or infestation of crops, and then crops then go back into the non-crop reservoirs. So biological control really has to be very effective in contributing to reducing populations in these reservoir areas. One of the main approaches we like to look for first is classical biological control, in which we're relying on adapted native natural enemies from Asia that could be regarded as safe enough to release into the invaded areas. Also, there's augmentative biological control, which could be done either with the Asian natural enemies or with resident natural enemies that occur in the invaded ranges. And then conservation biological control, which relies on preserving and encouraging populations of locally occurring natural enemies through a variety of cultural and manipulative management. And this can also include application of entomopathogens, which are often treated as biological insecticides, but there are a variety of techniques that can be used, and we'll mention a little bit of some of the research that's been done in that area. We know a lot about Drosophila parasitoids in general because of the widespread use of Drosophila as a lab animal. Quite a bit is known about parasitoids of certain species of Drosophila, in particular Drosophila melanogaster. And well over 50 species of parasitoids are known now that attack various species of Drosophila. These include larval parasites, most of which are Burconids in the genus Asobara, or Phagidids in Leptopolina and Ganaspis. And others are pupal parasitoids in a variety of genera, mostly pteromalids, but also this Trichopria, Drosophila and related species, and Diapriidae. There are no known egg parasites or adult parasites of Drosophila. When Drosophila Suzukii first appeared in North America and then also in Europe, extensive surveys were started to see whether or not there were any native natural enemies in the invaded ranges that could be contributing significant amounts of mortality to spotted-winged Drosophila. And in North America, we know that there are over half a dozen species of larval parasitoids and several species of pupal parasitoids that will attack spotted-winged Drosophila. However, none of the larval parasitoids successfully develop to any degree or to an important degree from spotted-winged Drosophila. And the reason is because of the spotted-winged Drosophila's strong physiological defense against invading organisms. Parasitoid eggs are typically encapsulated very early on, and you can see them on this slide here as the dark spots. So the encapsulated eggs or young larvae never develop to full maturity, so the parasite dies, and generally the fly is capable of continuing its life, and it doesn't kill the parasite when the encapsulation occurs that early in its life. On the other hand, the pupal parasites readily attack and develop quite well in spotted-winged Drosophila. But unfortunately, the rate of attack in field populations tends to be very, very low, usually not more than a couple of % in natural populations. These two pupal parasites also are very, very widespread, and they're virtually cosmopolitan, and the same two species are found in the native range of spotted-winged Drosophila in Asian countries, as well as most of the invaded ranges. And it's not that they've been following spotted-winged Drosophila around the world, but they have a very wide host range, as I'll mention in a moment. So just as the surveys in North America have shown, surveys of parasitoids that could be attacking Drosophila suzuki in Europe showed the same thing. Extensive surveys found that only the two cosmopolitan pupal parasites were able to develop in Europe on Drosophila suzuki. And there were a number of larval parasitoids that would try to attack Drosophila suzuki larvae, but the high immune response resistance of Suzuki prevented them from successfully developing. So both for North America and Europe, with the lack of effective native natural enemies, interest turned to classical biological control and the possibility of finding effective natural enemies in the native range in China and Korea, and to some extent in Japan. So our next step was to conduct explorations with cooperators in Korea and China, and beginning in 2013 and continuing for several years, for 2014, trips in 2016 and 17, in South Korea and in China. And many of these trips were made in collaboration with South Korean and Chinese scientists, some of whom are probably attending the Korean Society meetings now and might be listening in. And you can see the collection points in different years in South Korea and in China. Most of our collections in China were made in Yunnan province, where there are high resident populations, but there have been other collections made in some of the other provinces as well. But they find pretty much the same agents that we find in Yunnan and as well as in South Korea. So after these half a dozen years or so of prospection in South Korea and in China, we have a pretty good idea now of the diversity of natural enemies that are found. And this is what we find in our studies. There are the Asobara japonica, the braconnid, and two species of phygitids, Leptopolina japonica and Ganaspis brasiliensis, that form the majority of parasites that are found both in South Korea and in China. And in particular, Ganaspis brasiliensis is the dominant in most locations, forming the largest proportion of parasites in the populations of fly. And over the past decade, considerable numbers of laboratory studies have been conducted on these three species, Asobara and the two phygitids, Ganaspis and Leptopolina, studies on the various aspects of the biology and the efficacy, host specificity studies for safety, and of course these are particularly important for the possibility of obtaining permission to release an exotic agent in a new area such as North America, on their climatic adaptability and their competitive interactions and so on. A lot of these, the largest proportion of these studies have been done in Kent Dana's lab at the University of California, Berkeley, in the field station in the Central Valley. And Shinkeng Wang was important in conducting a lot of those when he was at Kent's lab before he came to our lab. This slide shows the summary of the most important results. The top part shows the host specificity of the primary agents that were tested. And you can see this is a list of the non-target species that were tested, about 24 species of Drosophilids, 2 pupal parasites. This Pachycropoideus is a pteromalid and Trichopria is the diapriid. And you can see that both of them attack nearly all of the non-target host test species in pretty good numbers. So these are generalists, and they exist in the environment because they attack so many different species of fly pupae. These over here are the larval parasites, and you can see that among the same group of non-target test species, the Brachanid, Asobara japonica, is also pretty much a generalist in terms of its host use. It attacks a wide variety of Drosophilid species. On the other hand, the two Phagidids, Leptopolina and Ganaspis brasiliensis, are very, very specific. And they attack only the species that are closely related to Drosophilus suzuki'i, melanogaster, and simulans, and in the field, a couple of other ones that weren't part of this test list but occur in Asia. Some of the slides down here show the results of some of the other biological studies, the fecundity of Asobara versus the Leptopolina as a Phagidid, the olfactometer studies with these wide tube olfactometers that show that the species respond very strongly to odors that are produced by the host berries, blueberries or raspberries or blackberries, whether or not they're attacked by spotted-winged Drosophila. They have a very strong response. So quite a bit is known about the biology of these species now, and as a result, we chose to submit a petition for field release of Ganaspis brasiliensis. This has been pending now for several years because it takes a couple of years to go through the review process. The initial petition submission was sent back asking for more information, and then it was resubmitted in 2019, and word has come back that it's been tentatively approved and it's now undergoing the final public comment period. And if no new objections are raised, we hope to be able to receive permission to make field releases beginning in the 2021 field season. So that's very encouraging. And meanwhile, very interestingly, last year, it was reported that these same two Phagidid species and another exotic species of Asobara were discovered in the wild in Canada and British Columbia. So they now exist as adventive populations in North America. And there's the potential that these should eventually spread into adjacent parts of the US from Canada. So it'll be very interesting to see how these populations spread and what they do on their own. Now, in addition to classical biocontrol, we have the possibility for augmenting, and there's been quite a bit of interest in the potential for augmenting native parasitoids that don't need permission for field release. And this would include the two cosmopolitan pupal parasites, Trichopria and Pachycropoideus. So a number of field studies have already been conducted in different European countries, in Mexico, and in California. And there's strong interest from some commercial insectaries, particularly in Europe, in the possibility of producing some of these, such as Trichopria, and selling them to producers for release into protected crops or covered crops. This picture here in the lower left-hand corner shows an example of an enclosed crop where the crops at risk are grown under some sort of screening to try to keep Drosophila from entering the crop, but they also help to enclose any natural enemies that might be released into the enclosed area. So some of these studies in Italy and Mexico have shown or been able to demonstrate that releasing augmentively these pupal parasites has been able to significantly reduce populations and damage due to spotted-winged Drosophila. But a lot more studies are needed to study their timing and the numbers of individuals that need to be released and so on in different crops. Some of the studies that are ongoing in California right now are addressing this in California situations. For example, in berry fields adjacent to riparian habitat, the riparian habitat serves as a tremendous reservoir for spotted-winged Drosophila populations that increase dramatically into the summer and fall when there are very, very high numbers. And any releases of natural enemies need to begin to be effective early on before those numbers can increase. So far, the releases of Trichoprea drosophila and Pachycropoides vendiniae in California have shown mixed results. With Trichoprea, some positive response of release in covered crops at 3 study sites. Not such great results with Pachycropoides. With over 60,000 parasitoids have been released. With augmentative studies, a lot more work that needs to be done with these. Now, there are also possibilities of working with predators. A number of predators have been found attacking spotted wing Drosophila in various survey studies, and these show some of these natural enemies. Predators, there are several ways of using them that are being investigated. Many of the approaches are conservation related, trying to reduce the use of broad spectrum pesticides and rely on organic pesticides, providing the predators with conditions that give them shelter and food or alternate food. And then some studies that have looked at the possibility of augmenting predators. And there are several predators that feed on spotted wing Drosophila, such as pirate bugs and rove beetles, lacewing larvae that can be purchased, but their cost is relatively high, quite high, and it's yet to be determined whether or not they can be effective economically. However, the impacts of predators in some of these field studies are shown to be relatively significant. So the predators have been shown to prey on and remove significant proportions of the spotted wing Drosophila pupae and larvae from plants and on the soil. So they could be a very important part of an integrated pest management scheme to conserve these predators, in addition to the possible use of parasitoids and other management techniques. We certainly don't want to discourage the use of predators or their populations. There have also been some efforts made to look at the entomopathogens with fungi, and there have been several tests made with widely available fungi such as Beauveria, Escherichia, and Metarhizium. In lab tests, the results vary widely. Using entomopathogens in the field present a lot of challenges. Entomopathogens typically have low residual activity, and they degrade very fast, so methods have to be taken in order to protect them in some way so that their residual activity extends long enough for them to be able to actually infect significant numbers of flies and larvae in the field. One of the techniques that has been suggested is an auto-inoculation device where fungal spores are put out in structures that protect them from ultraviolet and from rain, that allow flies to enter and then pick up the fungal spores and then leave and then reinfect other flies in the population, in the crops and in the reservoir population. There have also been studies shown that nematodes can be effective at attacking spotted wing Drosophila populations, and several species have been tested. Nematodes require moist environments, but they're typically applied as soil drenches, sometimes on foliage, but since a large part of the spotted wing Drosophila population resides in the soil, this could be an important way of infecting a lot of the pupae and a lot of the adults as they emerge from pupae. A little bit of work on viruses, not so much. Several viruses have been found. Same thing with bacteria. Not a lot of positive results from bacteria that have been tested, mostly Bacillus thuringiensis. So in summary, we have generally in the invaded areas, no effective native arthropod natural enemies. We do have good natural enemies identified from the native range in Asia, in particular Ganaspis brasiliensis, which we hope to begin releasing more widely in North America. There are good possibilities for augmenting field populations of the pupal populations and possibly predators as well. And further work might show that entomopathogens have utility. And of course, as is always the case, there's a lot of opportunities for further studies. that will improve our knowledge of the ecology and the behavior of the natural enemies, that will help us develop and improve the rearing techniques to be able to rear and release large numbers of natural enemies effectively and more inexpensively, conserve their populations in the field, and in particular, find ways of integrating all these different biocontrol tactics into ecologically suitable IPM programs. And with that, I'll end this presentation, and just with a list of, in particular, this is the team that works primarily on classical biocontrol aspects of spotted wing Drosophila. Of course, there are many, many other people that have been involved in the research that I've talked about, way too many to mention. And with that, if there are time for any questions, do you want to deal with the questions now or during the break?
Maybe we can have one short question if you have any, and also the after the all speakers, we can have more additional time for discussion. Anybody
Okay.
has the question for the Kim's? Yep.
Yes, I can.
Okay. You suggest several biological control agents, and when you apply IPM, how we can choose specific biological control agent among so many biological factors, and for example, BT, or you said parasitoid, and depending on situation. But what is the main criterion to choose a specific biological control agent?
Well, I think with any IPM program, it has to be tailored to the specific situation of the crop and the location. The researchers or the extension agents and the growers in the affected crop need to have a good idea of what the local conditions are, the presence in populations of locally occurring natural enemies that could be utilized. Whether they're, if the grower wants to be able to use enclosed crops, that introduces possibilities. If the grower is not using an enclosure to exclude spotted wing Drosophila, then that makes different series of choices available. In North America, where we don't have the naturally occurring agents that are already active in wild populations, we're going to be relying more heavily on classical biocontrol agents with the Asian parasitoids. In Asia, where you already have these natural enemies present and active, then you want to look more strongly at what can be done to conserve and promote your existing natural enemies and how to fit them into with other IPM strategies. So every situation is somewhat unique and has to be constructed individually, I think.
Okay. Yes, thank you for the question and answer. And then I think we have to move on the next presentation because it's time. Let me introduce the second. Thank you very much, Dr. Kim Holmer.
My pleasure.
Yeah, we will have more questions using the break time or after the symposium. Okay. Yeah. And then let me introduce the second speakers from US, Dr. Julie Gold. She's working in the USDA Front Protection and Quarantine, Massachusetts. She's talking about the biological control of the emerald ash borer, progress and future research. Julie, are you ready?
Yes, I am. I'll start sharing my screen.
Yes. You can use the 30 minutes, probably 20 minutes presentation and 10 minutes discussion will be nice. Yes. Yeah,
oh no, probably not.
Okay.
We'll see.
Yeah.
Okay. Can you see my screen? Okay.
Yes.
Yes.
Good.
Okay. Great. So I've been working on biological control of emerald ash borer for almost 20 years now. It doesn't seem like that long, but it's been a long time and we've had a lot of success. So I'm going to tell you about some of the history of the whole project and the successes that we've had. The emerald ash borer was found in 2002 feeding on ash in southwestern Michigan. And it is thought to have been introduced to North America at least 10 years prior, so probably around 1990. And it probably came to the United States in solid wood packing material. These are pallets that bring a lot of heavy equipment to our country, and we think that the insect came in the solid wood packing material. And this is a very beautiful beetle. The buprested beetles are called jewel beetles because they're so pretty. And the emerald ash borer can have a 1-year life cycle, or where it's very cold in the north, it can take 2 years to develop. But generally, it has a typical life history. It emerges in the spring. The adults feed on foliage, but that's not a problem. The adults don't cause any damage. The insects will then mate and lay eggs in the crevices of ash bark. And these are some of the eggs that are very flat. They're laid between the layers of the bark. And it's the larvae that are damaging. The larvae feed under the bark, and they destroy the phloem of the tree and cause a lot of damage. If they have a 1-year life cycle, they then overwinter in the wood, a little deeper into the tree. In the spring, they'll become pupae and emerge as adults. As I said, it's the larvae that cause the damage. They produce these galleries under the bark, and eventually the bark will just fall off the tree because there's nothing left to hold the bark on. And they can kill the branches of the tree, and eventually they kill the entire tree. And in our country, hundreds of millions of trees have died, and there's been huge economic and environmental losses due to this very devastating pest. You can see here, these are all dead ash trees. And it has considerable ecological damage. Ash in general is only about 2% of our trees, but in riparian areas, in wetlands, where there's things like rivers, you can see this river running through. Where there's rivers and wetlands, ash is a very, very important species and can be most of the trees in an area. And you can see how many dead trees there are here. It's very important economically. In the United States, ash is a very popular street tree. And you can see what happened in this neighborhood. We had beautiful leafy trees, and then emerald ash borer came in and all the trees died. Ash is also a useful wood product. I know that they play baseball in Korea like they do in the United States, and baseball bats are made out of ash wood. It's a very good wood for baseball bats. Our native peoples also use ash bark to weave these beautiful baskets, and it's very important to the native cultures in our country. And when emerald ash borer first came into our country, we tried mechanical control. They set up these huge areas where they would bring in trees and chip them into tiny pieces. And they tried regulating, telling people don't move firewood, don't move ash trees. And they tried some chemical control. But the problem is that emerald ash borer is a very strong flyer. It can go very far, but even more important is artificial spread. The nearby, where Kim and Shinking live, is nearby to a state called Maryland. A nursery shipped infested nursery stock. These are trees in pots shipped from Michigan to Maryland. All of a sudden, Maryland had this pest. People also in our country like to go camping. They move firewood and they move logs. So we have not been able to control emerald ash borer. 20 some years after it was found in this country, it is now in 35 states, and pretty much in every state. We have a big range of mountains right here called the Rocky Mountains, and it's basically pretty much east of those mountains. So very early on, it became obvious to many of us that we were not going to control this pest. So we turned to classical biological control, which is the importation of specialist natural enemies for the sustained control of a previously introduced pest. And there have been some very striking examples of success. The Vidalia beetle, this little beautiful beetle, really controlled cottony cushion scale in our citrus orchards in California in the 1800s. Classical biocontrol can be very specific, unlike some pesticides. It's self-sustaining. The insects reproduce on their own without human intervention. In the long run, it can be very cost-effective, and it can be extremely effective. So we decided to pursue biocontrol of the emerald ash borer. And as Kim mentioned, one of the first things you want to do is look and see if there are any natural enemies that are attacking the pest in the new range. And we did a survey and we found almost nothing. We found a few parasitoids that attacked EAB, but the parasitism levels were less than 1%. So we turned to China and we started surveying in the northeastern parts of China where emerald ash borer could be found. Needless to say, emerald ash borer is not very common in China because it's under control. The trees are resistant to emerald ash borer in China, and we actually this is right here. These are American ash trees that were planted in China, and they were hit by emerald ash borer, and that's where we found some of these parasites. So we found 3 promising agents. One of them is a quite small eulophid parasitoid called Tetrasticus planopenisi. It's gregarious in that each female lays multiple eggs in one emerald ash borer, and you can see inside here, you can see where the arrow is pointing. This is a larva of Tetrasticus inside an emerald ash borer. These larvae develop and they eventually will burst out of the EAB larva in their gallery, and then they become adults and they repeat the cycle. And they do this several times a year. These things have 3 or 4 generations per year. So that's very promising. Spaethius agrilli is a braconid. It's a larger wasp with a longer ovipositor and can go through thicker ash bark. It's also gregarious in that it lays multiple eggs per EAB, but they're laid on the outside instead of the inside. And you can see the larvae feeding here, and they form cocoons. And again, they do this several times per year, which is a good attribute for biocontrol. And then there's this tiny little wasp called Oobius agrilli, which attacks the eggs. And thank goodness I didn't have to work on this because I couldn't see it. It's very, very tiny. But it has 2 generations per year on emerald ash borer eggs. So, as Kim mentioned in his talk, in our country, the regulations are very strict on making sure that there is safety, that the insects that you want to import are not going to attack a native insect. So we had to do host specificity testing, and we found that all 3 of the parasitoids attack significantly more emerald ash borer or EAB than non-target insects. We did some olfactometer work and found that Spaethius agrilli was attacked to ash leaves, but no other leaves. And we actually did surveys in China looking for these parasitoids, and we found over 2,000 larvae in the same genus as emerald ash borer and found none of these larval parasitoids. So in 2007, we were given permission and got a permit from the government to start releasing these parasites. Well, one of the big problems is you have to rear these things in really large numbers to have a biocontrol program. And we're talking about rearing an insect that lives under the bark of a tree. So how do you do that? Well, how we do it is we collect logs in the winter that contain emerald ash borer pupae, and we bring them inside and warm them up. And you can see here that there are emerald ash borer adults that are being collected using this method. We then get ash foliage. We sometimes have it shipped from Florida and Hawaii, shipped across the country. We ship this foliage. We rear the emerald ash borers, and they lay eggs. You see these lids here? They lay eggs under this screening on coffee filters. So we get the emerald ash borer eggs, and then we put them on logs using parafilm, like you all use in your lab. And we keep these logs wet, so the logs are moist, and the EAB develop normally, just like they would in a tree out in the woods, but they're growing in the lab. We then give these, either the pieces of paper with the eggs, we give them to the egg parasitoid, or we put the logs with larvae with the parasitoids, and we successfully rear them. So that was a big step. And we rear, even though this is a really hard system, we rear over 1 million parasitoids per year for this project, which is quite a feat. So we've currently released parasitoids in 25 of the 35 infested states. Even clear out here, this is our state of Colorado, and we have a satellite infestation, and we've released out here, and we've released throughout our country. And so we've done all these releases. Well, you have to ask the question, is it doing any good? And so this is Jian Duan, who works with Kim and Shin King, and he's peeling the tree. He's taking a draw knife and peeling the tree to find larvae. And we also can sift the bark to find emerald ash borer eggs to see if they're parasitized by Owobius. We can put logs or bark in tubes and collect emerging adults. And we've developed these yellow pan traps. Parasitic insects are very attracted to yellow. And so we put some preservative in these little bowls, and we collect a lot of parasites this way. And it's actually very easy and very cheap. So we use this technique a lot. And we have found that Tetrasticus planopenisi has been recovered in 17 states. Owobius agrilli has been found in 15 states. And both of those species are persisting and dispersing throughout the north of our country. But the problem was Spathius agrilli. We would find it the year after release, and then we would never see it again. So that was a real mystery. You know, why weren't we finding it? Well, climate matching is an important tool for biocontrol. And when we did the climate matching between Tianjin, China, where we found Spathius agrilli, and the climate in the United States, we found that Spathius agrilli was really matched better to the middle and southern part of the United States. And we were releasing up here in the gray area where it's not a good climate match. So we decided that we would only release Spathius agrilli in the south, but we had a problem. Tetrasticus is really, really tiny, and it can only parasitize in very small ash trees. So what happens when the emerald ash borer gets into big trees? We need protection. Well, and we need something like Spathius agrilli. Fortunately, we found Spathius galeni. Kim and Shin King's colleague Jian Duan worked with people in Russia, in the Russian Far East, and found Spathius galeni, which has a very long ovipositor, and it can go through very thick ash bark. And look at the climate matching. It's much better matched to the northern part of the United States. We found that this species was quite specific, so it wasn't going to attack our natives. And we got a release. We had a release petition. It was approved, and we started releasing this insect in 2015. And Spathius galeni has now been recovered in 8 states. So it's moving up there, and we think it can be really successful. And one of the things that anybody who works with biocontrol for a while knows is that parasitoids and their hosts have very complicated life cycles, and they need to be synchronous. And we found that Spatheus galeni and Tetrasticus planopenisi emerge really early in the spring, and they have to have larvae to attack. And they have to be emerald ash borer that have a 2-year life cycle. If all the emerald ash borer are in a 1-year life cycle, there's nothing for them and they die out. And so we did a really large survey of the United States, and we surveyed 92 sites over 2 years, and we developed this map which predicts the probability of emerald ash borer having a 2-year life cycle, which we need. And so if you look at and the red dots show where Tetrasticus planopenisi has established, and you'll see that it's much more likely to establish in the north. The further south you go, the warmer it gets, the emerald ash borer develops really, really quickly, and Tetrasticus cannot survive. In fact, we got in this yellow area, light yellow area in the bottom of our country, we saw no establishment of Tetrasticus. So this model has actually helped us determine where to release it so that we're not wasting money releasing this parasitoid where it can't establish. So this model has been very, very helpful to our program. So we know where we know we've got an establishment, we can find these parasitoids, but will they do any good to slow down the population growth? Just because they establish doesn't mean that they're going to affect the population. The data I'm showing are from Michigan, and this was a study by Jian Duan. And what he found is that this is what happened to the emerald ash borer population. In 2009, hi, I wonder what happened.
Oh, I think it's because about the network system or something. I don't know, but sorry for this confusion, and you will have like additional 10 minutes. I don't know, but yeah. So
Okay.
we'll wait. Other like participants will come. So I'm here.
Oh, did everybody get kicked off?
Yeah. The rooms rooms exploded.
Oh dear.
Yeah. So we have Wang here, Hannah here, Julie here, Kim is here.
Yes, please, Julie, upload your file again. Excuse me.
Yes.
Yes, please use your the PPT file again. And then,
Oh, share my screen again?
yes, that's right. Somebody in the conference, please shut down.
I saw something happen to my computer.
Okay. Yes, you can continue.
Okay. So emerald ash borer, the outbreak peaked in 2009 at about 45 larvae per meter squared. Within 2015, within that 1 year, they had moved, the parasitoids were found 2 kilometers away. Remember I showed you those yellow pan traps? That's what we were doing. We put yellow pan traps all over these cities. So they moved 2 kilometers in 1 year. And this graph shows the buildup of the parasitoids year by year. We had a release plot and a control plot. And by 2017, the parasitoids had invaded our control plot. So this is Tetrasticus planopenisi. It was establishing, it moved very, very quickly. Spatheus galeni we released later. It's also establishing and spreading. We're also finding Aobius agrilli. And by 2019, emerald ash borer densities were very, very low. So you know what we did? We stopped treating with insecticide. And now we're continuing to monitor these populations because we think that these biocontrol agents are going to be able to keep the emerald ash borer down and that these large trees will continue to be healthy. This year, we are actually moving this concept into natural forests. The study we just did was in a city, and we're trying to do this also in natural forests. One of the things about ash is that their seeds are very short-lived. Some tree species have seeds that live in the forest floor. Ash does not. Their seeds do not live long. And so we are treating, again, big trees, trees that are reproductive. And we're doing this in black ash because it's very important economically and culturally, and it's very, very susceptible to emerald ash borer. So in summary, our results are very promising so far for biocontrol of emerald ash borer. Tetrasticus planopenisi and Aobius agrilli are establishing and they're dispersing. We are continuing to release Spatheus agrilli in the south and Spatheus galeni in the north. EAB in young trees are suffering very high mortality from woodpeckers and Tetrasticus. And this is our next generation of ash, which we think we are going to be able to protect. And Tetrasticus and Spatheus galeni are establishing and spreading in urban areas and can contribute to integrated pest management. And this project really illustrates well that biocontrol is very complicated. It requires time, it requires patience, and it requires a lot of research. But when it works, it can be very successful. So that is it. Thank you for your attention. I enjoyed this symposium.
Yes, thank you very much, Julie Gold. It was very interesting presentation. And then, sorry about interruption. Probably we had at about 10 minutes interruptions. That's,
Yeah.
I will keep in mind for the time. Okay, so if some participants have questions to the Julie, please. You can have your microphone used, or you can give a message by the chattings. Anyone has the question?
Julie,
Yes,
thank you for your presentation.
you're very welcome.
Yeah. How do people monitor EAB in the stage? Using traps or?
Well, there is a trap. It's called a green funnel trap. I don't know if you know what a funnel is. A funnel is what you put oil in your car in. There's stacks of funnels, and they're green, and it represents like a tree. It looks like a tree, and it has a lure that's attractive to emerald ash borers. So they do have adult traps that they put out. The other thing people actually do is emerald ash borer really, really likes stressed trees. So people will go out and cut the bark of a tree to make the tree smell really good. And the emerald ash borer will come in and then they'll peel the tree. They call it a trap tree.
Okay, thank you.
Yep.
Anyone else? Yeah, personally, I have a question about the host range of the emerald ash borer in US, because I was thinking, the Fraxinus, the ash tree is only the host, but looking at, the presentation, I am just so the betula, like, the birch, is it host range or just checking somebody checking the skins of the birch? What is
birch is absolutely not a host for emerald ash borer. Okay. We have a tree called white fringe tree that I don't know. It's also an Oleaceae. It's closely related, and it's a tree that's kind of in the southern United States. And emerald ash borer will attack that, but not birch. We have an insect in the genus Agrilus called the bronze birch borer, and bronze birch borer attacks birch, but not emerald ash borer.
Okay, good. Yep. Anyone else has a question additionally? We have probably one questions we can have. Yes.
Julie, just a quick question.
Yes.
You mentioned about protecting the next generation of ash trees, right?
Yes.
So, do people or government actively replant ash trees in the area where they are affected?
No, no, because what happens is that emerald ash borer cannot attack a tree that's smaller than about 2.5 centimeters. So when emerald ash borer comes in and kills all the big trees, there are still small trees there that it cannot attack that are too small, and they come up. So no, there's no planting.
Okay, that makes sense. Thank you.
And what is the origin of the true parasitoid, like the genus Spatius species you're releasing to the the nature, and then where is the origin of the true species of the Spatius?
It's interesting because my Chinese collaborators also say Spatius. they have the same pronunciation. Spathius agrilli is from Tianjin, China, and Spathius galeni is from Vladivostok, Russia. So much farther north, and they are climatically adapted to different parts of our country. The two different species.
Okay.
They have Spathius. One of the Spathius species was also found in Korea. And I can't remember which one. I can't remember which species, but we also found it in Korea.
Okay. Thank you very much.
You're very welcome.
Yes. Okay, and then we are moving to the next presentation. May I introduce the next speaker, Seung-hyun Lee from Seoul National University, Korea. We were talking about the Asian longhorn beetle in Korea and population genetics and biological control. Seung-hyun, you're ready. Or you use the microphone. Open the microphone. Use the microphone, yes.
Hi. So you hear me?
Yes.
Okay. So, yeah, let's get started. Hi, I'm Seung-hyun Lee from Seoul National University, Korea, and the title of my talk today is Asian longhorn beetles in Korea, population genetics and biological control. And there are two main subjects of my talk. One is about the brief introduction of Korean.
Seung-hyun, probably you use two microphones. Probably you have to turn off your the cellular phone. Yes.
Hello. Is it better?
Yeah.
Okay. Sorry. So, yeah, there are two subjects of my talk. One is about the brief introduction or brief overview of Korean Asian longhorn beetles and their population genetic structures. And the other part is about my ongoing effort on finding natural enemy of Asian longhorn beetle with ARS. especially focusing on finding egg parasitoids. So let's get started. I believe all of you will know this beetle. So let me be very brief in the introduction part. Asian lowland beetle, also known as ALB, is one of the most notorious invasive pests in the world. They're really highly polyphagous, damaging more than 36 plant families, and at the same time, they attacked mostly living trees. And their native range is China and Korean Peninsula, and they have invaded to North America and Europe. But in both countries of its origin, namely China and in Korea, the ALB outbreak situation has been quite different. China went through many ALB outbreaks. After the first outbreak record from 1980s, due to this reforestation policy that makes the situation much more worse. And in contrast, ALB outbreak has never been reported in South Korea until recently. Korean ALB has been only found in the deep forest of the northeastern part of the mountains, and their population level is very low in these areas. So if you collect one Asian lowland beetles in the forest, that means you're super lucky in that day. And they mostly found on the Acer species, especially Acer mono. So it's really weird that we didn't have ALB outbreak here in South Korea, but China has many outbreaks. Some really vaguely guessed that there are some reasons for non-outbreak in South Korea, but some say ALB may outbreak in the future. But it really happens? It did happen, and we have ALB outbreak here. Very recently, not recently, in 2014, we first found ALB outbreaks in South Korea. We found them in Incheon, Seoul, the western urban cities, and Ulsan and Busan, the southern cities. And these cities are two of the largest harbor cities in South Korea and its adjacent cities. So it's very suspicious that these notorious beetles found in these kind of harbor cities. But we cannot jump into the conclusion that they are invasive because we already have ALBs here, right? We already have the native populations here. So they can be either invasive populations or they're just moved from the native range, especially namely the northeastern forest. They moved from the forest by human acts or something. So the first thing I did was to figure out the current distribution of this outbreak-like populations. So I started social media, literally, like Facebook, Instagram, and some blogs. I searched it with every possible keywords, like from Asian lowland beetle, Chinese citrus lowland beetles, Cerambycids, Dynastis, Licanids, even roaches. Because many people uploaded this species online because they think these species are super rare. So it did help for my research. And in the past 5 years, I have collected more than 200 individuals from 18 different locations throughout the whole country. So we can easily divide this South Korean population into 3 subgroups with geographical, habitat, and I would say observed host plants. They are western urban populations and southern urban populations and northeastern forest populations. So the northeastern forest populations named after its province name, Gangwon. So I call them KW subgroups. They all found in these kind of natural forests. And as I said, their population level is low and very difficult to collect. And they mostly found on the Acer species. And the western urban populations were named after 2 big cities here, Incheon and Seoul. So I call them IS population. You can see the difference of habitat type. They are all found in the urban area, and they're mostly found on the horse chestnut and rarely Salix or Acer saccharinum, maybe. And the southern urban populations are the most destructive population subgroups in South Korea. They mostly found on the Salix species and rarely on ulmus. And they were found alongside the river, like this, here, here, and here. So these populations are really big, so we can easily collect like 100 individuals a day. And moreover, the native populations and the IS populations look somewhat different. I didn't measure it in the scientific fashion, but yeah, maybe Wang will feel in the same way. They look somehow different. So the newly found populations are very suspicious to be invasive because of their outbreak area and their biological features and the difference in appearance. So we made a hypothesis they are invaded from other region, namely from China. So we conducted molecular experiments, population genetics. So we used all the samples we collected from like 4 or 5 years of collection, field survey. And this is the process of molecular work. This is the process of the molecular work, collect the specimens and we extract the muscles and make the vouchers as a dried specimen. And we extract the DNA. We used 800 base pair of CO1 and conducted molecular analysis. Actually, my paper contains like more data set. I have like global data set with Chinese populations and North American populations in Europe. And I added more analysis into my paper, like DNASP or DIY ABC, like that. But I want to make it very simple here. So I used only Korean samples. So this is the phylogenetic tree, and neighbor joining and maximum likelihood tree have the same topology. And you can see the clear clustering according to the geographical subgroup. You can see here. And this one is the haplotype diversity of each location. And the right one is, the haplotype network. We found the moderate genetic diversity in the putative native range, KW. See, you can see that proportion of the haplotypes here, and there are many haplotypes here. And what's interesting is more than 50 individuals collected in 4 different locations and 4 different years, they have all the identical haplotypes in the IS subgroup. And BU, we collected more than 120 samples from here. They only have 2 haplotypes, and that are very distinct each other. So we think this IS populations and BU populations are really a good example of founder effect that in many cases observed in invasive populations. So these are the summary of molecular analysis, and we can conclude by like 4 independent evidences that these populations are like possibly invasive. So they are invaded here and here independently 2 times, and yeah, the domestic hypothesis can be rejected. So now you got the situation in South Korea. We are no longer safe place from Asian long-horned beetle outbreak. And now let's talk about the natural enemy things. So this is, I think Dr. Wang will present this more in his presentation. This is what they did. Like they induced oviposition inside. They made a sentinel log with oviposition, so the eggs are in that sentinel logs. They place those sentinel logs in the natural condition, and they lure the parasitoids. But I thought it's more important to induce this oviposition outside, because there will be some chemical volatiles or like physical vibration that those kind of signals can allure the parasitoids, I think. So I developed a simple new trap that allows small parasitoids can get in, but ALB cannot get out. So yeah, and this costs like $7 or $8, so it's really cheap and effective. As I told, these outbreak area, we can easily find like 100 individuals a day. So I collected females from these outbreak area, so like 50, 100 females, and launch the new version of trap into the native site. So I think it's super, super important to keep ALBs in the cage safely. So I did everything on my own. So this is the photograph I launching my traps. And all the traps are safely maintained in last 2 years. No breaks, no escape of Asian lowland beetle. So it's quite cheap and safe trap, I think. And we collected, we launched the Sentinel logs and collected twice in 2019. And this year we did this for 3 times. And yes, we got the egg parasitoids of Asian lowland beetle. I think this is one of the most important contribution to the invasive entomology community I have made. Yeah, this is the photograph. This is the ALB egg, and this is the egg parasitoid. This is another one. We only collected 2 individuals in 2019. So we tried to make it to the adults. So we brought them and reared in that laboratory condition, but we failed. The one died in the larval stage, and this is the second one, died during pupation. So yeah, this is a dead pupa. So we tried to get the CO1 barcode sequence using many universal primers of these parasitoids. And this is the result. I was surprised that most of the sequence were ALB itself. So we extracted the DNA from the whole body. So if anyone knows what happens here, just please let me know after my talk. But anyhow, only one primer, SLAP and LEP primer stat, successfully provide out a CO1 barcode of parasitoids. And through the blast, we found that this species is most close to Aprostocetus species. That genus contains the citrus lowland beetle parasitoids, egg parasitoids. So yeah, we think it's really plausible. But yeah, still like really different from any public sequence available. So if you have any unpublished sequence of these genus or this family, so please let me know. And if you know somebody who may have this, please let us know, because the identification of this parasitoids is the most important part of the publication. So yeah. So we found 1 egg parasitoids of Asian lowland beetle, and yeah, this is not a big deal. The larval parasitoids from Acer mono, and we through DNA barcode and morphological identification, we identify this species as Spathius in US accent and Spathius in Korean accent maybe. And we also found some non-parasitic oviposition pit dwellers. So they live inside the oviposition pit, but they're not attacking the egg actually. So they are. We also found out this through DNA barcoding. So one is Drosophilidae and one is Acidomids. So yeah, this is what I prepared for today. So take-home lessons, 2 lessons, 3 populations in South Korea, 3 subgroups in South Korea, 2 are invasive and 1 is native. And yes, we have the egg parasitoids. Thank you. 교수님, you've got to use your microphone. Oh. 교수님, 네. maybe I've got to visit his office.
Oh, sorry. It's not working. Sorry about. I have to open my microphone. Yes, he's coming to my office now. Didn't I open this? Okay. Please wait 5 seconds. He will return. Yes. Now for the question for about 5 minutes we can have. Yes. Anybody has a question?
I have one question.
Yes.
So, what the genetic differences between two invasive groups? So, do you know the differences were? And I want to know too. So, whether they are serial introductions from some other places or so different independent introductions between them. Do you know that?
The two subgroup have these two circles are from BU area, the southern subgroups, and this one is the Western urban subgroup. So they're actually distinct, genetically distinct. Yeah, but two are more closer than these native populations, but that doesn't tell anything about like where these came from. And the more important thing here is the Chinese populations are genetically mixed because of the domestic reforestation. So in this part, move here, and so that's like total mess of like, I would say like melting pot of the Asian longhorn beetle. So we cannot trace the origin of this population because already mixed genetically in the China. So yeah.
So after this study, did you try to compare them with the Chinese population?
In my paper, I already have Chinese samples and North American samples and European samples. But this is the presentation, so I want to make it simple.
Okay, I understand.
Yes, we can have one more question.
Can I make a quick question?
Yep.
Yes, Tu-hyung.
I'm so glad to hear that you did not lose any of your sentinel traps in the field.
Oh yeah.
So I think my question is about the location number 12 in this map.
Okay.
Do you think that population actually moved from Busan or Ulsan area as an invasive population, or do you think they are just native population in Jeonju area?
No, I believe this is surely invasive because I think the most ALB populations from here to here, they won't move by themselves. I believe they move by like planting trees or like human things. So yeah, it's possible. That's why we only found them in the big cities, I think.
Okay, that's good to know. Thanks.
Okay, thank you, Sung-hyun. Let's move to the next talk. The next speakers from USDA ARS, Dr. Jingheng Wang is talking about progress toward biological control of Asian longhorn beetle. Please, Jingheng.
Okay. Okay, thank you, Professor Lee. Good morning to everyone in South Korea and good evening to my colleague here in the US. And so I'm going to talk about progress towards biological control of Asian longhorn beetle. Let me first introduce my co-authors. So they are Dr. Xiaoyi Wang from Chinese Academy of Forestry in Beijing, China, and Dr. Junior Good from USDA office in Massachusetts, USA, and also Dr. Songyang Li and Professor Songhao Li from Seoul National University. So we are naturally collaborating on the survey of the parasitoids in China and South Korea. And this is the outline of my talk. I will just give you a very brief introduction about the beetle, as Sun Yang already talked about this. And then I'm going to focus on the three sections. First, I will talk about what kind of parasitoids attacking waterborne and Thermin-bicid. This is the family of this beetle belongs to. And second, I will talk about classical biocontrol prospectus. And this will largely based on what kind of parasitoids we have found so far in China and South Korea. And then I will talk about novel association biocontrol prospectus. So this will based on some evaluation of the parasitoids that are native to North American, but are also capable of attacking this exotic beetle through novel association. And then finally, I will give a brief discussion about the future of this project. So let me start with a brief introduction of Asian long-haul beetle. I will just go short on ALB. So ALB is native to China and Korea. And this is, as Sun Yang mentioned, is a very highly prolific beetle. And there's more than 100 tree species have been recorded as hosts for this beetle. And although it prefers to attack Asana, like Popinus, Stachys, hard water trees. The beetle was first detected in 1996 in the US, in New York City area. Was later on also found in New Jersey and Massachusetts, Illinois, Ohio, and the US states, but also in 2003 was found in Ontario, Canada, and was most recently was found in South Carolina. It was this year. But in US, this the target population are very small, isolated population, and so in this, this beetle has been targeted quite extensively for education. Most of this the target population have been successfully eradicated, except some population probably still present in Massachusetts, Ohio, and also in New York City. But the beetle was also found since 2001 in more than 10 European countries. I think the population also was still present in Italy and several other countries in Europe. So I think also we are very successful to eradicate some invasive population, but still I think we are facing a constant threat of the invasion and established. So if this beetle became widely established in the US, then I think biocontrol, particularly by using parasitoids, could be a viable option for the area-wide control of this invasive species. And indeed, the biocontrol of AOB in China have been quite successful. And there's 2 genus parasitoids that have been mass-produced and released for the control of AOB in China. Or this beetle in China is not regarded as pest in nature forest, but it could be pest in some urban area, particularly in those plant forest. So those 2 parasitoids are Pastax have forest and also Scodamus squai. But unfortunately, the 2 parasitoids are very generous parasitoids. They're also being used for control the other beetles. And so we are not able to introduce them into the US as classical biocontrol agents because of the concern of non-target impact. So we are looking specifically, we're looking at the parasitoid not only effective, but must be host specific. And so when I started working on this project, I have done some nature review first, and I look at what kind of parasitoids attacking this specific host community and try to find, maybe give an idea of what kind of parasitoids we are looking for. So if you look at the host feeding echo, the beetle, they spend all the immature life hiding in the water. Like typically, adult beetle lay egg in the bark, and then young larvae developing from phloem, and then burrow into deeper in the xylem, also pupa inside in the water. So they can't protect from certain natural enemies, but maybe not from specialized parasitoids. We know some parasitoids, for example, some ecomodule, they have very implacable non-type opods that are able to draw into the sick bark and reach to attack the beetle larvae or pupae. So I think the parasitoids could be the first choice for biocontrol. And so there's totally about 575 parasitoid species have been recorded worldwide, associated with the waterborne Thermin-bicid. Most of them are novel parasitoids belonging to 2 families, the majority belonging to two families, Baconidae and Ecomolidae. These two families account almost 80% of novel pestoids. The egg pestoids are less diverse and coliform 5 family, but the majority of them belong to Insectividae and Eulofemidae. So these two families also account for over 80% of novel egg pestoids. Well, pure pestoids are less diverse. There are only total about 27 species called from 30 different families. Also, we found egg pestoids seem to be more host-specific compared with novel or pure pestoid in terms of the number of host species called pure pestoid species, as shown on the top graph. But this is probably due to the fact that all the egg pestoids are indoor pestoids, and the majority of the novel or pure pestoids are ectopestoids, as shown on the bottom graph. And so we know ectopestoids tend to be more generous because they lay egg outside of the host. And so they don't need necessarily to overcome the interior physiological defense by host. So they tend to be more capable of adapting to the novel hosts. So these are some general characteristics of the pestoids attacking this specific host community. And so this probably gives us some idea, what we are looking for. I think we're probably looking for some pestoids from attacking egg or young larvae from the perspective of classical bioconjugate because they tend to be more specific. But also it tells us, some native or resident pestoids to the US may be, some novel pests because most of them are ectopests may be able to adapt to attack this exotic beetle. So I'm going to talk about the two different perspectives about using the exotic pestoids and also the indigenous pestoids for the biocontrol of AOB. So let me start with the classical biocontrol perspective about using native pestoids from Asia. This work started by survey of AOB pestoids in China in 2015. This work actually started by Dr. Junior Gould in collaborating with Dr. Xiaoyi Wang in China. So far, I started on this project, working on this project 2 years ago. So far we have surveyed in 5 different locations in China, from Jining, Beijing, and Shanghai, and this is Guangzhou. And this year we add on Guizhou. So these 5 locations have been surveyed. And so the methodology we're using the very similar in China and South Korea, as Song Yang just talked about the methodology, very similar. Basically, we collect a lot of beetle in June or little May, depending on when the beetle start emerging in the field. And then we using this central logs that are normally using on like a maple or willow. And this log were infested with adult beetle eggs and also was young larvae and securing this metal screen cages and then launch this cage in the field. So after like 3 or 4 weeks, we collect back those logs and dissect them to recover the AOB eggs or larvae. Sometimes we found the pestoids larvae or cocoon and then lure them to adults for identification. Just quickly show you some photos of the field work. So this here are the 2 students, Zhang Xin and Li Fei from Dr. Xiaoyi Wang's lab, and this is colleague Dr. Gao Liangming. And so I was there last summer to join and then for the survey in China. And so sometimes we just doing the dissection of logs in the field, but also sometimes we doing dissection in the hotel room. That was the easy job, just peel off the bar and then you will see this AOB larvae or eggs. And this is how we lure the larvae collected back from field. So basically we just cut a piece of the bar open and then make a glove and insert the larvae and then using paraffin to wrap the flappers. So far we have found at least 12 pestoid species from China. This from the central logs. So they include 3 Baconidae. I don't want to overwhelm you with the name, just the family name. And so there's 1 Ecomolidae and 5 Tomonids, and I don't have a picture for the last one. and 2 eupemis and wing Eurotomida, and also wing. This is the common species I mentioned at the beginning. And so the parallelism from this central log are kind of low, but varies among the 3 different locations. So very similar between Shanghai and Beijing location, but very, very low in Jining, Jining. And so it's range about 4.7 to 11.16, and very similar different years. This data from 2015 to 2018. As this table just show you what did the parallelism by individual pest toys look like, and also was very similar among different years. This is the 2018, 2019. But it's interesting to note that some pest toys like the 3 I highlighted here in yellow are collected from both locations in Beijing and Shanghai. And we know Beijing and Shanghai is quite different. The weather is quite different. It's about 700 miles away. But this is interesting because this 3 species seems to have wide geographic range. So this is some good attribute for the pest biocontrol. So far we have imported 4 species into our lab for learning. And we have limited success with learning of the 3 species. Largely, we only receive very few specimens, like only 3-5 females. And so this is not very, this is kind of hard to learn them. But the one species, despite this, and Apophylla, we receive more cocoon, and this species has been learned so far for 6 generations in our lab. Also, this number is still very low, and I'm hoping to build the higher number maybe for some evaluation next year. And so this is the Song Yang. He did talk about his survey in South Korea. I was there last year with him. So now I want to talk about novel association biocontrol perspectives. And this is the potential using some indigenous pest toys from North America. And this work also started by a survey of indigenous pest toys of waterborne salmon bicysts in North America. And so we just cut down this hardwood tree in the Delaware forest, and we laid these balls on the ground from spring until fall, and then we just let the beetles and pests colonize on these balls, and then we collect them back in the fall and store them in those solid tubers. And so mostly these beetles or pests start emerging in the spring of the following year. And so far, we have done quite extensive survey. We sampled over 1,000 trees, and we cover over 14,000 individual beetles belonging to the 56 species. And we cover over 19,000 pest toys belonging to 12 families. Majority of them are Baconic pest toys. And so just give you example, this is an example from a subsample about 500 specimens of the Baconic pest toys. Most of them belong to the top 2 species, the Encina manipus and the Lepidostranchus picus. Those 2 species accounted for about 75% of the collection. And so this is why we're very interested in the 2 species, and this seems to most common novel pest toys in North America. And so we have started evaluating of these 2 pest toys. Actually, the first one we have done quite a lot of studies so far. And so this is native to North American, is gregarious ectopestoids. The other one is also ectopestoid. And this one is neither attack AOB in our quarantine learning. We have learned so far for over 100 generations. And so the evaluation procedure is very similar, as Kim and Julie all talk about, very similar in our quarantine loan. Basically, we just go through these procedures, look at the efficiency, and then the host specificity, and then also look at the climatic adaptability, just try to predict the potential geographic range if we're going to release them, and also try to develop some optimal learning methodologies. But so far, we haven't crossed this line, haven't started release any pest toys for the AOB. So I'm just going to give you quick 3 examples about the evaluation of this pest toy. So this study, we look at the host ninja test with this pest toy. And in this particular studies, we look at whether this parasite prefer to attack AOB or the 8 different non-target or alternative hosts. The top 7, those are common semian biases from North American. The last one is the citrus non-harbor is also invasive species. This one also originating from China has been detected in Europe, but it has not been detected in US yet. But this is very similar to AOB. It's also a high-risk invasive species. So what we found is this parasite didn't attack the top 3 beetles, but neither they attacked the bottom 5. And it didn't show a preference between AOB and each of the 5 attacked species. Well, the top 3 actually belong to the different subfamily than the bottom 5, which share the same subfamily as AOB. So still, this parasite seems to be, generous, but still prefer to attack the species more closely related to the AOB. The second experiment, we look at the host size effect and whether the parasite prefer different host size. So in this study, we also using choice test to look at the parasite preference between the 2 significant different size. The larger host about 5-6 in star and the small one about 7 in star. And we found that the parasite didn't prefer for any size. Also, the host size did not affect the survival and development time of the parasites. But it did affect the clutch size. I mean, the female parasite lay more egg on those larger hosts than the small ones. And the female wasp derived from larger hosts, also larger than those from small one. And consequently, the larger female produce more mature egg than those from small one. So overall, the parasite would benefit by attacking larger hosts. And this have some indication for learning. That's why we have been using a larger host for learning. And so the last example just show you some optimal learning parasites and host densities. So in this study, we manipulated different density of parasites and hosts, and then look at the overall parasitism as well as the per capita efficiency based on the number of biology produced per female wasps. So overall, the parasitism increase was increasing ratio of parasites to hosts, but the per capita efficient drop was increased ratio of parasites to hosts. So an optimal ratio would be 3 or 4 parasites to 4 hosts for efficient learning. And so I just want to show you a few examples about the evaluation of those parasites. They're very similar for the evaluating almost all the candy parasites. We just go through this similar process. So in conclusion, so far, there's about 29 parasites that are recorded from AOB or associated from AOB potentially. And most of them are recorded from China during our survey. Actually, most of them discovered during our survey in China. In South Korea, there was 1 species recorded, and Sonia just mentioned maybe there are another 2 different species. And then in North America, we have confirmed 5 species capable of attacking AOB based on the lab test, and also the other research in Europe have tested some indigenous parasites from Europe. At least 7 species are capable of attacking the AOB. But all of them are novel ectoparasitoids, except 1 novel indoor parasite. And most of them are recorded from Baconidae and Tomonis. So to conclude, we haven't found the egg parasite from China. Maybe Sun Yan found 1 from South Korea, and I'm really counting on him to collect more next year, hopefully, and so we can import into US for some evaluation. So we have found some novel parasites from China, and we just started about to evaluating the potential of this parasites for classical biocontrol. At the same time, we have evaluated some North American novel parasites. And so like the Encina memphis that I just mentioned could be augmented maybe to suppress AOB if AOB became established, or even help with current eradication efforts. So I would imagine in the future, the optimal schedule for control by control. It could be combination with both exotic egg or young larval pesticides with indigenous larval pesticides. And so this is hopefully we were able to find some effective pesticide from Asia. And so so I'm just going to end up here, and thank you for your attention.
Okay, Heng, thank you very much for the presentation. Yes, this is almost time, but we may have one short questions if anybody has. Okay, no question. You are helping. All right.
Thank you.
Thank you very much, Heng. And then, as it is scheduled, maybe we better start in 11:20 here in the morning, but in US it will be 10:20 in the morning. So we may take a break 5 minutes. So please return in 5 minutes. Thank you.
Okay, can you see my presentation file? Okay.
to the Zoom meeting again. Because we are scheduled 10 minutes interruption delayed, so we only use a very short break, only 5 minutes. This is the time. Continue the next talk. Yes. Next talk, Hyojung is here. The next presentation, may I introduce the next speaker, Professor Hyojung Kim from Kunsan National University. He will talk about the tracing the origin of the Korean invasive population of the spotted lanternfly. Are you ready?
Okay, do you hear me?
Yes.
Okay. So, I am Hyojung Kim at Kunsan National University. So, thank you for inviting me. So, this symposium. Today, I give a talk on my study of inferring the invasion route of and origin of SLF, spotted lanternfly. So titled as Tracing the Origin of Korean Invasive Populations of the Spotted Lanternfly. So this exotic species now widely spread in Korea and seriously damaging to grapes. So it is necessary to find its origin and invading route. Okay, so these are contents. So 4 sections. This species, spotted lanternfly, so SLF, suddenly came to Korea and rapidly spread throughout the middle and southern part of Korea since about 2005. The first official record of SLF in Korea as publication was published in 2006. Occurrence of SLF in vineyards and the neighboring area at the time of suddenly increase in 2009. So here. So by sap sucking, so damaged by these insects. So to grapes, although its main host is the native Chinese tree, as you know, Alliandus altissima, so which common name is tree of heaven. So as you know, but I think that this tree is tree of hell. So at the time, it had not been known where this species originated exactly from. So Animal and Plant Quarantine Agency in Korea, so wanting to know its origin and invading route and funding this research project during 2 years. So this photograph was taken by me in Cheonan and central area of Korea in 2006. So lanternfly distributes in Eastern Asia. So it is known to be originated from China. So its native range is very wide, Midwest, Middle, and Southeast, and South of China, and including Taiwan. And it also distributes in India and Vietnam and adjacent to China. But it is known that its population density is very low in these 2 countries. In Japan, this species likely invaded in Ishikawa Prefecture in 2009. So this introduction in Japan was confirmed through Japanese taxonomist. It is suspicious that this species was transferred by some transportation rather than own migration. So our investigation was focused on the eastern coastal area of China. So by trading, so they are so not migrate by natural condition, and so it is suspicious that they were transported by human activity. In this study, we want to know the genetic differentiation and relationships between geographically isolated population in Korea and China and Japan. So based on population genetic data, gene flow, and dispersal or the pattern of spatial distribution can be revealed. So finally, we confirm the invasion route and origin of Korean lanternfly populations. This material and methods. For population genetic study, first we collected lanternfly samples in Korea. The population samples were collected from 44 sites, which approved to 27 collective populations. This species now distributes everywhere now, except for some island regions in Korea, including Jeju. So strangely, SLF suddenly occurred in two separate regions, capital area and central area, and rapidly spread out. So we collected many samples at the time in two initial spots, like this. And we sampled lanternfly in China during 2 years. We visited to China twice, 2010 and 2011. So because it is predicted that lanternfly was transported by hitchhiking with transportation of importing materials into Korea, we targeted to collect samples in major port cities in China, eastern coastal area, yes. And then we collected samples in those Beijing, the South Tiantai, from 21 sites in 12 cities located in Hebei area and Shandong, Jiangsu, and Zhejiang. So just one population sample collected from Ishikawa, Japan, was sent from Japanese taxonomist Dr. Yoshitake. So how could we find the genetic variation between recent divergent groups? So we can apply population genetic study to reveal more recent divergent groups and trace invasive origin of exotic insect. So using microsatellite or an SMP SNP loci. So both estimations based on the different allelic condition. But in case of microsatellite, mutation model is fully developed. So as you know, compared with SMP, but we use this high cost specific marker. So we design microsatellite markers rather than SMP. So it is facilitated to resolve very recently radiated group like spotted lanternfly. To confirm, so first of all, to confirm mtDNA haplotype between collected populations, two mitochondrial genetic markers, ND2 and ND6, were tested. There are mtDNA regions known to be highly variable in hemipteran insect, as you see, so more than other regions. Therefore, we developed primer sets using the full empty genome sequence retrieved from the GenBank. And we isolated and developed new 15 microsatellite markers from lanternfly, as these loci were characterized in a single population in Korea. They were confirmed to be highly variable in allele mutation enough to do population genetic study. Then we performed fragment analysis, and raw data were analyzed in GeneMapper 3.7. And after we estimated 15 microsatellite loci, 12 were selected through for population genetic analysis. In the data analysis, 768 lanternfly adult individuals were used from 40 populations in Korea, China, and Japan. So 15 to 40 samples in each population were included. 1 or 2 samples in each population were sequenced using ND2 and ND6 markers, and haplotype were analyzed using neighbor joining method in Mega software. And then population genetic analysis was carried out based on multi-locus genotype of 12 microsatellite markers. So we used common population genetic software like such as GenPop and GenAlex, GenClass2, and Structure, and DIABC. So results. First, we analyzed two mtDNA marker, ND2 and ND6. So haplotype in ND2 and ND6 was 100% identical. So among Chinese, Beijing, Tianjin, and Shanghai, and all other Korean populations like this. So however, Linan and Ningbo and Tiantai population, so from Zhejiang province, had a big difference, genetic difference from those other populations. Second, we analyzed population samples using microsatellite F statistics and estimate over NM over all 40 populations were calculated in each locus. So estimation results show that the low FIS and relatively high FSD values in each locus. So, and NM values are very low. So it is useful markers to reveal the population genetic differences between lanternfly. So I accepted only 12 markers, and the 3 markers is not polymorphic within the populations. To display genetic distance and relationship between populations, first of all, principal coordinate analysis was performed. The first PCOA test included 27 Korean and 12 Chinese and 1 Japanese populations. In the plot drawn by the first 2 components, as you know, Zhejiang and Hebei area and also Shandong population in China were placed far from the other populations, as well as each other. Second PCOA analysis was performed, excluding the Hebei and Zhejiang population, these 2 populations. So Shanghai population is very close to other Korean populations than south of Shandong and Jiangsu populations. However, Samcheok is so close to Shandong populations, and the Japanese population is close to other 2 Korean populations. So collected from Yecheon and Sangju, sorry. And then I tested spatial autocorrelation analysis, which performed to know the spatial structure based on geographic and genetic distances. In China, local lanternfly group, which genetically connected between populations, distribute within approximately 250 kilometers and at least within 400 kilometers. Whereas Korean lanternfly groups spatially distribute within 9 kilometers, approximately 90 kilometers. Compared to Chinese spatial distribution in lanternfly native range, and it is possible that Korean populations will continuously expand further in Korea. So naturally spread out, so is equally to this condition, so genetic condition. Genetic structure was analyzed using structure program. The most likely the number of cluster K, which means number of ideal cluster among all samples, was estimated from 2 to 5. When K is 3, the delta K of likelihood value was maximized based on Ebano methods. Whereas when K is 5, the likelihood value is maximized. As shown in structure analysis, so graph structure of all Korean population here, corresponds to that of Shanghai population here. Yes. So you can see compare two populations here and here. So structure of Hebei area and Zhejiang population is clearly different structure. So like here, pink colored. So and then so initial population, one Korean population from Cheonan is rather similar to Beijing population. So and Samcheok here, Samcheok population is so similar to other Shandong populations. So I estimated that at least 3 different groups, subgroups in Korea, and there are the distinct also 3 groups in China. Structure assignment was mapped as pie graphs, and most Korean population, as you see, very similar to Shanghai population. So Japanese population similar to Beijing, and so already I said that Samcheok is similar to Shandong population here, blue colored. Genetic assignment test was performed using gene class 2 analysis. We compared each Chinese population with all Korean populations using individual assignment method. So as a result, Shanghai is most likely assigned to the source of Korean population. So like here. And then so other genetic assignment was used, and we compare the source of population. So like here, because Shanghai was estimated the most likely source of other Korean population, we inferred the migrate route between Shanghai and other Korean populations. We also use individual assignment, then gene class after test. So we selected the first and second likelihood values and connected that. So from source to invading populations. So as a result, to separate different place and capital area and the central area, and Incheon and Seoul and Busan and Nonsan. So we inferred the initial invaded regions. So assignment test also correspond to the FST, as you see, the Nonsan, Busan, Incheon, Cheongyang. So they have very close genetic differences between pairized FST to Shanghai population. Finally, we estimated 4 introduction scenarios using approximate Bayesian computation in DI-ABC program. We set 3 populations as POP1 to Shanghai as source, so POP2 the capital area, and then POP3 the 2 central area and the other invading population. The first and second scenario hypothesized to serial introduction, so POP3 introduced via POP2. So while the third and fourth were hypothesized to independent introductions, so this scenario hypothesized the temporally different condition, and this hypothesized being introduced at the same time. As a consequence, scenario 4 showed the highest probability in both tests, a direct estimate and the logistic regression. Therefore, the random fly population from Shanghai came to 2 separate Korean locations, so independently at the same time. They spread out and then nationwidely. So these results support that the random flight transport by human activity rather than natural migration. We collected and investigated the random flight initially in Shanghai twice. This collection site is an island named Chongming, so located in the northeast Shanghai, 15 kilometers far from the mainland. So I guess that this island is small the quarter size of Jeju Island, so small size. There is a large plantation area, Tree of Heaven, as you know, which is main host of lanternfly. We found many adult insects and egg masses in the bark. So population density was very high when we visited here. So we expected that this region is the source of so Korean lanternfly invasion. The last slide. In ND2 and ND6 haplotype analysis, or Korean population, so lanternfly matched to Hebei area and Shanghai population. So among them, Shanghai was most likely estimated to the source region of most of Korean population, except for Samcheok and Cheonan in 2006. The multiple introduction of the spotted lanternfly was detected at least 3 times and introduced into Cheonan and Seoul and Busan and Samcheok. Yes. Korean population was narrow spatial structure than Chinese population, and the dominant genetic group was inferred to be dispersed independently from 2 separate regions, the capital area and central area. Many scientists contributed work and support this project from Korea, China, and Japan. Thank you very much for your attention.
Thank you very much, Professor Hyojung Kim, for a nice presentation. And because the time is almost, as scheduled, we have to move to the next talk. And then probably we can have the questions, both of the, next speech also about the lanternfly, we can have questions together. Okay, let me introduce the next speaker. Dr. Hannah Broadley, USDA ARS, the plant protection and quarantine. There's Buzzard Bay, Massachusetts, probably. And then she will talk about the development of the biological control method against the spotted lanternfly in US, research on egg and nymphal parasitoids. Hannah, are you ready?
Are you getting the speaker view or the full view of it?
Sorry? You can.
Do you see the slides right, or is it just the speaker view with the notes and stuff?
Yeah, I think it's the slide already ready. Yes, no problem.
Okay. Yeah, because when it shares for me, I just see the speaker view with the notes and all that. But if you guys are good, I'll use this. Yeah. All right. Thank you. And you can hear me okay?
Yes, good.
Okay, great. Well, thank you very much for inviting me to part of this group. This has been fascinating. It's getting late here, but all these talks have been keeping me up. So thank you. So I'll be presenting on some of the work we have on the development of classical biocontrol methods against spotted lanternfly in the United States. So this works really well going after Dr. Kim's talk. This is work that I'm doing in close collaboration with Julie and Kim Holmer. I joined their group. They had started the work on this along with Dr. Wang in China. So I'm going to just go through this quite fast because Dr. Kim already covered some of the background of spotted lanternfly, but speaking to a little bit about the invasion of spotted lanternfly in the United States. And I will throughout, I'll be referring to spotted lanternfly, either saying the whole name out or calling it SLF. So in the United States, spotted lanternfly was first detected in the eastern United States in our state called Pennsylvania. I'll show you a map of that location on the next slide, but it was first noted in the fall of 2014. As Dr. Kim said, you find it on Tree of Heaven or Tree of Hell, as he mentioned, as its preferred host plant, but you also will find it on a number of other species, ranging from fruits, ornamental, woody trees. I think the list of species is 70 plus different species right now. It's a particular issue for growers of grapes, hops, and some stone fruits. It causes really heavy honeydew as they're feeding, and then you get sooty mold and damage from that. So here's the location of where spotted lanternfly currently is in the United States. This was just updated last month. It came from that epicenter around right in the middle of the blue area in this state of Pennsylvania here in the eastern United States. And it's been spreading this fall. You know, the adults are out now, and there's been a lot of new detections, not necessarily of populations, but new interceptions of the adults at various different states. So the population is by no means under control. There's also really big concern about the possible distribution of all the places in the United States that this could potentially establish. So moving across a lot of the East, moving into the Midwest of the United States. There's also big concern because it's highly suitable for California, out on the very West Coast. And that's a lot of growing in the United States. That's vast farmland and growing area there. So there's a lot of concern about how to manage spotted lanternfly in the United States. There's also a lot of challenges. I've mentioned a few of these, but just re-emphasizing challenges in how to contain spotted lanternfly. The egg masses are cryptic. They're very hidden on the bark. They're very hard to see. They also don't only lay on bark. They'll lay on wood or stone or rusty metal material. And so people will inadvertently or accidentally move the egg masses. It has a very broad host range, so it's hard to, if you're looking for it, there's not just one species of hosts that you can look at. The adults can also disperse long distances on themselves or by themselves. They can use wind or thermals to disperse. They also get help from human transportation, of course. So the populations are continuing to spread. Hitchhikers, so human-assisted movement. And also there's been so far very little mortality from any of our native or resident natural enemies. So Julie and Kim jumped right on this as soon as they could, starting to work on doing foreign exploration to try to develop a classical biocontrol method. And then I joined the team about 2 years ago. So I think a lot of us are pretty familiar with the general idea of how you develop biocontrol methods, but I just wanted to speak a little bit to our approach for spotted lanternfly because I'll be talking through some of these results that we have. So starting, some other researchers, and we were starting to work on this as well, have been evaluating native and resident parasitoids in the United States. In particular, with collecting egg masses over the winter, we've noted some wasp emergence holes in some of those egg masses, and we also inadvertently, so accidentally, got wasp DNA when we were trying to get spotted lanternfly DNA. So we're really interested in what specifically that is. Kim and Julie worked with Wong as soon as they could, starting to work on foreign exploration work. And then I had the opportunity to join that last year, doing some foreign exploration work. We've also been doing different studies both in the United States with our colony of wasps that we have in the lab, as well as work in the field in China, looking at the life history of the different parasitoids that we're working with, working on the life history, the life cycle, and population genetics of them, then developing a really good rearing system for these parasitoids, testing their host specificity, and then applying to get approval for release. Of course, then there's all the stages of releasing it and studying the effects of it, but we're still a ways from that. So briefly, some of the foreign exploration work that we've been collaborating on in China. So they've been working on, for the last few years, doing different surveys across China. So here's a map of 27 of the different provinces and regions in China and the surveys that, along with the collaborators there, they've been able to conduct over the last 5 years. And that's been surveys both for spotted lanternfly and then any associated egg parasitoids or nymphal parasitoids. So doing by collecting egg masses, deploying sentinel egg masses, using sticky traps, putting out bottle traps with nymphs in them. So a variety of different ways to try to find any of these parasitoids. Spotted lanternfly so far has been found in 27 of those different regions, and 2 parasitoids have been identified, and we're conducting further work with those. So these are the 2 parasitoids that we're focused on right now. Anastatis orientalis is an egg parasitoid, and then Dryaenus sinicus is a nymphal parasitoid. So I'll talk briefly about these 2 and some of the work we're doing with them. We have more work right now on Anastatis than we do with Dryaenid. We've started working on Dryaenid more recently. So Anastatis orientalis, as I said, is an egg parasitoid of spotted lanternfly. It was first discovered in northern China. It was reported to parasitize 30% of egg masses and then 40% of the eggs within each egg mass, and was reported to have 2 generations per year, one in the spring and then a generation in the fall. So it is reported to be able to use the spotted lanternfly eggs before the nymphs hatch, before the spotted lanternfly nymphs hatch, and then it can use the newly laid spotted lanternfly eggs again in the fall. It was collected and considered as a biocontrol agent for spotted lanternfly in South Korea, which I'll talk a little bit more about that because we applied for funding to work on the role of Anastatis in Korea. So in Korea, as Dr. Kim said, it was first noted in 2008 and then rapidly spread there, introduced from China. We're really interested in all the work that has come out of the Korean work on this about the spread, life cycle, overwintering mortality, behavior. We as in researchers in the United States is what I mean, have been looking a lot to this because Korea has been dealing with spotted lanternfly for many more years than the United States has. So we've been really relying on some of that work that has come out to learn more. And work with Anastatis is no exception to that. So as you probably know, so Anastatis parasitoids were discovered in Korea in 2011, as well as some other work with natural enemies. And then there was further work by Mija Sao on how to rear Anastatis, and also different life cycle information about Anastatis orientalis, I should say. So that report in 2011 was unknown Anastatis species. Dryaenid sinicus, so I'll just briefly touch upon this and then I'll go more into our research with Anastatis orientalis. So Dryaenid sinicus is this nymphal parasitoid. It's known to attack second and third instar nymphs and appears to be univoltine, so 1 generation per year. It grafts onto the nymphs, the prey, and then paralyzes them, deposits its egg, and then you get this structure called a thylacium. That's that white sort of bubble behind the wing pad. That's the developing parasitoid nymph, and there's reports of 23 to 60% parasitism of nymphs in the field with this species. We've had Anastatis orientalis in colony for a number of years now, so we've been able to conduct some research on the rearing of it, how to store it, longevity, fecundity, sex ratio of the species, so we can learn as much as possible. We're also working on testing the host range of this species to be able to consider it as a biocontrol agent. We have work going on the life cycle, so we understand how well synchronized it is with spotted lanternfly, the genetics. So we have some interesting work there where it looks like we may have discovered a cryptic species, or at least very different populations of Anastatis. Kim's lab and Joe Kaiser, who was on earlier, has been doing a lot of host selection behavior work. And then we have a lot of future plans looking at diaposonate, conducting more foreign exploration work. As I mentioned, understanding the population dynamics and the role of Anastatis orientalis in Korea, and then also host feeding. We know that Anastatis orientalis host feeds, but we don't have a very good understanding of what additional mortality, if any, that is causing. So just quickly, I just pulled out a few highlights of a paper that we have in press right now. So we were looking at the longevity and fecundity of Anastatis orientalis females. They're very long-lived. We were able to rear out a number of those females. We did 30 different replicates with this, looking at their fecundity and progeny produced over time. And they were living up to 16, 17 weeks. They weren't producing a lot of progeny per week. So you'll see there, you're only seeing about every half week, you see about 15 progeny produced. So 30 per week at their most productive time. If they're not provided with another mate, you have a very, start with a very female skewed progeny, and then likely because of sperm depletion, you end up with more males towards the end of their life without additional mating. And this third panel here is just showing that we were able to use year-old stored spotted lanternfly egg masses and got no significant difference in the number of progeny we were able to produce with the old egg masses versus the new egg masses, which is really useful for us because we don't have a good rearing system for spotted lanternfly. So we rely on collecting those egg masses in the field over the winter, storing them in chill, and that's what we keep our colony and all of our studies going with over time. In 2019, we did a study looking at the life cycle of Anastatis orientalis in our growth chambers. So we mimicked Beijing conditions, and we mimicked conditions, light and temperature conditions of the eastern United States. And we had population or collections of Anastatis parasitized egg masses that we ran through these different conditions to see how well synchronized they were with spotted lanternfly. The answer from this first year of study was not well synchronized. We had emergence in the spring like we were expecting, and then there was early summer emergence when there's no spotted lanternfly egg masses available for the wasp. So they presumably are using another alternate non-target host. And then there was emergence for the Beijing conditions again in the fall. The conditions, the eastern US conditions, we never saw another fall generation. So that's sort of a dead end there. So we were obviously disappointed about that because it suggests that they are using alternate hosts. But Julie initiated looking at better area that might be a better climatic match. And what came up with that is that Yantai looks like a much better climatic match to eastern United States area where spotted lanternfly is invasive. We're conducting another year of this life cycle study and interestingly getting different answers. It's very confusing, and so I'm not going to go into too much of the details now. There's just not enough time. But we are seeing this Yantai population is behaving differently. It's emerging after spotted lanternfly nymphs are coming out, which we don't quite understand if that means it's using an alternate host. Also, we're getting another round of emergence from those collections now, and we're running some DNA work on that. Some of the DNA work that we have looked at, though, is we are seeing different genetic groups of this Anastatis. what we have in colony is this haplotype or this group C, but there's also this other potentially cryptic species that we're calling this haplotype A, B, that group there, which using a portion of CO1 is about 1.3 to 1.8% different. So this is something that we're now trying to separate out into isofemale lines and conduct further work on to see if this different anastatis group or haplotype or species, whatever it is, may have a different life cycle and may or may not be more host specific than the haplotype we have in colony. We have host range testing underway. We have selected 11 priority species. These are closely related native plant hoppers, but from initial work that showed that Anastatis orientalis that we have in colony, that haplotype C, that it appears to be able to attack non-targets, we expanded out the range of species that we're testing. So now we have in no choice tests, we have 30 different species ranging from Hemiptera, Lepidoptera, mantids, Coleoptera, and stake bugs. So you can see we have quite a broad range that we're looking at now. We have gotten non-target parasitism, unfortunately, with some of those wasps producing females and male offspring, some of them just producing males. So those are probably less preferred hosts, but we are getting non-target parasitism, which here's a little bit more of the detail of all the non-targets. Out of those 30 species, these are what have been attacked so far. All of these species here that I'm showing here, all with non-target testing, all of those are now being moved into choice testing to see what happens in that case. We don't have results yet with those choice tests. As I mentioned, Kim Homer's lab is meanwhile conducting behavioral assays to look at how the Anastatis may be searching for hosts and showing preference in its behaviors using ethovision work, behavioral work. Some of that work some of the initial work is published. So Robert Malik published that out of Kim Homer's lab. Joe Kaiser's now heading up follow-up work with that. Robert Malik's work does show that that Anastatis is attracted to spotted lanternfly, their footprints and other cues that they leave behind, as well as the ootheca of the egg masses. With some of our initial work looking at behaviors against Publicia, which are most closely related species in Lycorma, in all of those cases, we see that Anastatis shows much stronger behavioral responses to cues from Lycorma than our native species, Publicia. So that does suggest that even though Anastatis orientalis can attack non-targets, those are not preferred hosts. I know I am almost out of time, but I just have a couple more slides that I'll go through quickly. And then there probably won't be time for questions, but I'll just finish up these last couple slides. So Dryaenid sinicus, we are still in the initial stages working with that. That's been a really difficult species to collect and rear. We now do have 70 cocoons in the lab. We are just watching those pupae. They should be out in the next couple of weeks now. We have nymphs ready for them to parasitize. So we're hoping that we'll get a colony of this up and going in the next, this fall. And then we can conduct further work with that. And I just wanted to bring up, so we have future research we're really interested, assuming we get funded with the proposal that we put in. We have future research that we're hoping to do to look at surveys across Korea, looking at the egg and nymphal parasitoids in Korea, and also conducting life table studies to understand what role natural enemies are playing in Korea. Because as I said before, we're falling behind Korea, trying to learn as much as we can from you guys are a number of years ahead of us. So we think there's a lot to learn there. So just in conclusion, we have two potential biocontrol agents we're working on. Anastatis orientalis that we have in colony appears to have some generalist behaviors, but we're looking at potentially a cryptic species. We're working on the nymphal parasitoid. You can see we're still at the relatively early stages on developing this biocontrol work. And we're also looking at some native species. This is all done with many, many collaborators. This is just a few of them. So I will wrap up there because that was a couple minutes over. So thank you for your time.
Okay, thank you, Hannah, for the very nice summary and introduction about the lanternfly and their parasitoids. And then let's have a question or comment for both two speakers, Hyojung and Hannah together, because this is for both for the lanternfly. If anybody has a question or comment, please. The time is already over than scheduled, but we may have one or two questions. Okay.
Can I make a question?
Yes, Do-hyung?
I think this question can go to Hannah and also Professor Kim. We actually try to establish a laboratory colony of the spotted lanternfly. Basically, we totally failed. Do you know anyone who successfully established and maintained the spotted lanternfly colony and also their secret?
Not in the United States, not to date. Julie worked on that for a number of years. She gave up on it, passed it over to another collaborator, Tracy Lesky. I think Tracy Lesky, she's ARS, is probably the closest. She has gotten it from all the way from egg to egg on a number of individuals, but by no means does she have a very productive or efficient way to rear it out. Julie and Kim, you may be able to add more information, but I think Tracy is probably the closest. But she's only reared from egg to egg a handful. I don't know, it's probably like a dozen that she's gotten from egg to egg.
I think the key is that you have to use living plants. We were at first trying to cut foliage in the field and bring it back, and that was totally unsuccessful. We are now Tracy Lesky has figured out how to take Tree of Heaven seeds and rear them up into plants. And I think that's going to be the key. Plant seeds, get a Tree of Heaven and rear the nymphs on that. I think that's going to be the best solution. And it's very easy to collect Tree of Heaven seeds, and it's very easy you have to chill them. You have to make sure they have a cold period, but then, so that's the key, I think.
So we are, at least for our dryinids, we have egg masses we collected in the field. Well, we collected all of for the anastotis and the dryinids, collected in the field, and then we're rearing out those to get nymphs that are second and third for the dryinids, but we're not even trying to go really beyond that. We just want something that the dryinid can parasitize. But it's a challenge. A lot of different people in the US have been working on it, and it's...
Okay. Thanks. I can definitely talk to Tracy Lesky. By the way, I did my postdoc with Tracy Lesky, so...
Oh, wonderful. I didn't realize that. Yeah, she's the one that's gotten it the furthest. So, yeah, talk to her.
I visited her in February, but I didn't see her lab colony. So, probably some progress and it's coming, probably. So, I can talk to Tracy. Good to know.
The eggs were just this fall, because she's rearing them, I think, in natural conditions, not accelerated. So the eggs were laid this fall. So she certainly will have updates for you. That's
there
wonderful.
There are a few researchers at Pennsylvania State University who are also working on lanternfly biology and have been working on rearing. And I think a couple of them also have made similar small steps at being able to rear lanternfly through to egg laying. I don't remember exactly who those persons are, but Julie Urban, who's leading the big SERI grant for lanternfly for Penn State, would be able to tell you which labs have made the most progress. But I think it's not significantly more than Hannah's described.
Okay, thanks.
Okay, yes, the organizing committee of this conference pushed me to quickly finish these sessions because there is another, the symposium starts again. So I would like to ask every participant to, to communicate through the other, the way, the email, or we can make another, that personal or separate satellite meetings if we need. I think that's another way. So we have one more talk by the Kyu Im. She's working in the National Quality Service of Korea. She will talk about the International Year of Plant Health and IPPC strategic framework. But this is a little bit another subject, but it was scheduled. But because of the conference place having problems of the internet access, she will give her talks through the video, so it will be known after finishing our meetings. So I would like to thank everybody, especially for the speakers, especially for the speakers from US. This is probably very late in the night, and then we have to go to bed now. Okay, and then also we have to collaborate more about the investigating national enemy here in China, and then also probably hopefully the COVID situation, getting better, and then you can come, to this place and then collaborate. So thank you very much for all the speakers, and then good night there in US, and then here in Korean members, have a good lunch. Thank you very much.
Thank you. Thank you. Bye-bye.
Thank you so much. This was great. Thank you.
Thank you very much. Thank you,
Bye,
Kim and Hannah and Wang.
Okay, thank you, Professor Lee. Bye-bye.
Bye.
Bye.
See you sometime.
Thank you.
Bye. See you later. Okay.
See you. Goodbye. Thank you.
Bye.
And then I will toss the host of the Zoom to Young-Suk Lee is here. Oh, he's not here. Yeah, we are here. And I will toss over the host to the Young-Suk Lee, who will run over the video of the Dr. Im Kyu-oh. Yes, he will run the videos.
Good day, everybody. This is a Q&A game from the Animal and Plant
이영석 씨 지금 발표시키나 지금 뭐가 하나 올라왔는데 근데 이렇게 올라오면 되나? 이영석 씨 이게 지금 비디오가 어떻게 돼 있죠? 너 들리지도 않는 비디오인데 지금 임규옥 박사님이 비디오 올리시기로 하셨다가 지금 나오셨나요? 제가 우리 홍기정 교수님한테 들었던 내용하고 좀 다르네. 바로 직접 하실 것 같은데? 지금 들리질 않아요. 그쪽에서 하는 얘기들이 전혀. 어떻게 됐지? 여기 지금 임규옥 박사님 나오셨잖아요.
네.
그러면 직접 하시는 거네. 얘기하고 달랐네.
예, 직접 또 하신답니다.
아이고, 죄송합니다. 그러면 지금, 누구야? 우리 이종호 박사님,
네.
예, 여기 들어오셨는데 사전에 좀 알려주시지. 이거 결례를 했네, 우리 임교
Good day,
박사님한테.
everybody.
그러니까 좀 왔다 갔다 좀 했습니다. 그래서
야,
지금
근데
소리가 들리세요?
우리는 저기 이종호 박사님 건 들리는데
네.
그 이영석 선생님 아이디로 들어온 임규옥 박사님은
네.
소리도 안 들리고
네.
소리도 안 들리고 전체 화면으로 보이질 않아요. 어떻게 해야 되지? 우리 이 박사님 그쪽에 아마 테크니컬리 문제가 있는 것 같아요. 전체 화면으로 뜨지를 않아, 임교옥 박사님께.
글쎄, 기술적으로 좀 문제가 있는 것 같은데요.
네.
지금 소리 들리세요?
이게 이종호 박사님 건 들리는데.
제 소리만 들리고 박수 소리 안 들리죠? 예. 안 들려, 지금.
이영석 박사님 이름으로 돼 있는 임규옥 박사님은
네.
그 화면 안에 이영석 박사님 그 창 안에 조그맣게 그 화면이 보이긴 하는데 소리도 안 들리고 전체 화면으로 보이질 않아.
이영석 박사님 아이디로 들어온 거에는 화면이 조그맣게만 보이고 전체 화면도 아니고 소리도 안 들린다는데? 임규옥 박사 얼굴은 조그만 화면으로는 보인대요. 그죠?
그러니까 보통 이제 그렇죠. 이게 지금 아마 거기 비디오를 카메라를 발표하는 그 화면으로만 보이고 프레젠테이션이 지금 안 올라와 있어요.
예.
소리가 안 들려요. 임규옥 박사님 소리가 안 들려요.
소리가 안 들려요?
네.
그러면
임규옥 박사님 말씀을 해보시겠어요?
임 박사님 말씀 한번 해보실래요?
Hello.
안 들려.
안 들려. 이런 교수님 녹화된 영상을 갖고 계시죠?
이거는 이제 우리가 피니시하면 그게 녹화가 될 거예요. 이 회의를 피니시하면.
아니, 아까 기존에 임규옥 박사님 발표 녹화해서 보내드린 거.
그거는 이영석 씨가 그쪽에서 아까 홍기정 교수 얘기로는 이영석 씨가 그쪽에서 올려주기로 했어요.
이쪽에서 올리기로 했었다고?
네. 그래서 우리가 호스트 지정을 해준 거거든.
뭔가 지금 문제가 있네.
아이고, 우리 임규옥 박사님한테 이거 너무 죄송한데. 아니, 결정을 했으면 그대로 하지 왜 자꾸 바꿔.
안 되죠. 그러면 원래 녹화된 거.
여기서 호스를 넘겨주고.
교수님.
네.
교수님, 아까 원래 이메일로 받은 거 갖고 계시죠?
그거는 이승현 박사가 지금 다운받아놨나?
아니, 다운받으려고 했는데, 다운받는 게 억세스 디나이드라고 떠가지고요. 저도 못 받았거든요.
그러면, 그 파일을 여기서 지금 다시 올려야 되네, 그럼?
그렇게 한다 그랬다고, 홍기정 교수.
예. 영관님, 아까 USB, 녹음된 거 그냥 그거 올릴 수밖에 없겠는데? 안 되는데.
그냥 아예 그쪽에도 시스템 다 끄고 그냥 이 방에 개인적으로 들어오셔서 파워포인트로 하시는 게
그게 제일 좋아요.
낫지 않을까요?
줌으로 들어와서. 근데 그게 또 여기서 기술적으로 안 됐다고.
거기 있는 거를 다 끄고 그냥 노트북으로만 하시는 게 낫지 않을까 싶어요.
그 방법.
여기 거 다 끄고 노트북으로만.
아니, 근데 그것도 지금 거기 방안이 인터넷 액세스가 불안해서 우리가 그랬던 거 아니에요? 그것도 쉽지 않을 거야.
여기서 말로 하면 나가나요?
잠시만요. 이거 접수 안 하셨죠? 내 거로 들어가셔서 인사만 하시고 그렇게 하시자고 그러죠. 그래, 가다도. 그러면 교수님.
예.
이동 교수님.
예.
지금 일단 그러면 세션은 끝내고요.
예.
끝내면서 그 나중에 우리 홈페이지에다가
네.
우리 그 발표 자료는 올려놓는 거로 이렇게 해서.
제가 봐도 그게 나을 것 같아요.
예, 그렇게 하셔야 될 것 같습니다. 기술적인 문제는.
이쪽에서 할 수 있는 게 아니기 때문에.
예, 그렇게 좀 안내 좀 부탁드리겠습니다.
뭐 안내나마나 여기 들어오신 분이
다
들었는데 뭐.
다 듣고 계신가요? 죄송합니다. 이게 기술적인 문제가 있어서 저희가 이 회의장에서 발표를 직접 준비를 하셨었는데
그러게.
안 될 것 같고요. 그래서 저희가 학회 홈페이지를 통해서 오늘 임기혁 박사님 발표하실 자료들을 올려놓도록 하겠습니다. 죄송합니다.
아닙니다. 수고하셨습니다. 자, 그러면 우리 이 세션, 심포지엄 세션 오전 거는 모두 클로즈 할게요. 우리 임기혁 박사님 거는 홈페이지에 반드시 올려줄 수 있게 좀 해주시고. 그리고
다운을 못 받아놨대.
그 신박사 지금 들어와 있는데 신 교수.
네, 교수님.
들려요?
네, 들립니다.
그리고 우리 1시에 시작하게 하잖아요.
네.
지금 바로 아마 테스트 좀 해봐야 될 거예요. 그쪽에 두에인 박사하고, 누구죠? 송호준, 교수.
네.
들어오게끔 우리가 바로 한 15분 전에 오픈하자고요. 45분에.
네, 45분에 오기로 했습니다.
네, 45분에 오픈해가지고 들어와서 테스트하고, 그러고 바로 1시에 시작하는 거예요.
네. 이거 계정은 그냥 그대로 쭉 사용하면
아니요, 계정이 달라져요.
다른가요, 교수님?
계정이 달라져요. 그 심포지움별로 계정이 다 열리게 돼 있거든요.
이게 그냥 연속으로 한 방에서 되는 거라서 아마 연결이 될 것 같은데요.
그거는 상관이 없는데 문제는 이제 심포지움 그거 계정을 다 따로 줬거든. 잠깐만요. 어떻게 돼 있나 이게 아마 다를걸. 심포지움 계정을 계정마다 다 다르죠.
똑같습니다. 저희랑 이 똑같은 방에서 다음에 진행되는 세션 똑같아서요. 그냥 이 방 유지하고 이제 여기
되네, 그러면.
여기에 두에인이랑 송호준 교수님 들어오시면 될 것 같아요.
근데 그거 신박사 안내해 줬죠, 저쪽에다가.
네, 얘기해놨습니다.
요 아이디하고 패스워드로.
네, 거기 학회.
네, 같습니다.
네, 그럼 문제 없어요. 그러면 45분에 봐요. 식사들 하시고.
네네, 45분에 뵙겠습니다.
네.
제가 그러면은 신승환 교수님을 호스트로 만들어 드리고 자리를 보겠습니다.
네, 그렇게 하는 게 낫죠.
호스트 신승환 교수님 되셨고요. 이제 사람들 음소거하거나 그런 거 다 하실 수 있습니다. 네.
신 교수님 몇 시 몇 분에 발표하세요?
제 거요?
예.
예, 제 거 중간에 껴 있습니다. 제 거는 여기 지금 3시 10분에서 3시 반
예.
이렇게 돼 있습니다. 네.
예. 제가 볼 수 있는 시간인가 해서 여쭤봤어요.
뭐 수업 있으신가요?
아니, 잠깐 나갔다 와야 돼가지고. 예.
알겠습니다.
수고하셨습니다.
예, 고생하셨습니다. 이동훈 교수님. 예.
수고하셨습니다.
예. 오후에 뵙겠습니다.
마이크 테스트. 하나, 둘.
or specialized plant feeding. And from there, I'll go ahead and I'll share some general summary and conclusion of kind of what we've learned to date at that insect or beetle plant interface. So one of the major reasons why beetles have long been of great interest to naturalists is, of course, their extraordinary taxonomic diversity. And I don't have to tell all of you as entomologists who are very familiar, but that the lineage itself is just extraordinary for its variety. Taxonomic
네, 형님
diversity.
식사하셨습니까? 네, 사무실이세요? 네, 보내주신 거 잘 받았고요. 그런데 혹시 이 착수기 보내주실 때 공문 첫 페이지 혹시 있으실까요?
So the other thing that's notable about the lineage, of course, is that a large fraction of species are associated with plants. So they exhibit specialized, often very intimate interactions with plants, in particular with flowering plants. So my work began in trying to establish the sort of foundation for the phylogeny. Where is it that beetles reside within the larger phylogeny of insects? And until relatively recently, we weren't very sure about this. We had some understanding that they were relatives of beetles and perhaps a few other groups of insects that are in that neighborhood in the phylogeny of Holometabula, but we weren't certain. Statistical support and resolution in phylogenies available to that point was not really definitive. So in around 2010, I started working with a group in Bonn, Germany, primarily Oliver Nijhuis and Bernhard Miesoff, but also involving Rolf Beudel and a number of other colleagues. We put together our own genomic resources that we had generated for beetles and their near relatives, and we generated a number of pretty unique data sets at that time. One of them was one of the largest phylogenomic data sets available for insects at that time. It was 4,485 nuclear genes from across exemplars of the Holometabula. We also incorporated genomic metacharacters, which, by the way, I would argue are vastly underutilized at the moment in reconstructing phylogenetic relationships. So these are things like near intron pairs and their distribution across the genomes of different insects. We looked at gene order as well. We also studied non-coding RNAs, and we re-evaluated Holometabulous insect morphology. This data set included the first draft genomes for the Neuropterida. This was a megalopteran genome, Chaliotes, that I sequenced in my lab, was ultimately published in 2014. A Strepsipteran genome, the first one from Mengenilla that came out of Oliver Nijhuis and Bernhard Miesoff's collaboration. And then the first Archostomaton beetle genome, which I sequenced, the genus Priacma. We showed with these data that quite definitively Strepsiptera is the sister group of the order Coleoptera. And while that was perhaps something that a lot had already been fairly convinced of, what it allowed us with confidence was to move forward with an outgroup, with a taxon against which we could polarize in-group character states within Coleoptera. Now, further work, collaborative work, large, great big, really fun communities of friends and colleagues working together resulted in further establishing the higher level phylogeny of the Insecta using similarly large phylogenomic data. And I'm just sharing this here in the spirit of this symposium, looking at international collaboration and the relationships that grow from it and the important role that is played in furthering our science. So we had established the relationship between Strepsiptera and Coleoptera, but this paper, published in 2014, using phylogenomic data largely from transcriptomes, cemented those relationships in the larger context of Holometabula and even more broadly across Insecta. Again, providing us context in which to ask questions then about the phylogeny and evolution of beetles. So my work for the last 10 years or so has been largely focused on reconstructing beetle phylogeny at various taxonomic levels. Existing studies, and there are a lot of them, had a number of interesting and sometimes even different weaknesses. They related to taxon sampling and the numbers of genes that were included in analyses. Generally speaking, the data sets that had been published until recently were extraordinarily well taxon sampled by representing most of the major lineages, but they often missed including near outgroups. Things like Strepsiptera were typically excluded for good reason in some cases because of long branch attraction and other issues, but at any rate, there were differences and challenges in interpreting data sets to date. One of the biggest weaknesses, though, was that while we could place families in the context of higher taxa, like superfamilies or series, we didn't really have great confidence in the interrelationships among those major lineages. Statistical support was very low, and resolution in some parts of the tree was very poor at best. So we took a somewhat different approach than had been taken so far. We used phylogenomic data from 147 exemplars. Ultimately, we analyzed data from nearly 5,000 nuclear genes and around 2 million amino acids. So our alignment was on the order of 2 million amino acids in length. These data came largely from transcriptomes, but also involved some low-coverage genomes that we used for genome skimming. We generated a phylogeny and then calibrated that phylogeny using well-established and carefully vetted fossils. The data set itself, though, was pretty unique because in the context of phylogenomic data that were available even as of this year for beetles, all the studies so far, if you look at this plot, you can see had a lot of genes and very few taxa, or relatively larger number of taxa, but not so many genes. In no case, though, did you have a combination of a large number of taxa and a large number of genes, which could be carefully evaluated, carefully vetted, and only those which were most suitable for phylogeny reconstruction used in building a tree. So the data set that Dr. Shin and I analyzed, resulting in the paper we published in Proceedings of the National Academy of Sciences last year, had both of these features. A large number of genes from which we could select those suitable for phylogeny reconstruction, and a large number of taxa representing all major lineages. So the phylogeny that we reconstructed is shown here. Not surprisingly, because we had already pretty well been confident of this, Strepsiptera were the sister group of Coleoptera, and the suborders Archostomata and Mixophaga were sister groups of one another. The Archostomata and Mixophaga were themselves sister to the Adephaga, the ground beetles, whirligigs, and others. The skirtoids and Darodontids, a very interesting group, actually formed a paraphyletic grade subtending the remaining polyphagan Coleoptera. So Polyphaga is comprised of this extraordinary diversity of forms that have really Okay, I'm happy to start back up whenever you guys let me know when you'd like me to.
I think we just go ahead and start it.
Okay. So the polyphagan Coleoptera then, in addition to those groups I've already mentioned, include familiar taxa like the metallic wood-boring beetles, the
누가 여기를 자꾸 이 같은 아이디를 사용해서 계정을 오픈하려고 하는 건가?
들어보니까 뭐 전체가 다 튕겨나간 것 같던데요. 근데
무료 심포지움이?
네네. So as I told, this happened already twice in the morning session, so please bear with us. And maybe after more people getting in, maybe you can start.
Sure. I'll pause and wait a minute.
저쪽에 전화해가지고 이게 그 홀에서 누가 매니저를 건드리는 것 같은데 그거 터치하지 말라 그랬으니까 한번 믿어봅시다.
다시 그럼 시작을 해볼까요?
네, 좋아요.
Okay, go ahead.
Okay. Well, where I left off was mentioning the elatoroids, so click beetles, fireflies, and their relatives. So carrying on through the polyphaga, we established the placement of Incertacetus taxa like Nosodendron, which turns out to be a very important, relatively early divergent lineage within polyphaga. The clown beetles, the hysteroids, the hydrophiloids, the rove beetles and carrion beetles, the staphylinids and sylphids. The scarabioids, sister to Staphylinoidea, something that had been proposed early on by morphologists, but had not been supported by molecular data to date. The bostracoids, which were sister to the remaining Cucujiformia, the claroids, the coccinelloids, the ladybird beetles, the ship timber beetles, superfamily Limexloidea, the tenebrianoids, the darkling beetles, Cucujoidea, which formed a paraphyletic grade, and this has, by the way, held up in our subsequent analyses. It's interesting because if you look at the grade of taxa that forms this former clade, what we see is some really interesting transitions relating to early kinds of associations with plants. The Cucujoids then subtend then the chrysomeloids and curculianoids, the Phytophaga, this remarkable sister group relationship between these two superfamilies, which together account for approximately 1/3 of all described extant beetles, and probably more like about half or more of all extant Coleoptera. If we look at nodal support across this tree, it's really robust. There are a handful of nodes with less than 97% maximum likelihood bootstrap support. So for the most part, this tree is very, very well supported, well resolved, and provides a substantial foundation for further work. So what I really think was probably most useful for us in this case was that unlike studies to date before this one, we aren't in a case of incremental increases in resolution and in nodal support. We finally have a very well-supported, exceedingly well-resolved phylogeny that we can use as the basis then for further evolutionary inferences. We generated another data set, which would allow us then to more carefully establish timing and patterns of beetle diversification. This data set included 521 taxa, representing 140 of the 190 beetle families. It contained data from 89 nuclear genes, and these data were obtained via genome skimming, transcriptomes, and previously published data in NCBI. We calibrated the resulting phylogeny, and then in a sort of an interesting way, because this hadn't been done before, we added 48 additional families on the basis of previously published phylogenetic data. I don't have time to tell you about how that was done. You can see it in the paper if you're interested, but it allowed us then to sample 188 of the 190 described extant families of Coleoptera. This is a fairly busy figure. Let me walk you through the important points. What we recovered, and this, by the way, is a family-level summary tree of the 521 taxon tree that I just mentioned. We recovered a Carboniferous origin for the order Coleoptera. Episodic radiations beginning in the late Carboniferous with Polyphaga, here number 24, and 13 post-Permian increases involving descendant lineages of Polyphaga. So the red circles indicate significant increases in diversification rate along branches or crown groups leading up to terminals. Coleoptera survived 3 of the big 5 Phanerozoic mass extinction events and diversified into nearly all extant families by the end of the Cretaceous. So, this continued evidence for extraordinary lineage preservation across the order. Diversification rates within Coleoptera were not exceptional, which is consistent with previous studies, although I will point out that the highest rates were recovered along the stem group of the Phytophaga, the leaf longhorn and weevil beetles. In terms of beetle relationships or interactions with plants over evolutionary time, we see on the basis of this data set that herbivorous beetles underwent substantial crown diversification at the same time that angiospermous plants rose to ecological dominance during the Cretaceous. In beetle groups that are associated with gymnospermous plant pollen, some of these plant-beetle interactions, apparently over 150 million years old, were some of the earliest documented interactions between pollinating holometabolous insects and plants. They predate bees and butterflies, and presumably some of these beetles were the earliest pollinators of flowering plants as well. We see a prevalence of transitions from generalized diets like detritivory and saprophagy to more specialized diets like feeding on plants, phytophagy, and mycophagy, or feeding on fungi. This is similar to what I saw in studies published in 2015. So this is results from the Beetle Tree of Life project, where we showed that during the early mid-Cretaceous, there was an extraordinary rise in beetle diversity contributed primarily by plant-feeding lineages, the scarabs, the longhorn beetles, the metallic wood-boring beetles, the weevils, and the leaf beetles. And so that summary, published actually just the year before, sort of looking at these data, we see the evolution of ever more varied and specialized interactions with plants, especially flowering plants, as key to the apparent success of beetles. But it turns out that's not the whole story, and I'll get back to that in just a moment. So I'm going to move on by talking a little bit more then about the extraordinary diversification of this one major lineage that we call the Phytophaga, comprised of the sister superfamilies Chrysomeloidea and Curculianoidea. There are over 125,000 described species in this clade, easily 500,000 or more in total if you include undescribed taxa, an extraordinary radiation exceedingly successful in association with plants. These beetles are primarily specialized plant or fungus feeders, but there's a lot of variation in the ways that they interact with plants. I'm going to focus a lot of my talk, this part of my talk, on weevils because that's where we've done most of our work. Weevils, this itself is an extraordinary radiation, 5,800 genera, more than 62,000 described species. Fossils of all living families are known from the Cretaceous, again, evidence for extraordinary lineage preservation. The chrysomeloids are similarly diverse, more than 7,750 genera, with fossils from the early lower Cretaceous. Of this whole radiation, most striking perhaps in many ways is the interactions between the true weevils and plants. And in particular, this extraordinary radiation has resulted in just a huge number of species. 51,000 or more described extant species in this one family alone. It's arguably as large as the staphylinids. Earliest fossils from the Middle Cretaceous, which is about the same time that flowering plants rapidly rose to ecological dominance. It's also a mess. There is an extraordinary amount of work that needs to be done. None of the subfamilies of Curculionidae, this largest family of plant-feeding insects, is demonstrably monophyletic on the basis of morphological or molecular data. So we have a lot to learn. Extraordinarily diverse, extremely important ecologically and economically, but in fact, most of the groups as we define them are not monophyletic. So we sought to sample very deeply across the phylogeny of the weevils, as well as their sister group, the chrysomeloids, the leaf and longhorn beetles. I'm not going to dig too deeply into these data. We generated a number of different kinds of data, which I'll talk a little bit about over the course of the coming slides. So first of all, I want to share with you a data set that was a bit of a proof of concept. It was based on target enrichment data, from which, though, we captured off-target data as well, which allowed us then to have representative sequence data from as many as 5,085 genes across the exemplars that were sampled. So we recover first, I'm going to show you first, the Chrysomeloidea, sister to the weevils, the Curculianoidea. The Nemonichids were not monophyletic. The anthribids, the fungus weevils, the bellids, adelabids, the leaf-rolling weevils, the carids, which are associated with some gymnosperms in the Old World, the brentids, and then this extraordinary radiation of the Curculionidae. What we showed with this data set is that you can work with large numbers of genes across these taxa, which are relatively more closely related than all of the beetles were in some of our other analyses, and that we can recover very strong statistical support for these relationships. We did some other analyses focused primarily on the target enrichment data. This is a tree based on 522 genes just for the weevils. And here we have more resolution, still nowhere near the large number of taxa we set out to sequence, but another step in that direction. Strong nodal support across the tree. We generated a time tree, which I could compare then to work that I had done many years ago with just a few genes to see what additional resolution and additional confidence we could obtain in the timing of these splits within the phylogeny. So within the weevils, we find most relationships recovered with maximal statistical support, so that's good. We find a couple of interesting groups that had long been debated in terms of their phylogenetic position were placed definitively. In this case, the Ichthyosaurini and Microsaurini were recovered in Brentidae. The Nemonichids were not monophyletic, as I mentioned. The subfamily Cymberidini, the pine flower weevils, we raised or elevated to family rank. They turn out to be sister to all the remaining weevils. So a really interesting early divergent lineage with some implications for interpretation of the fossil record as well. In the family Curculionidae, this one extraordinary radiation within the weevils, we see that most relationships, again, are well supported. Some interesting things going on within that we need to resolve in terms of which groups warrant recognition as subfamilies. The pinhole borers, the Platypodini, were recovered sister to the Dryopterines. That's something I recovered first in 2009. A lot of people didn't believe it, but thankfully we continue to recover that relationship. It does appear to be robust. It's also consistent with some things that we've learned from larval morphology as well. This is work done by Adriano Marvaldi and colleagues. We also found that these were distant from the scolatines, the bark beetles, a group with which they had been thought to be closely related in the past. In terms of weevil interactions with plants, we find that weevils, and this is again consistent with some things we had learned using fewer data, diversified into families almost certainly in association with gymnospermous plants. And they did so during the latest Jurassic and early Cretaceous. The family Curculionidae has early Cretaceous origins, and taxonomic diversification started while flowering plants rose to ecological dominance during the Middle-Late Cretaceous. The ancestral hosts of Curculionidae were angiosperms as well. So this extraordinary lineage, specialized plant feeders, probably the most diverse family of animals on Earth, is deeply rooted in its interrelationships with angiospermous plants and clearly co-diversified with them. What we see within this group, though, is that later arising lineages of Curculionidae are more specialized. They follow the general sequence in terms of increasing specialization of feeding on roots and stems, to feeding on wood, to feeding on leaves, seeds, and so on. And I'll return to that again in some of the work that we've done looking at the genes that it turns out are underlying these specialized plant feeding habits. Now, this is as well consistent with what I saw in 2009, some work that I had done on weevils, where I showed that if you look at this green curve here, it's showing you angiosperm pollen over geological time. And what you see is that 135 million years ago, there's virtually none. All of the pollen is from gymnospermous plants and others, non-angiosperms. But by 70 million years ago, angiospermous plants comprise around 95% of palynological or pollen deposits. So during this time frame, we see the rise of angiosperms to ecological dominance. In fact, this coincides with when we see the origin and early diversification of all major lineages of weevils that are feeding on plants. So again, further evidence for this deep and really intimate interaction between flowering plants and weevils, and in particular, the family Curculionidae. So to move on, I want to share with you what we're learning about other parts of the Phytophaga phylogeny. So within Chrysomeloidea, we're digging deeply into the interrelationships of the leaf, longhorn, and weevil beetles. These two, of course, being associated with plants, very specialized and interesting interactions. So we've generated phylogenomic data and phylogenies, which have been published for the Chrysomeloidea. So far, a backbone only has been published. This based on 541 genes. We're currently studying the evolution of host plant, tissue, and taxon associations, woody plants versus herbaceous plants, different woody plant tissues, different plant taxa, laticifers versus resin canals, and we're also studying things like volatile sex and aggregation pheromones and their evolution across the lineage. We found a lot of interesting things about relationships already. Some of these we have yet to publish, but are consistent with what we're learning based on morphological work as well, in terms of realigning relationships and helping us to sort of rethink the evolution of morphology as well across the lineage. What we see is that the cerambysoid assemblage contains orthosodactyly and the megalopodids. Megalopodids are rendered paraphyletic by the Oxypeltidae, which is a very interesting family associated with Nothofagus in South America, long considered a group of longhorn beetles. We see generally very good support for relationships across this clade. There's a lot of interesting nuance I'm not going to dig into here. It's really only of particular interest to specialists. Again, happy to correspond or talk with anyone about any of this. But what I do want to share with you is the preliminary results of our large-scale phylogeny that incorporates all the data I've talked to you about so far and more. So we do now have data, thanks to the significant efforts in my lab of primarily Dr. Shin, but also some others, contributing data on both the weevils, the leaf, and the longhorn beetle. So we do now have a pretty substantial phylogeny which reconstructs these relationships and recovers a lot of other interesting nuance of the interaction between these beetles and plants over evolutionary time. So my hope is you'll be hearing more about this relatively through papers that have been published or that will be published. Okay, so I'm going to change gears a little bit. So that was sort of a view of where we're headed. And where we're headed with all of that is more extensive, more thoroughly taxon sampled phylogenies, time calibrated to produce time trees or chronograms, which allows us then to evaluate things like timing and patterns of diversification across that lineage in association with plants in a way and with a resolution that's never been possible before. But what I want to turn to now is comparative genomic work, where we've carefully looked at the evolution of specialized plant feeding. So beetles, it would seem, for a long time now, there's been this great interest in their interactions with plants. And their diversity as a whole, the whole of the lineage Coleoptera, has been chiefly attributed to adaptive radiations of specialized herbivorous or plant-associated beetles because of their interactions with flowering plants. But angiosperm-feeding beetles are generally not more species-rich than their relatives with other feeding habits. And in fact, that's also true for herbivorous insects in general and for other hexapods. Now, it does appear pretty clear that angiospermous plants were important to the diversification of beetles. But what's becoming clear by these other analyses that have been done is that there's a little more to this story. It isn't just an association with angiospermous plants that appears to be correlated with diversity and diversification within Coleoptera, because not all plant feeding or even all angiosperm feeding beetle lineages are diverse. Well, what we've come to learn is that the genomes of some herbivorous beetles encode enzymes that can degrade plant cell walls and can otherwise facilitate specialized plant feeding habits, habits like leaf mining, stem, and wood boring. These so-called plant cell wall degrading enzymes are mainly carbohydrate esterases, polysaccharide lyases, and glycoside hydrolases. They enable beetles to digest cellulose, hemicelluloses, and pectin in plant cell walls. In doing so, they liberate sugars, amino acids, and other essential nutrients. And if you're not a plant person, one of the important things to know is that plant cell walls are the Earth's most abundant source of carbohydrates. Being able to eat them and to do so efficiently is an extraordinary innovation. And it's actually something that relatively few organisms can do well. Plant cell wall degrading enzymes are known from 26 beetle species in 5 families, or at least they had been until the work I'm going to share with you today. They were originally obtained by horizontal gene transfer from fungi or bacteria. Again, limited studies, but that's the extent to which we knew it at this point. Gene duplication and functional diversification of these genes after horizontal transfer resulted in multi-gene families with complementary catalytic activities. Basically, if you look at the suites of genes that we know of in these beetles, they complement one another and allow them to break down an extraordinary diversity of plant cell wall components. But keep in mind that they're only known from a small fraction of the 190 beetle families. It is clear, though, we do know that these genes are required by some beetles for successful herbivory. Other insects, like termites and wood-feeding cockroaches, can do similar things, but those metabolic capabilities are primarily conveyed by symbionts that live in them, in their guts or in other parts of the body. So plant cell wall degrading enzymes in beetle genomes are a proposed key evolutionary innovation in the adaptive radiation of phytophagous beetles. The thing is, though, we don't know much about them, or we didn't. Their phylogenetic distribution in beetles and impacts on beetle diversification were virtually unknown until the work that we published last year. So we documented the numbers, kinds, phylogenetic distribution, and timing of first appearance of beetle genes encoding putative plant cell wall degrading enzymes. And we asked whether these were correlated with taxonomic radiations in beetles and shifts in host range or feeding niche. We also studied genes that encode putative invertases. Invertases are cool. They catalyze the conversion of sucrose to glucose and fructose. Now that's important because sucrose is the primary form of photoassimilated carbon in plant vascular tissues. So that's a big deal. Not all insects can do that. That, in combination with the ability to break down plant cell walls, is pretty unique. It is an extraordinary potential innovation. What I'm showing you here is the phylogeny that we previously generated, but I've shown across the top the transcriptomes and low coverage genomes that we mined to do comparative genomic work on these genes. What I'm showing you here, and I'm just going to briefly walk through it, I'm not going through all the details, is the phylogenetic distribution of these genes on the right. On the left, sort of in the middle here in this colorized plot, I'm just showing you a close-up of the phytophaga, which are down here on the lower right. In the boxes, I show you the number of copies of each gene family that are present in the genome or transcriptome of the taxon that was studied. Notice we don't have these data for most because most of these are transcriptomes from which we don't get copy number data. But what we did find that was notable is that plant cell wall degrading enzymes and invertases were found almost exclusively in the metallic wood-boring beetles, the Buprestoidea, up here shown on the right, and the Phytophaga on the lower right. What's cool about this is if you look across all beetles, these are the only two really specialized plant-feeding lineages. And not just that, they're the most species-rich lineages of plant-feeding beetles. So they're diverse and they have the ability to feed on plants in specialized ways that other lineages cannot. I haven't talked much about Buprestoids, so for those that don't know them well, there's more than 15,000 species. They're specialized wood feeders generally. The ancestral habit appears to have been external root feeding. This is from a paper we published about 6 years ago. Leaf mining occurs within the lineage. There's multiple origins, in fact, interesting and specialized ways of interacting with plants. What we see is that the Buprestoids and the Phytophaga encode expansive and remarkably similar arrays of plant cell wall degrading enzymes. They were independently acquired, and so they have similar genes, but with different origins. Most plant cell wall degrading enzyme gene families are represented by multiple homologs, meaning that once these genes made their way into beetle genomes, they diversified. The two major origins of specialized herbivory that we find in beetles are the ones shown in green. They are these two lineages. There are other plant feeders, but they're not specialized. They are not doing the same remarkable things that these two lineages are. What's cool about these two lineages too is that the origins of these genes are coincident in time and in phylogenetic position with the first appearance of these genes in their genomes and with increases in diversification rate. So basically what it seems is that when these genes originated in beetle genomes, the lineages which have them and have a diversity of them rapidly diversified. They were able to utilize plants in specialized and unique ways. We inferred the origins of these genes, basically where they came from, and their origins are in bacteria and fungi. The mechanisms behind the horizontal gene transfers that originated, or where these genes originated, remain unknown. Although we are digging into that now a little bit with the help of a number of collaborators worldwide. So in summary, diversification rate increases are coincident with origins of an array of novel plant cell wall degrading enzymes, suggesting that these genes triggered adaptive radiations that resulted in nearly half of all living beetle species. These two lineages together are about half of all living beetles. The appearance of similar arrays of these genes in the Buprestoids and the Phytophaga are separated by over 250 million years of evolution. This is convergence. These were not present in the common ancestor of these two lineages. Plant cell wall degrading enzymes enabled efficient, symbiont-independent digestion of plant biomass, promoting the evolution of uniquely specialized plant feeding habits in these lineages. What it also further reinforces is the intimate and complex evolutionary relationships between insects, plants, and microorganisms. That latter member of this sort of three-part interaction having been largely ignored for quite some time. So let me just sort of give you a brief summary of what we understand to be the sort of macroevolutionary history then of beetles, in particular in their interaction with plants. So we see the origin of hexapods during the Ordovician around 480 million years ago, flowering plants more or less the same time, or excuse me, plants more or less the same time. So you have hexapods and terrestrial plants emerging from, if you will, marine environments at the same time, more or less, co-evolving then throughout the history of their terrestrial life on Earth. Coleoptera appearing around 150 million years later during the Carboniferous, at the same time as gymnospermous plants. So again, a deep history of interaction here between these lineages as well. Beetles living through the end Permian mass extinction event. The first dinosaurs appearing 100 million years after the first beetles. The first flowering plants appearing 200 million years after the first beetles, around 160 million years ago. Angiosperms rising to ecological dominance in such a way that they've now occupied about a third of the evolutionary history of the beetles. And a mid-Cretaceous increase in diversity and diversification rate of plant-feeding beetles. So as flowering plants rose to ecological dominance, beetles diversified along with. But keep in mind that beetles did not diversify rapidly at the same time that angiosperms first appeared on the scene. There was a lag between the origin of angiospermous plants and the diversification of angiospermous-associated beetles. It would seem it was not until angiosperms were an ecologically relevant, if not dominant member of the flora, that beetles diversified on them. The Chicxulub asteroid impact and end Cretaceous mass extinction didn't have a huge impact on the higher level lineages of Coleoptera, although no doubt eliminating many lower level taxa. During the Paleocene-Eocene thermal maximum, and this is something I've written a little bit about, we see a rapid increase in diversification across beetle lineages at lower taxonomic levels, below the level of family or even subfamily. And then, of course, we're now in the midst of the Anthropocene with really remarkable impacts on beetle diversity. And one thing I like to point out to audiences is that there are beetle families now on the brink of extinction, maybe even that have gone extinct in very small isolated areas of Australia, that have been on Earth for literally 150 million years. This is a group at the ordinal level which has exhibited remarkable resilience over evolutionary time, but is in a position now where we are losing higher level lineages, and potentially will lose quite a few in the near future. So in conclusion, beetle diversity apparently resulted from multiple factors: a low rate of lineage extinction over a long evolutionary history, co-diversification with angiosperms and in the novel habitats they created, an adaptive radiation of these very specialized herbivorous beetles in association with all kinds of plants, not just angiosperms, by the way. These genes facilitated all kinds of plant feeding, and this was following the independent origin of these plant cell wall degrading enzymes and invertases from microbes via horizontal gene transfer. So I'd be remiss not to dwell quite a while on my colleagues who made this work possible. I want to specifically mention Rolf Beudel, Bernard Misoff, Adam Slipinski, and then especially Dr. Seung-Hwan Shin. Dr. Shin figured prominently in almost all of the work that I presented to you today. We continue to collaborate. He's a wonderful friend and colleague. I also want to thank the U.S. National Science Foundation and the United States Department of Agriculture for funding for this work. I'm excited that we're continuing a lot of it. We have really what I would consider very exciting new work going on at that interface between, if you will, beetle macroevolution and comparative genomics. So I enjoy and look forward to sharing more of that with you as that gets published. With that, I also need to thank collaborators in several large consortia, the One Kite Project, Bernard Misoff, Xin Zhao, and many others who have played a very important role in allowing me access to data, elaboration, and establishing new analytical pipelines, and so on. The 1K Weevils project and my collaborators on that project, the Insect 5000 Genomes Project, and many friends and colleagues that have shared their expertise, knowledge, and data in the context of insect genomics. So with that, I want to stop there, and I'm not sure if there's time for questions, but I'm certainly happy to field them if there is. Thank you.
Thanks so much, Dr. Mekana, and thanks so much for the great talk. And I apologize for the bad connection. I think you did a great job for just continuing our talk, your talk. So I think we have about 5 minutes, so we can have maybe 1 or 2 questions.
Okay, great.
If there is no question, may I ask you one?
Of course.
Yeah, it's kind of about that I'm asking question because most of what we did together. But I would like to know what is your next plan? Like you resolved the beetle phylogeny with the most robust tree, and we found really great example of the evolution. And I would like to know what is your next plan about the beetle phylogenomics and evolution?
Well, I think the exciting place where we are now is we're able to add more taxa at lower levels, which brings us much closer to biology. When you're working at such a high level, the patterns that are so fascinating as a natural historian are sometimes a little bit lost. But when you start to sample very, very deeply, for example, within the longhorn beetles or the leaf beetles or the weevils, you start to see fascinating patterns of host association. For example, within the longhorns, remarkable transitions in host association by nature of shared morphological defenses in plants. So plants with resin canals, for example, being colonized more readily by a given lineage that's good at that than plants that might have similar chemistry. And there's qualifiers around all of that, but it becomes really very fascinating. So what I'm enjoying doing now is we are sampling much more deeply within some of these lineages. The other thing is, I think we can all relate. Many people have focused on conspicuous, well-known lineages when there's quite a few things out there that have been ignored that have really interesting biology, some of which are ancient lineages, isolated lineages as well. And when we include those in the phylogeny, they inform considerably our understanding of the evolution of the lineage. So we've been finding and including a lot of those groups as well. And then the last thing I'll share is that as we get closer to biology, we're also able to generate genomic data. That genomic data then allowing us to sort of come full circle, right, to understand the genomic basis of these remarkable biological traits, which have interested us since we were children, right? I mean, why is it that there is such striking morphological variety and variation? How is it that they feed on so many different kinds of plants? So those questions become much more satisfying to address when you have the genomes, really, if you will, that underlie the biology. So we're doing a lot of that, generating genomes, transcriptomes, asking questions which are effectively comparative or evolutionary genomic questions about pheromones, about host associations, and so on. So a long answer, I apologize, but that's the fun things that we're doing right now.
Great. Thanks so much. Any other question?
Yeah, just thank you for the nice presentations. Just to cut a short question about the fossil. And when you're making the phylogenetic, trees or the evolutionary, the trace, I mean the calibration using the fossil. In case of the beetles, is it does it help to analyze these things, compare with the genomic approach, and also using that fossil probably help. But what is the conditions? How can you, using those kinds of where the beetles coming, the fossil beetles coming?
Let me know if I don't fully answer your question, but as I understand what you're asking, how is it that we do this? What are sort of best practices, and maybe how is it that we specifically do it?
Yeah.
One thing I've found is that most studies don't pay adequate attention to evaluating the fossils that are used. And so what I have found is that by absolutely and thoroughly revisiting the fossils that are proposed to be used anyway, really thinking about which ones we think are best to be used, determining whether they meet certain specific criteria before applying them, not just leaning on published papers that say they are what they are and they're as old as they're supposed to be, but rather looking at the fossil when we can, actually getting it in hand, or at least looking at the original description and determining whether it's adequate to answer a number of important questions about the fossil before we use it is critically important. And it seems really obvious that that's what one would do, but the reality is that most workers, and I won't single anyone out here, but the reality is most of our peers don't do that. And so the real benefit of it, of course, is that without even generating any molecular data, by having a really robust fossil data set, we can often significantly inform our understanding of the macroevolution of these groups. And as a great example, within the weevils, we have found very clearly that a specific fossil that has been widely used, that's in amber, is misidentified as being a Burmese amber fossil, and it is not. And when we learned this both by looking at fluorescence of that fossil, and Dave Clark, a postdoc in my lab, carefully studied that fossil, we've shown definitively it's not even in the deposit it was thought to have been in. And it has really important implications for minimum and maximum age constraints within, in this case, the curculeonity. I'll stop there. I hope I answered your question. If you have a follow-up, I'm happy to try to address it.
Okay, thank you.
Okay, so I think it's time to move on. Thank you so much. And we will start the next keynote to speak. The next speaker is Dr. Ho Jun-sung. He is currently Texas A&M University professor, and he will talk about the Orthoptera tree of life, elucidating patterns and processes of Orthoptera evolution. Okay, so please share the screen. Yeah, is the setup okay? Okay, yeah.
All right, so thank you for the invitation. It's actually, it's great to be invited to this symposium. And I also like to share that it's fun to be with Dwayne. We were graduate students at the same time. We were this postdoc at the same grant, and then we have a parallel career going on. And then now we have relationship with Seung-Kwan, and we have we're sharing a stage together. So it's interesting. 그리고 한국말로 좀 할 텐데요. 지금 제가 하는 이 발표는 그 드웨인이 듣기 때문에 영어로 하겠지만 초대해 주신 거 너무나 감사드리고, 그리고 제가 하는 발표를 통해서 또 여러 가지 많은 그 새로운 공유하는 길이 열릴 수 있었으면 좋겠습니다. Then I'll start. So today I am going to talk about the Orthoptera tree of life, elucidating patterns and processes of Orthopteran evolution. The way I am going to talk about this talk is, I have structured this talk is a bit different from how Dwayne has done for his work, because I like to go into historical aspect as well as highlighting the diversity of Orthoptera. So first, we are going to talk about the diversity of Orthoptera, and then I am going to talk about brief history of Orthopteran systematics, and then move on to the phylogenomics of Orthoptera, and then the evolution of acoustic communication, which is well known in the order Orthoptera. Orthoptera is the most diverse lineage within group Pauliniaptera. Of course, compared to beetles, the number of species in Orthoptera is minuscule. We have about 28,000 species known. However, it is the largest and most diverse lineage within Pauliniaptera, which includes various lineages like walking sticks, cockroaches, and termites, and earwigs, and so forth. And Orthoptera is a group that I have been very interested for a long time, ever since I was little. and now I'm pushing through the phylogeny of Orthoptera. And a lot of people are familiar with Orthoptera. Grasshoppers, crickets, and katydids are the ones that we grew up with. When I go to different countries, people know what grasshoppers are, what katydids are, not only because they are familiar, but they are oftentimes agricultural pests. However, not many people understand the diversity of Orthoptera. And here, whenever I talk about Orthoptera diversity, I like to sort of highlight how diverse they are, how strange they are. And other than the things that you're familiar with, there are some things that are super weird. So let me just highlight those different groups. Orthoptera has two suborders, Ensiphra and Sellifera. Within Ensiphra, we have a superfamily Grilloidea, in Korean, 귀뚜라미 상과, and we have several different families in there. This is one of the larger superfamilies within Orthoptera, and crickets are found everywhere in many different habitats, in caves and in different parts, and they're well known for their ability to sing. Gryllothalpoidea includes Gryllothalpids, which is a subterranean species, as well as Myrmecophilidae, which lives inside the ant colonies. A pretty interesting group. Fairly small superfamily, but interesting nonetheless. And then we have Stenopelmatoidea, which includes several Gondwana-land distribution lineages, including Grillocritids and giant weta from New Zealand and Jerusalem crickets and such sort of really interesting groups. And this group is not known to sing at all. However, they have interesting biology and they have defensive signaling. And then we have Rhaphidophoroidea, or camel crickets or cave crickets that are found all throughout the world. And then we go into some of the obscure small groups that are found in different parts of the world. And in South Africa and in the Middle East, we have Schizodactyloidea, or splay-footed crickets, which are carnivorous cricket that is found. Not much is known about them, and only 16 species known. Hageloidea is another group that is found in the cold northern temperature in northern North America, Russia, China, India. And this is commonly known as ambidextrous cricket. They are able to use their wings left or right, or right over left to create sound. And is known a lot from Jurassic fossils. So these are considered living fossils. And then, of course, we move into Pterigonioidea, which includes katydids. Katydid is a single family, but it is the largest family within Ensiphra, and they're everywhere. And they're well known for their crypsis, as well as their ability to sing and so forth. Very familiar group. Then now we move on to Sellifera, and then we have a small group known as Tridactyloidea, the familiar insect. They're found in riparian region, tridactylids, and there's some ones that are found in South America, in the tropical region. And then this weird-looking, kind of worm-looking thing is a group that is endemic to Gondwana land and found in South America as well as Australia. It's completely subterranean, lost its jumping leg, and so forth. And then we have Tetragoidea, and this is one family. It's about 2,000 species, and the familiar form is this, with the elongated pronotum. However, there's enormous diversity in the form of pronotum, and I view this as a kind of membracity of Orthoptera. Pretty interesting group. And then we have a Eumasticoidea, which is commonly known as monkey grasshoppers, and I'm not really sure why that name came, but the face looks like a monkey. Very colorful, found throughout the neotropics, and one of the least studied groups within Orthoptera. And then also within South America, related to the monkey grasshoppers, we have a jumping sticks. It looks like a walking stick, but it's actually grasshopper that can jump. And then, just like Ensiphra, we have some obscure relic groups. Within Sellifera, we have some relic groups, such as this desert-dwelling Tenosauridae, which is found in western U.S. And then bladder grasshopper Dumorix, they're found in South Africa. And then here we see a female, but male has an enlarged abdomen to create sound. And then we have some other relic groups found in Southeast Asia as well as Mexico, called the Triglopterygoidea. And then we go into a bit charismatic group. Here is a Pergomorphoidea, which includes Pergomorphidae. And they are large, showy, very colorful, oftentimes feed on milkweed and toxic. And they're always featured in large insect collections as a showy insect. And then finally, we have grasshoppers that are found throughout the world, and the group that I'm most familiar with. So I'd like to take a moment to actually talk about the history of Orthopteran systematics. Although this symposium is about the global collaboration, I think it's important to trace back and think about the history of our discipline, our taxon-specific discipline, in terms of how we got to where we are. If we actually think about the species description trend over the past 250 years since the publication of Systema Naturae by Carlos Linnaeus, we see that the number of species described per decade kind of goes up. By 1800s, we have one family of Orthoptera known, and then there's a proliferation of a species description and understanding of the diversity. By 1900s, we have 26 families known. But then, there are world events. So we have World War I and World War II, where the number of species description went down because people were suffering. And then eventually by 2000, we have 42 families recognized, which is the number of families within Orthoptera that we know today. However, this interesting part here is a drop. The number of species that described in the last decade has dropped significantly from the past decades. And that is not to say that we have described all the species. This is a reflection of the loss of taxonomic expertise. So this is a signature of taxonomic impediment that we're facing today within Orthoptera. Of course, when we do a cumulative line of number of species described, is going up and has not hit an asymptote. So it is possible that there are more than 30,000 species of Orthoptera, and there are many more species to describe. And there are regions that we have not explored, like Southeast Asia, Western Central Africa, and so forth. And also places that we have explored quite a bit, still we come up with new species. There are still more species to discover. And it's actually an interesting exercise to go back and see who's who in Orthopteran systematics. Early in the 1700s and 1800s, European taxonomists, these were museum taxonomists, they studied the specimens collected by the explorers when these countries, these empires sent out explorers to Africa and Asia and different parts of the world and collected specimens. So these are not actually explorers. These are museum scientists started to discover and describe species. And then later on in the sort of late 1800s, people started to go out and describe. But it's interesting that these guys are still kind of stern-looking taxonomists or European white males. And that trend continued. And later, different scientists started to go to different parts of the world and describe more species, and that corresponds with the number of new species that were described in late 1800s and 1900s. And then we move into what I refer to as the period of geographical specialization. Within North America, we have two prolific taxonomists that went out and collected and described a bunch of species throughout North America, and we find that in Africa, Europe by Europeans, and we found that in Europe and other parts of the world as well. And then later, once we have described a lot of general diversity, there's a period of what I call the taxonomic specialization, where rather than knowing about all the different groups, now different scientists are specializing on certain groups. So Sir Boris Suvorov on grasshoppers, Dr. Lucien Chopard for crickets, and so forth. And then we have this giant of 20th century, and in the middle of 1900s, 1950s, 60s, 70s, we have a renaissance period of Orthopteran taxonomy. And these are the big names in the Orthopteran taxonomy who describe many species and established the current taxonomic classification and the phylogeny of Orthoptera based on museum specimens. And then these are people who are still alive. Dr. Carbonell passed away last year, but all the other people are still alive, and they're in their 70s and 80s. However, many of these scientists were museum scientists or curators and did not have the ability to train the next generation of taxonomists. So what happened was that drop in the number of species described, because no new taxonomists, next generation taxonomists were not trained. However, there were several other groups. Some of them, this lore from Paris Museum was trained by, for example, Marius de Camp and Amagnaro and so forth. They're now these are the active scientists who are in their 50s and 60s, who are doing active work. And then now we have a younger crop of scientists, taxonomists in their 30s and 40s. And what you can see is, in the old days, it was kind of European white male, and now we actually begin to see the diversity of systematists in different parts of the world, focusing on their region in South America, in Asia, and so forth. And we are in the process of generating additional new generation of Orthopteran taxonomists for the future. Let me just have a quick moment to talk about what I do in my lab. My lab is the only lab in the U.S. That can train Orthopteran systematics and taxonomists, which is kind of unfortunate because it's a fascinating group, charismatic, agriculturally important. But when I was trying to get my PhD, there was nobody that I could go to get trained. So I took that as a mission for me to train the next generation of Orthopteran systematists, and that's what I've been doing for the past 10 years. When we start to study Orthoptera, we have to actually look at it globally. So I do a lot of expeditions around the world with my students, trying to transfer my knowledge, and I also learn from my students as well. Here's a picture with my students and my family. We went to Costa Rica for studying. And then recently we went to Namibia try to find this obscure species. We couldn't find it, but it was a fun expedition as well. Here we went to South Africa, and we have another NSF project to look at the diversity of grasshoppers on top of the Drakensberg escarpment in South Africa. So this was a kind of fun study where we took helicopter ride up to about 3,000 meter high and study wingless grasshoppers that are found only on the top of these islands, these mountaintop islands. And then I've done a lot of work in Australia, and here we're actually retracing old taxonomist collecting route, tried to see what happened over the past 70 years due to climate change, did the grasshopper composition change and so forth. So we're doing a lot of international collaboration with different experts around the world. Of course, we can talk about the students that I've trained and the advances that I have made, because there's not many people who work on orthoptera and systematics, especially using molecular data. I've been pushing for training students to become the expert in next generation. So one of the earliest work that I've done with this was to look at the phylogeny of Katydis and Pterygoniidae, and then which resulted in a number of papers looking at the phylogeny of Katydid. And what we find is that just like what Dwayne has explained for Curculionidae, most of the subfamilies within Pterygoniidae are paraphyletic. So there's a lot of work to be done as well. Pergomorphidae is a small group. So less than 500 species known, but they're fascinating group. And my student, Ricardo and Salo, they've been working together to increase our knowledge. And especially for Ricardo, what I've done is, before he joined, there was absolutely no expert in the world who could identify these species, this group. So he spent many years learning the diversity and the taxonomy of this group, which resulted in the first morphological phylogeny. And then we moved on to using molecular phylogeny to understand its biogeography. So mostly an old world group, but now there's some new world species. And we studied based on mitochondrial genome data and tried to reconstruct the biogeographical history of this group, and found that there are multiple invasions from the old world to the new world to give rise to the diversity within the new world. One of the interesting things about this family, Pergomorphidae, is that these are colorful. And when you see something like this, and you say, oh, they might be toxic. It is true, they're aposematic, and they feed on milkweeds, Apocynaceae plants. And interestingly, this is a collaboration with Peter and Derfato, that there are a number of different insect groups that have evolved insensitivity to cardiac glycoside, which is found in milkweed or cardinalides. And so there are beetles, there are flies, and there are lepidopterans, and there's some hymenopterans and hemipterans that have convergently evolved the insensitivity to the cardinalide. And what's really fascinating about this is that they have an enzyme, ATPase, which has a mutation in the same amino acid position independently in the same mutation. So this is a convergence at the molecular level. And we've known about these for many years, but nobody had bothered to look at whether Orthoptera had the same type of conversion molecular evolution. And we looked at several of these species, and we found that, yes, those species that feed on toxic plants, feed on the milkweed, indeed have the same type of mutations in the same amino acid. So this is an amazing finding to have this more molecular level convergence for in different orders. Of course, we push through the phylogeny of grasshoppers, and I'm not going to spend too much time on it, but grasshopper is my favorite group. I've studied them for a while. They are youngest group within Orthoptera. They diversified within the Cenozoic, and the diversification seems to coincide with the diversification of the grass. And what we find is that the modern grasshoppers that we know today, many people used to think that they came from Africa, but what we definitely showed was that they actually originated from South America. And then because all the the early diverging lineages within Acrididae are endemic to South America. So we came with a bunch of biogeographical scenarios to explain how the modern grasshoppers got to be where they are today. And this is a large group, and this phylogeny, although it looks, it looks big, it's a small fraction. There's a lot of work to be done for this phylogeny of grasshoppers and understanding their diversity, camouflage, ecomorph evolution, and so forth. So there's a lot of work to be done for this group. Now, I'd like to shift to the talk that is a paper that was recently published in Nature Communication, looking at the phylogeny of Orthoptera as a whole, and try to use that phylogeny to understand how hearing and sound production evolved in Orthoptera. So, acoustic communication, ability to use acoustic signal for communication, is widespread in nature. We see that in mammals, we see that in birds, we see that in amphibians, and of course, we humans use our sound, our voice to talk to each other. However, the most diverse lineage within insects, within the animals that use acoustic signaling is the insect. So here's a kind of rough phylogeny of insects. And when we look at the distribution of the organisms that are able to hear using tympanum, we see that the tympanal hearing has evolved multiple times independently throughout the phylogeny of Orthoptera. And tympanum is found in different parts of the body in different groups. Now, when we think about sound production, and here I define sound production as a far field sound production, ability to produce sound that is audible. And I'm not talking about vibration, which is also quite prevalent across many different groups of insects. Here we find that some groups have ability to hear and ability to produce sound, and other groups have just ability to hear or only ability to produce sound. When we look at, and then of all these groups, Orthoptera is the most diverse group. There are 28,000 species of Orthoptera known, of which about 16,000 species are known to use acoustic signaling. That's more than the number of mammals, more than the number of birds, more than the number of amphibians. So it is the largest and most diverse group of lineage with an animal that use acoustic signaling. When we look at the history of the acoustic communication, here when we look at the fossil records, we find that Orthopterans are actually one of the earliest diverging lineages found in the Triassic period that are capable of producing sound for mating purpose. There were other groups that produce sound that maybe as a defensive signaling, but actual stridulatory coursing for the mating purpose, Orthoptera is known to be the oldest. When we talk about the evolution of acoustic communication, we actually have to think about the components that make acoustic communication possible. And though that's a sound producing organ, and for Orthoptera, it's a stridulatory organ, and then hearing organ, which we call ears or tympanal ears, right? Mm-hmm. So within cricket, this is a tree cricket. Here's a stridulatory file and a scraper on the wings, and then there are two tibial tympana here. When we think about these two organs, and these two organs must interact with each other. The signal sender must produce the sound, and the receiver must have the ability to hear the sound. And how did these two organs, two interconnected organs that are involved in acoustic communication evolve. And there are two different hypotheses put forth by people who study sexual selection and acoustic communication, or communication in general. One hypothesis is that it's based on sensory bias, which means that, for example, here the cricket or any early insect could have evolved some ability to produce sound in a different context, for example, for defense and so forth, and then could have evolved hearing in a different context to detect predators and so forth. Now that you have evolved these structures, it could be co-opted for another purpose, and therefore you can evolve acoustic communication. Another competing hypothesis is what's known as Fisherian coevolutionary mechanism, where the hearing organ and the sound producing organ actually evolved together, and they co-evolved because these are important for communication. So these are two competing hypotheses, and they have been put forth by various scientists, but we didn't really have a good way of testing that until now. And here we have just to show you the diversity between different groups. I'm not sure if the sound comes out, but if not, that's fine. Crickets, as you can see, can produce sound by rubbing their wings together. And then grasshoppers do so by moving their legs up and down. And then there are some groups like the Jerusalem crickets that can drum their abdomen on the surface. So these are the ability to produce sound. In terms of hearing, the encephalons have a tibial tympanum, and on their front tibia, and there's a membranous part, but inside there's a structure that is modified from a subgenual organ, which is for detecting vibration, and then there are some additional components that allow to process acoustic signaling. In grasshoppers, some grasshoppers, there's an abdominal tympanum in the first abdominal segment, and different parts of that tympanum is tuned to different sound and so forth. However, within Orthoptera, there are also some non-conventional ways of producing sound and hearing. Here is a bladder grasshopper from South Africa, and the males are able to rub the hind leg against the abdomen to produce sound, which amplifies by this enlarged bladder, and they produce low frequency sound that can travel up to 2 kilometers. And interesting thing is that they do not have ears. In fact, what we find is that each abdominal segment has a sensory organ, so entire abdomen functions as a ear. So this is an example of atympanic hearing. And then there are some other groups that we know little about, but they have structures that suggest that they are able to produce sound. For Pamphagidae, we have structures that are known as Krauss's organ that allow them to produce sound by rubbing legs against the abdomen, but they also have various other ways of producing sound. We have this sand groper from Australia, and then between mandible and maxilla, there's this stridulatory file, and nobody has been able to demonstrate that they were used for producing sound, but structures are there. Even things like tridactylid, which is pretty common, underneath the forewing, you have the stridulatory file, but nobody has recorded this. So there's a lot to learn about the diversity and the mechanism of sound production and also hearing. There's a giant weta from New Zealand and Australia, and these guys do not have wings, so they do not produce sound by rubbing the wings. However, they have the sugatory file on the abdomen, which they rub their hind leg against to produce defensive sound and so forth. So try to understand the evolution of acoustic communication. What we need is a robust phylogeny, and that has been actually kind of difficult for Orthoptera, mainly because not many people studied Orthopteran phylogenetics using molecular data. Earlier study was by Fluke and et al. In 1999, very small representative taxa, and there's some morphological work. And in 2015, I published a larger phylogeny based on mitochondrial genome data, which was nice. However, the backbone relationship was poorly supported, so it was really hard to understand the polarity of how these structures could have evolved. In 2014, there was a larger, there's a kind of major paper that was published as a cover of Science, which is years of work by a consortium known as Kite 1000 Insect Transcriptome Evolution, which we heard from Dwayne's talk. And so I became involved in this kind of late. Original One Kite team did not have a specific project on Orthoptera. I've been working on Orthoptera, but I was not part of the team before. However, in 2015, I went to Germany, addressed the meeting of insect phylogeny, which was a really nice meeting on kind of cutting edge development of insect phylogeny, where here's me, here's Dwayne, and but several other people. And this was a year after 2014, after a Science paper. So there are a lot of talks about One Kite, and I became so impressed with the promise of transcriptomic data and phylogenetic data for resolving the backbone relationship. So there we started to collaborate, and then end result is a paper that just came out a few weeks ago in Nature Communication, where I'm the first author, but Dr. Shin is part of it, as well as Dwayne, as well as several people from the One Kite Consortium. So I'm not going to talk too much about the analytics, but I'm just going to dwell on the results. Here is a phylogeny, a time tree based on the transcriptome data. It's a rather small taxon sampling, about 5,000 loci. And then here we have, there are a lot of Orthopteran fossils. And there are a lot because there's a lot of fragmentary wings. However, many of these fossils were not vetted. In fact, in 2015, when I tried to publish it, I did what most people do. I went to the literature and found some fossil evidence, but this time I wanted to do it right. So I collaborated with a paleontologist, Olivier Betu, and he went through all the fossils, and it actually came up with just 11 fossils, including several of them from Paulneoptera, to have a calibration. These are the fossils that he was able to, he confidently say that these are good calibration point. So we performed the divergence time estimate analysis, and here is the tree. Of course, we have a monophyletic Ensiphera and monophyletic Cellifera, and the relationships are very resolved with 100% bootstrap support in all the clades. What we find is that Orthoptera originated in Carboniferous about 350 million years ago. And that's a bit pushed back from the previous estimate, which was about 300 million years ago. And then both Ensiphera and Cellifera diverged in the Permian period. And then the crickets started to diversify from the Triassic. There's a long time ago. So of the living lineages of Orthoptera, crickets have been around for the longest time. And as you'll see, it's important to think about the the context they evolved, and the evolution of acoustic communication, and how ancient this mode of communication is. Katydids diversified and diversified in the Cretaceous period and on. And here's a classic example of what we call the Cretaceous terrestrial revolution. There's an explosion of the angiosperm and trees, and then diversification of katydids, and the mimicry and so forth. Grasshoppers, their latest arrival in the phylogeny of Orthoptera. Grasshoppers diverse in the Cenozoic, and then diversified, and it's a kind of rapid adaptive radiation that we see within Orthoptera. On the transcriptome data, and using that as a backbone phylogeny, we tried to increase the taxon sampling by adding mitochondrial genome data. And in the beginning, I was kind of not sure how this will play out, because transcriptome data has so much data, and then mitochondrial genome data, so little data comparatively. However, when we did this analysis, we were pleasantly surprised that the backbone relationship stayed, and then mitochondrial genome data were able to basically fill in the blank and reveal the relationship internally. So this was a kind of good exercise, and using this as a reference phylogeny, we are now able to understand the evolution of sound-producing and hearing organs throughout the phylogeny. So what I've done is, of all the in-group samples, I've gone through the literature, I've looked at the specimens, and tried to code the characters, the behavioral characters, the morphological characters important for sound production and hearing. So what I have done is a ancestral character reconstruction, and what we find is that here we have the evolution of what I call tegmino stridulation, which is ability to move the wings together to produce sound, appears to be an ancestral condition for Ensiphera. For crickets and grillotalpids, the right over left stridulation evolved. And then for katydids, left over right stridulation evolved. In the middle, there was one loss in the camel crickets, actually another loss in the myrmecophilids, but this has to do with the association with ant colony, and then these guys basically lost the ability they probably lost the developmental genes for the producing wings. And then Stenopelmatoidea, many of them do retain wings, but they have lost their ability to sing, and then they do not have stridulatory file on the wings, but instead have evolved the ability to produce sound by rubbing their legs against the abdomen. And if you think about this, how old this is, we are thinking about the anciency of this group, which we estimate to be a Permian, about 300 million years ago. And when we look at the Permian fossils of stem Orthoptera, there are actually several groups, several fossils that indicate that it might be able to produce sound. Here's a fossil of a Titanoptera. Here's a fossil of a sort of stem group Ensiphera. And we have examples of males having stridulatory file and females having none. So there is sexual dimorphism already present in the Permian and Triassic period. And then there are different lineages that have that use different wing veins to produce sound. So there's kind of diversification of structures that produce sound in the Permian and Triassic period. So what we infer is that the sexual dimorphism, the male having the ability to produce sound and female not, was an ancestral state for Encephora. When we think about the hearing organ, when we do ancestral character state reconstruction, at the base of phylogeny, there's a small probability that all the ancestral Encephalans could have evolved the ability to hear using tibial tympana. However, what we can definitely say is that the common ancestor of crickets evolved it, and then common ancestor of katydids and haglids also evolved it, as well as some other wettas and so forth. So it could indicate that there's a multiple evolution of a tibial tympana, but when we actually look at the position of these, the way it's positioned is on their front legs, and this is kind of awkward place to have ears just to hear the sound of the predators. In fact, the position here is really well suited for directional hearing. So the modern crickets have the tibial tympana connected by the trachea, which is adapted for hearing, and then there's a thoracic spiracle that is modified as acoustic spiracle. So this ability to hear sound and locate the source of sound seems to be coinciding with the ability to find mate. So the directional hearing evolved earlier on. When we actually look at the internal nervous structures for creating ability to hear, you find that there are actually two independent evolution of hearing. One is a modification of a neuron to create what we call the tympanal organ. And then in katydids and wettas and haglids, we have a crista acoustica, a sort of third component for hearing, processing the sound. So all in all, what we find is that it appears that there's actually a coevolution going on. So strigotrophiles and tibial tympana likely co-evolved in a sexual context early on in the phylogeny Encephora, going back 300 million years. So this pattern corresponds well with the Fisherian coevolutionary mechanism for understanding the evolution of hearing and sound production in Encephora. However, when we look at Cellifera, we have a completely different pattern. What we find is that when we look at sound production, different groups have independently evolved different ways of producing sound, which is quite contrasting with Encephora. Encephora, we have basically two modes of sound producing by rubbing their wings against each other or rubbing their legs against the abdomen. Here we have different morphological parts that are involved in sound production independently. What's also interesting is that most of the early diverging lineages within Cellifera don't have ears, right? They do not have the ability to hear, and they still diversified. The plot is a bit deceiving, but we have a couple thousand species of tetragids and a couple thousand species of eumastacids that are fairly diverse. That are successful, but they do not have the ability to hear. Hearing evolved in the common ancestor of Pergamorphoidea and Acridoidea, and that it evolved once at the common ancestor, and because right now the ancestral character state reconstruction shows that there's a small probability that it evolved once in the first abdominal segment, but it is likely that it is true. The position of this is quite interesting, and when people try to explain the origin of the hearing, they usually say, possibly is to detect predators and so forth, and that could be possible. However, there's a strong indication that these two lineages, common ancestor, was the first large-bodied grasshoppers that evolved with a strong ability to fly. And all these other things are either wingless or they're weak flyers. So these are first larger things. And then the abdominal tympana actually plays a really important function in hearing the wing beat frequency. So they are important for flight modulation. So we think that the first ears in grasshoppers probably evolved in the context of flight modulation rather than detecting predators. Once you have ears, then it could have been co-opted for some other, co-opted later for acoustic communication. So in this case, in Celifrons, hearing evolved first in a non-sexual context, then later co-opted for mating purpose. So in this case, sensory bias mechanism is probably the likely hypothesis for explaining the evolution of these two structures. One of the interesting questions now that you have a phylogeny, you have a time tree, you have a number of species, is to ask, did the lineages characterized by acoustic communication experience increased rates of speciation? As you know, characters under the species that show sexual selection and characters that are under sexual selection tend to evolve rapidly. So perhaps that have an important role in increasing the rate of diversification. So we did a BAM analysis, Bayesian analysis of macroevolutionary mixture. And what we find is that there are two episodes of increased speciation rate within Orthoptera, one found in katydids, one found in pamphagid grasshoppers, and one found in the grasshoppers. And then here, the line are the ones that use acoustic communication. Here, crickets and grigs and katydids, grasshoppers, and pamphagid grasshoppers. So what we find is that there's no one-to-one correlation between increased rate of diversification with ability to produce acoustic communication. And there are some other possibilities for example, katydids, the ability to evolve mimicry and camouflage could be a major innovation that might have driven the evolution of diversification and high speciation rate rather than acoustic communication. So, and then within grasshoppers, not all grasshoppers can sing, only a small number, but we don't see a particularly large speciation rate here, meaning that there's kind of little correlation between speciation rate and then ability to produce sound. So in conclusion, what we find is that the evolution of hearing and sound production in Orthoptera shows complex lineage-specific patterns. Within Ensiphera, we find that hearing and sound-producing organs co-evolved early in the diversification. Within Celifera, we find that hearing evolved first in a non-sexual context, but then later co-opted for mating when sound production evolved. And then evolution of acoustic communication did not increase the diversification rate in this Orthoptera. So what's next? So now that we have this phylogeny, we have published a nice paper. So what's next? The number of species that we used for this study is less than 250, which is, and then there are 28,000 described species of Orthoptera, a very small percentage, 1% of the known diversity, and we are inferring the evolution. So it is important that we increase the sample size. And recently, we have received NSF grant, National Science Foundation grant, to push this field forward. So Dr. Shin is part of this as a co-PI. And also, not only the phylogeny, but we want to try to incorporate biomechanics and biophysics and functional genomics to really push forward. So this is a Fernando Montealegre
here Nathan Bailey and Mike Whiting, and we have come up with a plan to target 1,600 species of Ensiphera, about 10% of the known diversity, and then using over 1,000 genes that can be generated using anchor hybrid enrichment to really have a robust phylogeny of Orthoptera. And then using that phylogeny to trace the evolution of hearing and sound production. And what we can do is using the phylogeny and using the stridulatory file morphology, we are able to reconstruct and model the sound so that we can not only look at and estimate the sound that can be produced by many of these singing extant species, but we are able to also model the sound that could have been produced by the extinct species from the fossil. We're also trying to understand what are the important genes for developing hearing ability. And we know that there are about 70% of katydids can sing at ultrasound and hear at ultrasound. So maybe there's important mutations in the genes related to hearing that might have promoted the diversification of that group. Also, there are some crickets that are found in Hawaii that have lost their ability to sing because of the selective pressure from parasitic flies. And using that as a model system, we try to understand what are the genes responsible for creating these structures that produce sound. And this is a very exciting aspect of the research that we're going to push forward for the next 5 years. Just a couple last things. Across among different insect groups, Orthoptera so far has fallen behind in terms of generating the new tools for molecular phylogenetics. Compared to beetles, Dwayne is leading the effort. There are researchers working on Lepidoptera, researchers working on Hymenoptera, and researchers working on Diptera that are pushing forward, and we're doing the same thing for Orthoptera. And we have developed these tools for generating over 1,000 genes to broadly resolve the relationship. And here's a kind of preliminary phylogeny part of it for Ensiphera. And here are the number of genes that we are using to get at this resolution. Another very interesting aspect of these new tools, which is based on the target enrichment, is that it is not only able to resolve this deep relationship, but it's able to resolve species relationship. So here's another study that we have done using the phylogeny of Stenopelmatis, which is a Jerusalem cricket. Across different species, there are very close related species, but we're able to use the same tool set to resolve the relationship. So it's a very versatile tool. Now the issue is the taxon sampling. Now I think we have developed a robust tool set to generate large phylogenetic data. So I'm very excited to push this research forward. So with that, I'd like to thank the Korean Applied Entomological Society for inviting, as well as all my collaborators from the One Kite team, and all my students for funding, for helping me with the project, as well as several funding agencies, and also for Dr. Shin for the invitation. So I know I have basically used up my time, but if you guys have any question, I'd be happy to answer.
Thanks so much for a great talk. I think actually the time is over and is exceeding about 10 minutes, but this is so interesting, and I think it's a rare chance to having you here. So if there is anyone who have any question, just one question for for this time, and then we will actually we have a break, but we may not use the break. But if anyone really need to go bathroom or something, then just to do that when you need to do. Don't worry about interrupting the talk. So is there any question? Anyone?
And 한국말로 하셔도 됩니다.
네네. 질문 있으신 분, 예, 한국말로 하셔도 되고 영어로 하셔도 됩니다.
저 질문 있는데요.
네.
So can you use the BAAM analysis to find the diversification rate of the clades? So can you tell us briefly about the analytical background of that analysis?
Okay, so the question is about BAM analysis.
Yeah.
It is without it is not without the controversy. Some people do not like it, and actually, when we went through the review process, one reviewer didn't really like it, so it asked me to do another analysis. But in order to do the BAM analysis, you first need to have the time tree, divergence time estimate. There is ultrametric tree, and then you need to have the number of species information, and that could be proportional to the number of species in your phylogeny, or if you actually have a really full phylogeny, it could be just a regular number of species. And then and the actual analysis is quite simple. You run through this R package, and then you I mean, I'm not really an expert on R, but I can run it. And the challenge is the interpretation, because what we find is what the output is that there's different episodes of increased rate of diversification, but that doesn't mean that you now that's the pattern, and you have to find whether there's an innovation that can explain that increased rate of speciation. It is one of many available methods for phylogenetic comparative methods, but it is, again, as I said before, it is not without controversy. So one of the reviewers suggested that we use another method called the HIC, or HIC, or BC, which are basically trait-based diversification analysis, right? So did the evolution of certain trait, a certain character, increase the diversification, not necessarily just the number of species? Mm-hmm. And we have done that, and that is even more difficult to interpret. But I can tell you more about that later.
Okay. So I guess the HIC, is that right?
HIC, yep.
HIC program is like more I think with McKenna's data, because he have the tree and he have the traits like phytophagous or non-phytophagous. So that'll be more like useful analysis in his data, right?
I believe so. I think that Dwayne's data set is just full of data, full of rich things. So there are so many things you can do. So yes, once you have the phylogeny, once you have the trait data, then there are many things you can do.
Thank you.
네, 그러면은 여기까지 하고 그리고 다음 세션 넘어가면 될 것 같아요. 저희
채세 질문 가능한가요? 그러니까 지금 휴식 시간인가요?
예, 그렇긴 한데 사실은 지나긴 했는데요. 이상훈 교수님 어떻게 할까요? 지금 뮤트로 돼 있는데 켜고 말씀해 주시겠어요?
예, 혹시 들리시나요?
네.
예. Thank you.
한국말로 하셔도 돼요. 네.
One of the things I was really wondering is in the case of taxonomically resembling species or like sibling species, I guess they can be sharing similar structures in the acoustic organs or something. I guess it's right. But I think the acoustic sounds should be easily distinguished from the sounds from similar species. So my question is, what makes that distinct differences I mean, it could be derived from highly differentiated structures of organs or from a specific behaviors or something. So, yes, I was wondering like that.
So, if I understand correctly, the close related species have similar structures, so they must produce similar sound, but what makes it different?
Yes, right.
So when we actually look at the theory behind sexual selection, there's a huge selective pressure for sexually selected characters to diverge really quickly by fishery and runaway selection. Mm-hmm. Female has a preference, male has the ability to follow that preference. So it is often the case that two very closely related species have very divergent songs, and they're constrained by the morphology, but they are able to maybe move their wings much faster or a different pattern to create different sound. So at that level, if you actually map those characters onto the phylogeny, it is actually not, or if you were to build a phylogeny just based on the songs, then you might actually end up with a star phylogeny rather than a strong phylogenetic signal, because at that level, at the species level, there's a huge selective pressure to diverge those traits in many different ways.
Thank you.
네, 감사합니다.
네, 감사합니다.
오늘 너무 좋은 톡 감사드리고, 예, 시간이 많이 지났기 때문에 이제 좌장을 이수환 교수님께 넘겨서 아마 저희가 10분씩 딜레이한 예정으로 진행을 해야 될 것 같습니다, 교수님.
그래요? 10분씩 딜레이를 한다고?
예, 지금 원래 50분 시작인데요. 지금 거의 3시가 다 됐기 때문에 그래서 다 10분씩 딜레이해서
스케줄보다 10분씩 딜레이가 돼서 6시쯤 4시가 좀 넘어서 끝나야 되겠네. 예, 뭐 그렇게 될 것 같네요. 그러면 쉬는 시간 없이 바로 시작하는 걸로.
네, 중간에 혹시 뭐 화장실 가실 분들은 그냥 잠깐 갔다 오시면 될 것 같아요.
오케이요. 그런데 지금 좀 우리가 결정을 해야 될 게 있을 것 같네. I wonder the two of the members from US whether they are staying here or because it is already 2 o'clock in the morning there. It is very tired for them. I suppose maybe what about Dr. Ho-Jun Song? Do you stay here or just?
네, 저 두웨이는 벌써 간 것 같고요.
아, 그래요?
네, 저는 아까 낮잠을 자가지고.
아, 그렇습니까?
네, 저는 그냥 있을 예정입니다. 네.
예. 그러면 뭐 사실은 두웨인을 제외하고는 우리 송 교수님은 뭐 전혀 문제가 없으시니까.
네.
나는 이걸 영어로 계속 유지를 해야 되느냐 하는 부분 때문에 그랬는데.
한국말로 하세요.
네, 그렇게 되시는 게 나을 것 같습니다. 여기 뭐 지금 있는 사람들도 그렇고. 그러면 쉬는 시간 없이 바로 진행을 할게요. 조금씩 좀 스피드를 내주시는 게 좋을 것 같고, 지금부터 발표하시는 분들이 20분씩 발표를 예정이 돼 있는데, 뭐 아마 20분은 다 넘길 가능성이 많아요. 준비하신 내용들도 꽤 되고, 또 이렇게 발표를 하다 보면 또 질문도 받고 해야 되니까. 어쨌든 그러다가도 좀 발표 내용은 좀 스피드하게 줄여서, 뭐 줄인다기보다는 빨리 좀 진행을 시켜주세요. 그럼 이어서 바로 소개를 해드릴게요. 다음 발표자는 우리 이승현 박사님이 Recent Phylogenetic Study on Cerambycidae and Nitidulidae 두 딱정벌레에 대한 개통 연구를 한 걸 가지고 발표를 해주시겠습니다. 바로 진행을 하시죠.
네, 알겠습니다. 보이시나요?
오케이, 좋아요.
네, 그러면은 발표를 시작을 하겠습니다. 네. 안녕하세요. 저는 Recent Phylogenetic Studies on Serendipity and Repeatability라는 제목으로 발표를 준비한 서울대학교 이승현이라고 하고요. 바로 시작하겠습니다. 제가 개인적으로 하늘소를 훨씬 좋아하기는 하지만 뭐 여태까지 쓴 논문을 보면은 대충 반반 정도 되는 것 같아요. Serendipity,랑 Repeatability가. 그래서 이 두 분류군에 대한 어떤 계통학적 연구의 현황과 우리가 어떤 걸 알고 있고 뭐 아직까지 모르는 부분은 어떤 것들이고 뭐 이런 것들에 대한 내용으로 발표를 진행을 해보려고 합니다. 그래서 제 발표도 그렇고 다른 것들도 그렇고 이렇게 애널리틱한 것들보다는 생각의 흐름이 저렇구나, 뭐 그래서 저런 논문을 썼구나, 뭐 그래서 저런 분석을 했구나 이런 것들을 보시면은 훨씬 더 도움이 되지 않을까 하는 생각으로 이 발표를 준비를 했습니다. 하늘소에 대해서 간단히 설명을 드리면요, 하늘소 상과라고도 하고 그냥 뭐 그 하늘소 상과가 이제 그 모노파일레틱하지 않다고 알려져 있으니까 Serendipity Assemblage라고 하는데 얘네는 이제 전 세계에 35,000종 이상이 알려져 있고요. 그러니까 35,000종이면 지구 전체 다양성의 한 3% 정도 되는 거죠. 굉장히 거대한 그룹이라고 할 수 있습니다. 이 친구들은 워낙 다양한 생태와 형태적인 것들을 갖고 있어서 뭐 하나하나 다 설명드릴 수는 없지만 뭐 산란하는 방식도 굉장히 다양하고, 그리고 유충이 섭식하는 식물의 부위, 그리고 섭식하는 방법, 뭐 섭식하는 식물의 상태, 뭐 이런 것들도 굉장히 다양합니다. 그래서 뭐 보시면은 초본류를 먹는 애들도 있고, 얇은 가지부터 두꺼운 가지까지, 살아있는 나무, 뭐 죽어있는 식물, 뭐 이런 것들 가리지 않고 특정한 상태, 특정한 그 플랜트 텍사에 맞는 종들이 이렇게 호스트 스페시피티를 가지고 이렇게 그 산다고 생각을 하시면 될 것 같고요. 번데기방 같은 거를 짓는 위치나 섭식하는 그 수목 내부에서의 위치도 굉장히 다양합니다. 아무튼 이런 하늘소는 이런 생태적인 특징을 갖고 있는 분류군이라는 거를 간단히만 알고 가시면 될 것 같고요. 그 하늘소 어셈블리지 같은 경우는 총 4개 과를 보통 이야기를 합니다. 베스페리디, 디스테니디, 옥스펠티디, 세라비시디 이렇게 4개의 과를 이야기를 주로 하는데 근데 그 다양성의 대부분은 이 세라비시디가 가지고 있어요. 그래서 거의 뭐 99.9% 정도, 90 뭐 몇 프로 정도를 차지하는 다양성을 가지고 있고요. 근데 이 하늘소에 대한 전체적인 계통 관계는 사실 그렇게 이 하이어 레벨 릴레이션십 같은 경우는 최근까지도 밝혀진 바가 거의 없었습니다. 그러니까 2014년에 나온 이 핸북 오브 조올로지에 나온 건데 이게 스파차랑 로렌스가 성충, 유충, 번데기의 형태와 생태 같은 걸로 이렇게 유추한 이 트리가 있었고요. 그리고 맥케나 랩에서 나왔던 어떤 그 딱정벌레 전체의 메가트리에서 그 위치를 확인하는 정도만 가능했습니다. 그러니까 여기 보시면은 여기 옥시펠티데, 베스페리디, 뭐 디스테니디, 세라비시디 이렇게 베스페리디 이렇게 있잖아요. 이런 것들만 그 큰 틀이 안에서 어디에 위치하고 얘네의 관계를 집중해서 봤던 논문들은 최근까지 나온 적이 없었는데, 네. 최근에 이 맥케나 랩에서 하나, 그러니까 멤피스에 있는 맥케나 그 팀에서 하나, 그리고 중국의 CAS에 있는 팀에서 하나 이렇게 앵커 하이브리드 이니치먼트를 사용하고 이 마이토콘드리얼 지놈을 사용을 해서 전체적인 그 하늘소의 그림을 본 논문들이 나왔습니다. 보시면은 이 앵커 AH랑 그 마이토지놈은 사실 조금 다른 관점, 그 유전적인 다른 관점에서 본 논문들이기 때문에 이 두 논문에서 어떤 부분들이 컨그런스하고 어떤 부분들이 그 불일치가 있었는지를 확인하면은 이제 앞으로 뭘 해야 될지를 좀 알 수 있을 것 같은데요. 보면은 첫 번째로 아까 말씀드렸다시피 세란바이코이드라고 불렀던 이 세란바이시데, 베스페리데, 옥시펠티디, 그리고 디스테니디 이렇게 4개의 과는 둘 다 분석해서 모노파일레틱하지 않았어요. 그러니까 전체적인 요 큰 덩어리가 있고 요 안쪽에 메갈로포디데랑 올스다크니데라는 과는 약간 그 잎벌레랑 가까운 친구들이 들어간 요런 형태를 둘 다 띠고 있었습니다. 그래서 우리가 세란바이코이드라고 했던 것들은 모노파일레틱한 클레이드가 아니구나라는 생각을 할 수가 있을 것 같고요. 그리고 바깥에서 조금 더 이제 이 그 안쪽 그 셜로우 노드들로 나가 보면은 이제 각 악과들의 어피니티들이 확인이 되거든요. 라미네랑 그 목하늘소 악과랑 넙축하늘소 악과가 이 붙어 있고 랩츄리네랑 그 네키달리라고 꽃하늘소, 번하늘소가 붙어 있고, 세란바이시대 프라잉린에 토파늘소, 토파늘소, 하늘소가 이렇게 붙어 있는 이런 어피니티들은 두 개에서 공통적으로 확인됩니다. 그리고 제가 썼던 이 멀티로커스 데이터를 이용한 페이퍼에서도 공통적으로 확인되는 부분이고요. 그래서 이런 것들은 요 각각의 어피니티들은 꽤 맞는 게 아닌가라는 생각을 할 수가 있을 것 같고, 근데 뭐 다른 점들도 있습니다. 이 왼쪽에 이 CAS에서 나온 논문 같은 경우는 하늘소와 세란바이시대 센수 스트릭토가 파라팔레틱하게 나왔어요. 이렇게 두 개의 클레이드가 여기다가 빠져있는 게 있는. 그래서 이렇게 나왔고, 여긴 모노팔레틱하게 나왔고, 그리고 나머지 전체적인 토폴로지들이 뭐 조금씩 달랐다. 뭐 이런 것들이 달랐고요. 그래서 아직까지는 여태까지 퍼블리시된 데이터로는 우리가 뭐 로버스트한 파일로제네틱 릴레이션십이 이런 거다라고 아직 내세울 만한 거는 없다는 얘기를 드릴 수가 있을 것 같고, 아마 아까 전에 맥케나한테 언퍼블리시 데이터가 여러 개가 있었으니까 그런 것들을 보면은 조금 더 로버스트한 릴레이션십에 대한 그 이해를 할 수가 있지 않겠나 생각을 하고 있습니다. 네. 그래서 그럼 다음으로는 이제 전체적인 그림에 대한 파일로제니는 아까 그 두 개의 페이퍼가 있었고, 그러면 요 안쪽에 보시면은 이 탑 4 서패밀리들이 거의 전체를 차지하고 있거든요. 2만, 11,000, 1,500, 1,000, 나머지는 뭐 100단위, 뭐 3, 이렇잖아요. 그래서 이 나머지 4개의 악과 혹은 이 악과 수준에서의 혹은 그 하위 수준에서의 계통 연구는 얼마나 되어 있는지를 잠깐 짚고 넘어갈게요. 네. 그 전에 이게 피터 스바차라고 하늘소 핸드북 오브 주얼로지 혼자 쓴 사람인데 맨날 하는 얘기가 뭐냐면 이 큰 세람비스 서패밀리에서는 요 분류 체계나 이런 것들이 완전 엉망이다라는 이야기를 매일매일 하거든요. 근데 보통 릴레이한 클래시피케이션이 이 계통적인 관계를 잘 반영해야 된다는 아이디어를 좀 역으로 생각해 보면은 계통적으로도 엉망이고 그렇기 때문에 클래시피케이션도 엉망이다라는 이야기가 되겠죠. 그래서 이것도 뭐 놀랍지 않게도 굉장히 최근에서야 이 악과 내의 그 관계들이 밝혀지기 시작했습니다. 네. 보시면은 뭐 여기 그 방금 전까지 지노믹스 얘기해가지고 이게 뭐 5,200 로사이를 얘기하는 거라고 생각하실 수 있는데 로사이 아니고 베이스 페어고요. 네. 8개짜리 로사이 파일로제네틱스입니다. 그래서 뭐 저희가 보시면 따로 뭐 연락을 하거나 이렇게 한 건 아닌데 비슷한 진셋을 사용했는데 그 이유는 아마도 뭐 C1이나 18S, 28S 같은 경우는 많이들 쓰는 마커고 예전에 그 프로틴 코딩 지인들에 대한 그 계통 시그널이 얼마나 얘네가 잘 주느냐에 대한 논문이 나온 게 있고 그 지인들에 대한 유니버설 프라이머들이 출판이 된 적이 있어서 아마 이런 논문들 때문에 비슷한 진셋을 사용했던 것 같고요. 그래서 아마 저희가 갖고 있는 데이터들을 이렇게 합쳐서 좀 더 큰 트리들도 만들 수 있을 것 같다고 생각을 하고 있습니다. 일단 그렇고요. 네. 근데 사실 이 논문들 보면 이 페이퍼의 애스펙트가 분류학적인 것보다는 훨씬 진화적인 부분에 포커스가 돼 있어요. 왜냐면은 커다란 악과들은 분류학적으로 엉망이라서 이게 도대체 어디서부터 손을 대야 될지 감도 안 오고, 그렇다고 뭐 보시면은 130종을 사용했지만 전체 다이버시티의 1%도 안 되고 120종을 사용했지만 전체 다이버시티의 1%도 안 돼가지고 이거 가지고 전체적인 그림에 대한 무슨 얘기를 할 수가 있냐라는 약간 자신감 없는 그런 것들이 있어서 아마 작은 그룹부터 정리해 나가는 게 텍소노믹에서 그 에스펙트에서는 좋을 것 같다고 생각을 하고요. 네, 그래서 이 중에서 제가 썼던 이 하늘소 악과 논문에 대한 설명을 간단하게 들어보려고 합니다. 이거에 대한 이야기를 드리기 전에 오늘 그 송호준 교수님께서도 방금 전에 발표하셨을 때 Ancestral States Reconstruction이라는 거에 대한 말씀을 하셨어가지고 그거에 대한 간단한 설명을 한번 드리고 진행을 해볼게요. 이 Ancestral, 저도 역시 이제 주로 Ancestral States Reconstruction이라는 방법을 이용해서 이제 거시적인 진화사를 확인을 했는데요. 그게 뭐냐면 좀 쉬운 예를 들어볼게요. 뭐 이런 식으로 여기 계통도가 있고 그리고 여기 이제 뭐 트레이트들이 있어요. 각각의 그 형질들이 얘는 다 육식이고 얘는 초식이야. 그러면 육식은 대충 이 노드 어딘가에서 기원한 것 같다고 예측을 할 수가 있잖아요. 조금 뭐 과학적으로는 조금 불확실할 수 있어도 이해를 좀 편하게 하기 위해서 좀 쉽게 드리는 말씀이에요. 네. 그래서 이렇게 쉽게 유출을 할 수 있지만 이게 만약에 뭐 이런 식으로 돼 있다면 아까 전에 그 뭐 히어링 organ에 대한 내용도 좀 비슷하죠. 여기에서 막 나타나잖아요. 그래서 이런 경우에는 그럼 우리가 ancestral states를 어떻게 뭐 유추하느냐 하면은 뭐 여러 가지 프로그램과 통계적인 방법을 이용을 해서 각각의 노드에서 육식이 ancestral이었을 확률, 여기에서 초식이었을 확률 뭐 이런 것들을 계산할 수가 있어요. 그래서 아까 전에 보셨던 파이 차트가 아마 그런 것들을 의미하는 걸 거예요. 그래서 만약에 이 노드가 초식이었다고 가정을 해보면 육식이라는 거는 여기서 한 번, 여기서 한 번, 여기서 한 번 총 3번 기원한 게 됩니다. 그리고 초식에서 육식으로 왔다가 육식에서 초식으로 간 건 없어요. 근데 반대로 여기가 육식이었다라고 하면은 육식은 서로 다른 시기에 독립적으로 2번 기원한 게 될 거고, 그리고 초식에서 육식으로 갔다가 다시 초식이 되는 것도 이런 현상도 한번 일어났다라는 결론이 나오게 되는 거라서 대충 이런 것들이 ancestral states reconstruction이라고 생각하시면 됩니다. 그래서 같은 트레이트와 같은 토폴로지를 갖고 있어도 이게 뭐 이것 때문에 이 해석이 달라질 수 있다. 그래서 아까 전에 맥케나가 뭐 이런 바이올로지컬한 그거를 하려고 하면은 뭐 샘플링을 촘촘하게 뭐 많이 하는 게 좋다 뭐 이런 얘기를 했는데, 그게 왜 그러냐면 예를 들어서 여기서 초식이 빠져버리면 여기서 육식이 빠져버리면 전혀 다른 얘기가 되는 거잖아요. 그래서 뭐 그런 얘기를 했던 것 같아요. 아무튼 이런 방법으로 분자계통도를 가지고 여기다 형질들을 여러 가지 입힐 수가 있습니다. 이따가 나올 건데 이제 뭐 예를 들어서 A, B, C, D, 뭐 1, 2, 3, 4, 가나다라, 빨강, 초록 이런 디스크리트한 형질들을 이렇게 입혀가지고 그것에 대한 이 ancestral states reconstruction을 해서 트렌드를 볼 수가 있고, 그리고 마찬가지로 지역이라는 것도 사실 디스크리트하게 바꿀 수가 있거든요. 보시면 아까 전에도 나왔지만 뭐 팔레아틱, 뭐 네아틱, 뭐 네오트로픽, 아프토픽 이렇게 나눌 수도 있고, 뭐 호주 북부, 호주 남부 이렇게 나눌 수도 있고. 네. 그래서 똑같이 디스크리트하게 변환해서 똑같은 방식으로 하면은 뭐 그게 파일로지그래피라고 할 수 있어요. 뭐 별게 아닌 거죠. 그리고 이 ABCD 같은 거 말고 연속적인 변수들도 이게 매핑이 가능합니다. 그래서 이건 이따가 설명을 드릴 텐데, 뭐 ABCD가 아니라 뭐 0.74, 뭐 0.99, 뭐 4.71, 뭐 이런 캐릭터들도 전체적으로 시각적으로 볼 수가 있다. 라는 이야기고요. 그래서 이런 아이디어를 갖고 제 논문을 보시면은 이해하는 게 조금 더 편할 것 같아요. 이거 뭐 MP에 나왔던 논문이고, 하늘소의 산란과 관련된 트레이트들에 대한 그 진화사를 추론한 논문입니다. 그래서 하늘소에 대해서 다시 간단히 정리를 해서 설명을 드리면, 유충에 대한 얘기는 안 했으니까요. 하늘소 유충 같은 경우는 다음과 같은 특징이 있습니다. 이 다리가 일단 짧거나 아예 없고, 두 번째는 수목 내부에서 섭식을 합니다. 일반적으로. 그리고 종마다 호스트 스페시피시티가 있어요. 그러니까 특정 종이 특정 나무의 상태, 특정 종이 특정 나무의 종류를 선호한다는 이야기죠. 그래서 이 세 가지를 종합해보면은 유충의 생존은 100% 이 엄마한테 달려있다고 볼 수가 있습니다. 왜냐하면 잘못된 데서 태어났는데 다리도 없는데 나무 안에서 먹는데 뭐 나와가지고 다시 이동을 할 수가 없으니까. 그래서 산란 관련된 전략에 대해서 굉장히 높은 선택을 받을 수밖에 없습니다. 그래서 요런 여러 트레이트들에 대한 연구를 진행을 했고요. 네. 그러니까 제가 이걸 하기 전에는 사실 뭐 거의 하늘소에 대해서도 모르고, 하늘소의 뭐 기주 관계에 대해서도 모르고, 하늘소 앞과 내부의 인터널 팔로제니도 전혀 우리는 아무것도 몰랐던 상황이라고 할 수 있겠죠. 그래서 이런 것들을 해결하고자 했고요. 시간이 벌써 많이 지나서 빨리빨리 진행을 하겠습니다. 네. 이 연구에 120종 넘게 사용이 됐고요. 총 6개의 진셋을 사용을 했습니다. 애널리틱한 건 별로 중요하지 않고, 여러 가지 트리들이 그렇게 큰 토폴로지컬 디퍼런스가 거의 없었고, 되게 마이너한 한 칸 바뀌는 거, 2칸 바뀌는 거 이런 것들밖에 없어서 이 정도면은 멀티로커스 데이터 치고는 꽤 괜찮은 거 아닌가라는 생각을 했었고요. 네, 그래서 이런 트리를 얻을 수가 있었습니다. 네. 그래서 뭐 이 모노파일리랑 이런 거 파라파일리에 대한 내용은 넘어가고, 여튼쨌든 저희는 꽤 괜찮은 트리를 얻었다라고 가정을 하고 ancestral states reconstruction을 진행을 했습니다. 총 3개에 대해서 진행을 했는데요. 기주와 관련된 것, 그리고 산란 전략에 관련된 것, 산란관의 길이에 관련된 것을 매핑을 해서 진행을 했고요. 프로그램들은 이런 거를 썼습니다. 아마 다 비슷한 프로그램들 쓰시는 것 같은데, 그래서 호스트 같은 경우는 기주 식물의 종류와 상태로 총 7개로 나눴습니다. 살아있냐, 뭐 쇠약했냐, 뭐 잘린 지 얼마 안 됐냐, 뭐 죽은 지 오래돼서 썩었냐, 뭐 이런 식으로 저뿐만 아니라 많은 하늘수 하는 사람들이 나누는 카테고리를 사용을 해서 이게 호스트 컨디션을 나눴고요. 그래서 얘네는 그 화렵수에서 시작해서 요런 클레이드들에서 커니퍼로 간, 혹은 요런 클레이드에서 허비큐스 플랜트로 간 애들이구나. 그리고 스트레스 호스트에서 시작해서 살아있는 거나 죽어있는 걸로 약간 중간에서 시작해서 이렇게 약간 극단으로 가는 그런 트렌드를 갖고 있구나라는 사실을 알 수가 있었고요. 산란 방식은 총 4개로 나누어서 진행을 했습니다. 이 식물 외부에 붙이는 것, 그리고 식물 그 내부에 살짝 그 바크레비스 사이에 끼워 넣는 것, 그리고 큰 턱을 이용해서 구멍을 뚫고 오비포지션 핏을 만들어가지고 거기다가 산란관을 넣고 나무 안에다가 산란하는 것, 그리고 나무가 갈라지거나 이러면 깊은 틈이 생기거든요. 그래서 그런 데에다가 쑥 하고 쑤셔박아서 산란하는 이렇게 4가지 형질로 나눴고요. 이것도 마찬가지로 약간 중간에서 극단으로 가는 바크레비스에다가 이렇게 살짝 꽂아 넣는 것에서 바깥에 붙이는 것, 그리고 안쪽으로 깊이 넣는 것, 뭐 이런 식으로 진화했구나라는 트렌드를 볼 수가 있었습니다. 그리고 다음은 산란관의 형태에 대한 걸 봤는데요. 뭘 봤냐면은 가로와 세로, 그러니까 길이와 너비의 비율을 측정을 했습니다. 총 85%의 인그룹에 대한 그들을 다이섹션을 해서 실제로 메저를 했고, 그리고 제가 구하지 못했던 것들은 논문을 참고해서 가로 세로 비율을 쟀습니다. 그래서 역시 아래에서 사용되는 파이투스라는 패키지에서 이런 그 결과를 얻을 수가 있었고요. 마찬가지로 약간 중간에 시작해서 극단으로 가는, 그리고 극단적으로 짧아진 산란관 같은 경우는 하나, 둘, 셋, 4번, 최소 4번 이상 나타났다라는 사실을 알 수가 있었고, 이 친구들은 다 알을 밖에다 붙이는 그 분류군이었습니다. 그래서 보면은 얘네가 알에 붙일 때 사용하는 특별한 기관들이 진화를 했는데, 여기 보시면은 이제 이게 산란관이고 이게 산란관이에요. 상동한 기관이고, 이게 마지막 배마디, 이게 마지막 배마디입니다. 그러니까 둘이 상동한 기관인데 형태가 굉장히 다르죠. 여기 약간 저희는 브러싱 올간이라고 하는데, 이 알을 붙일 때 풀 같은 걸 이렇게 막 약간 바르고 막 덮고 하는 그런 올간들이 마지막 마디에 진화를 한 클레이드들이 이렇게 3개가 있었고요. 사실 얘네 둘은 요거를 갖고 있었기 때문에 2개가 굉장히 가까운 애들이다라고 생각이 됐는데, 보시면 계통적으로 굉장히 멀죠. 이거 아까 전에 동민수 선생님이 질문했던 내용이랑 비슷한 것 같아요. 아무튼 그리고 배 마지막 마디가 아니라 그러니까 앞에서부터 4번째 스트라나이트가 이렇게 브러싱 올간으로 진화한 경우도 있었습니다. 이게 암컷은 있고 수컷은 없거든요. 보시면 여기가 이제 브러싱 올간이고요. 그래서 이렇게 3가지에 대한 ancestral stage reconstruction을 했고요. 그리고 보시면은 아까 전에 송호준 교수님의 트리도 역시 마찬가지 옆에 이렇게 2개 딱 놓고 봤을 때 되게 트레이트 이볼루션이 관계가 있는 것 같네 라고 생각하셔서, 역시 그 네이처 커뮤니케이션 논문에서도 그 correlated evolution 테스트를 하신 것 같은데, 그래서 저도 했고요. 이거 뭐 내용은 중요하지 않은 것 같고, 대충 산란관이 길고 수목 내부에 산란하는 것에서 시작해서 산란관이 짧아지고, 그리고 밖에다가 산란하는 방식으로 이렇게 진화적인 흐름이 왔었다, 뭐 이런 결과를 얻을 수가 있었던 논문입니다. 그래서 이 논문 간단히 요약을 해드리면은, 뭐 하늘수학과의 파일로젠이 처음으로 이루어졌던 논문이고요. 그리고 여러 가지 그 산란과 관련된 트레이트에 대한 그 진화사가 추론이 됐고, 가장 그럴듯한 이볼루셔너리 패스웨이가 추론이 되었던 논문입니다. 그래서 이 연구를 끝내고 나서 뭐 하나를 끝냈는데 사실 해야 될 게 몇 개가 생겨서, 이걸 끝내고 뭐가 생겼는지를 좀 설명을 드리자면요. 뭐 폴리파일레틱한 그룹들이 일단 나타나서 이런 것들에 대한 텍소노미 트리트먼트를 다시 한번 해야겠다라는 생각을 했고요. 같은 칼리딘이라고 하는 같은 트라이브에 묶여 있는데, 형태적으로도 유충과 번데기 형태에서 굉장히 큰 차이가 나고, 그리고 계통도에서도 굉장히 멀리 떨어져 있고, 뭐 이런 형태적인 성충의 차이도 얘네가 가지고 있어서 얘네들을 다시 더 많은 샘플을 가지고 다시 트리 만들어서 형태적인 것들이 어떻게 트라이브를 나누는데 역할을 할 수 있을까라는 내용을 살펴보면 좋을 것 같고요. 뭐 그리고 아까 트리, 전체적인 트리에서 모노파일레틱하게 나타났던 지너스나 트라이브를 가지고 바이올로지컬 트레이트를 가지고 그 ancestral states reconstruction을 하면은 또 재밌는 이야기들을 많이 할 수 있을 거라고 생각합니다. 그러니까 저도 뭐 여러 가지 형태적인, 뭐 이건 유충이고, 이건 번데기, 그리고 생태적인 데이터들도 여러 개를 갖고 있으니까 이런 것들을 트리를 그려서 입혀서 이볼루셔널 히스토리를 추론하는 연구들을 계속 하려고 생각하고 있습니다. 그리고 아까 보시면은 크게 메이저 4개 있다고 했는데 그 중에 3개는 한 번씩 누가 건드렸고 아직 안 건드린 게 하나 있어요. 그래서 이것도 재밌지 않을까라는 생각을 하고 있습니다. 밑빠진 벌레 이야기를 해야 되는데 시간이 지금 거의 다 됐죠.
너무 오버돼서.
네네. 그러면은 이거는 그냥 궁금하신 분들이 있으면 준비는 했지만 네. 아무튼 이것도 연구를 한번 했더니 할 것들이 여러 개가 생겼다. 네. 하나 한다고 끝이 아니고 여러 개 더 생긴다는 이야기를 드리면서 그냥 마무리를 짓는 게 좋을 것 같습니다. 질문 있으시면 질문 받으면서 후딱딱 얘기하고 끝내면 좋을 것 같아요.
그리고 질문을 또 발표했는데 질문을 안 받으면 섭섭할 테니까 시간은 지났어요. 근데 한 분만 질문 짧게 좀 해줬으면 좋겠어요. 질문이 우리 없으신 것 같으니까 혹시 뭐 질문 개인적으로 좀들 해주시고 우리 이 박사 수고 많았습니다. 그럼
감사합니다.
이어서 다음 분 소개를 해드리겠습니다. 그다음 그 발표를 해주실 분은 신승관 교수님인데요. 제목은 Phylogenomics reveals the systematics and evolution of 비비오모파 딥테라. 그래서 파리목 중에서 앞부분에 속하는 파리류들에 대한 파일로지노믹스 결과를 발표해 주실 거고, 참고로 우리 신 교수님은 앞부분에서 발표하셨던 두 분 미국 연사분들하고 같이 활동을 지금 하시다가 최근에 서울대학교 생명과학부에 9월달부터 그 일을 시작을 하셨어요. 여러분들 참고로 하고 우리 신 박사님 발표 부탁합니다.
네. 안녕하세요. 지금 화면 잘 보이시죠?
네, 잘 보여요.
네, 발표 시작하도록 하겠습니다. 사실 저희가 시간이 많이 오버돼서 제가 가능하면은 내용을 가능하면 좀 짧게 해서 뒤에 이제 저희가 초청한 연사분이 할 수 있게 하도록 최대한 노력을 해보도록 하겠습니다. 예, 저는 그 소개해 주신 대로 지금 현재 서울대 생명과학부에 이번 9월달, 2020년 9월달부터 부임을 하게 된 신승관이고요. 제가 오늘 발표할 주제는 Phylogenomics reveals the systematics and evolution of 비비오노파로 해서 비비오노모파는 한국에서는 털파리 하목이라고 하는 종이에요. 이 파리 쪽으로 제가 그 박사 학위를 연구를 하면서 이 털파리 하목 안에 들어있는 그 사이알리데라고 하는 검정 날개버섯 파리를 연구를 했었는데 여기에서 제가 노스캐롤라이나 스테이트 유니버시티로 포닥을 가게 되면서 거기에서 좀 더 상위 분류군 개념으로 연구를 했던 그런 내용하고 그 다음에 앞으로 제가 연구하고자 하는 그런 내용들에 대해서 한번 설명을 드려보도록 하겠습니다. 네, 컨텐츠는 다음과 같습니다. 처음에 인트로덕션하고 뭐 가설에 관해서 좀 설명을 드리고 그리고 계통 관계를 파일로지노믹스로 이 털파리 항목에서는 사실상 계통 관계를 정립한 게 아주 최근에 멀티진 파일로진이 하나 정도밖에 없거든요. 그래서 저희가 이제 두 번째로 파일로지노믹스를 지금 연구를 하고 있고 지금 논문을 내려고 준비 중에 있습니다. 그리고 퍼더 스터디 부분에서는 이볼루션과 관련해서 이 파일로지노믹 트리를 이용해 우리가 할 수 있는 것들이 무엇이 있는지 그런 부분에 대해서 한번 디스커션을 하는 시간을 가져보도록 하겠습니다. 일단 그 털파리 하목에 대한 계통학적인 위치는 기존에 우리가 네마토세라하고 브라키세라라고 그 크게 파리목을 두 개로 나눴었거든요. 그래서 네마토세라는 장가가목, 한국에서는 그렇게 해석을 하는데 장가가목이라는 뜻이 여기 보시는 것처럼 이 더듬이가 여러 마디로 되어 있는 긴 더듬이를 가지고 있다. 사실 뭐 이런 그룹은 짧아 보이지만 여러 마디로 되어 있어요. 그래서 이 비비오노모파를 포함하는 이 위쪽에 있는 티플리모파, 각다기하고 그 다음에 샌드플라이, 뭐 나방파리 종류들하고 모기하고 그리고 털파리 하목이 그 로얼 브라키세라, 로얼 딥테라 혹은 네마토세라라고 하는 그룹으로 묶여져 있었어요. 하지만 2011년도에 멀티진 팔로제니를 통해서 이 비비오노모파라는 그룹이 그리고 이 어떤 네마토세라라는 그룹 자체가 모노팔리가 아닌 것을 확인을 하게 됩니다. 비비오노모파에서는 기존에 모노팔로 제시됐던 몇 가지 과가 좀 여기 있는 펄미소마티데 같은 경우에 좀 애니그마틱, 약간 컨퓨즈한 그룹으로 서제션이 되고, 그리고 이 네마토세라 자체는 모노팔리가 전혀 아니고 이 브라키세라고 하는 우리가 흔히 파리처럼 생긴 파리들은 확실히 모노팔리지만 이에 대한 시스터 그룹으로 비비오모파가 제시가 되면서 비비오모파랑 그 다음에 브라키세라를 합쳐서 네오딥테라라고 하는 새로운 그룹이 제시가 되게 되죠. 그래서 이런 그 비비오모파의 계통학적인 위치는 사실상 대부분 분해자의 위치에 있고 그냥 우리 주변에서 흔하게 볼 수 있지만 특별히 뭔가 피해를 주거나 그러진 않기 때문에 많이 주목받아오지 못했던 그룹 중에 하나예요. 여기 보시면은 이 헤비테트인데 헤비테트도 비교적 단순하고 그리고 한 가지 특징 중에 하나는 지금 아쿠아틱으로 되어 있는 그런 헤비테트가 거의 없다는 걸 보실 수가 있어요. 여기 엑스마이드 같은 경우는 저희 연구에서는 사실상 이 부분이 다른 쪽으로 가서 전혀 비비오모파에 속하지 않는 그룹으로 파악이 되기 때문에 이 그룹을 빼고 나면은 이 어떤 그 대부분이 육상 생활을 하지만 굉장히 축축한 그런 곳에 생활을 하는 종들이고 그리고 헤비테트 개념으로 봤을 때 어떤 파리에서 좀 유명한 모기나 그다음에 뭐 지파리나 아니면 다른 뭐 채채파리 이런 애들은 어딘가 그 썩어가는 것에다 주로 알을 낳거나 뭐 병균을 옮기거나 그런 걸로 굉장히 유명하지만 그냥 이 부식질을 먹는 파리들이에요. 아주 일부가 여기 보시는 것처럼 이 식물을 먹는 그 사이알리드 같은 것들이 있고 그리고 혹파리, 골미찌 같은 것들이 또 식물의 혹을 만드는 그런 종류들이 조금 있기는 합니다. 그렇다 보니까 사실상 헤비테트가 굉장히 단조롭다고 볼 수도 있는데 비교적 심플한 헤비테트를 가지고 있기 때문에 이 안에서 어떤 우리가 진화적인 흐름을 볼 수 있지 않을까라는 개념에서 저는 털파리 하목에 대한 연구를 시작을 하게 되었어요. 여기 보시면은 이 윙리스라고 하는 형질하고 그 다음에 이 파이토파거스, 그러니까 식물 섭식성 그룹들이 이렇게 세 개가 있고 그 다음에 뭐 엔도파라자이트, 라비가 있는 그룹들이 있고 이런 식으로 되어 있는데 그 외에는 사실상 굉장히 그 특별한 뭐 피를 먹는다거나 그런 것들도 없고 엑토파라자이트도 없고 그런 굉장히 심플한 헤비테트를 가지고 있는 그룹이라고 보실 수가 있습니다. 일단 그래서 저희 연구팀에서 그 한 가지 관심을 가졌던 것 중에 하나가 이 비비오모파에서는 일단 식물을 먹는 게 그래도 비교적 한번 트레이스를 해볼 만한 특성이기 때문에 이 비비오모파 안에서도 식물을 먹는 그룹들이 어떤 특징들을 가지고 있는지를 쭉 보다 보니까 여기 보시는 이 골미찌 종류 중에 헬스앰플라이는 굉장히 유명한 해충 중에 하나인데 이런 것들이 대부분 어떤 균과의 공생관계를 통해서 식물을 먹을 수 있었다라는 것들이 연구로 밝혀지고 있었어요. 여기 보시는 연구는 그 비교적 오래전에 일어난 연구긴 한데 아주 간단하게 설명을 드리면 이 헬스앰플라이의 이 유충을 멸균이 된 식물을 먹이게 되면은 그리고 이제 이 알이나 이런 것도 다 깨끗하게 해서 균이 없는 상태로 먹이게 되면은 전혀 섭식을 하지 못했어요. 즉 얘들은 어떤 공생균이 없으면은 식물을 아예 섭식을 할 수 없었다라는 식으로 연구가 되고 있었어요. 제가 연구를 했던 검정날개 버섯파리 쪽에서도 예전에 어떤 아마 이게 남아프리카 쪽인가 거기에서 이 브라디시아라고 하는 쪽의 그룹에 여기에 이렇게 보시면 큰 가시로 되어 있는 이런 형질을 가지고 있는데 여기에 포집을 할 수 있는 형질이 있다는 것을 여기 샘 사진으로 여기서 이제 밝히면서 이런 그룹과 이런 형질이 없는 그룹을 이 페트리 디시에다가 동시에 한 마리씩 넣었을 때 얼마나 애들이 균을 퍼뜨리는가 이런 것들을 봤을 때 어떤 이런 그 가시들이 있는 형질들이 아마도 균을 퍼뜨리는 데 영향이 있지 않을까라는 것을 제시를 했어요. 그래서 제가 생각한 거는 이런 우리가 흔히 알고 있는 어떤 패스트나 그런 굉장히 다양한 질병들을 옮길 수 있는 이 하우스플라이의 경우 이 비비오모파의 그 자매 그룹으로 볼 수 있는데 이 그룹들이 가지는 공통적인 형질 중에 하나가 어떤 균을 옮긴다는 거예요. 물론 이쪽도 어떻게 뭐 사람을 위해를 가하려고 자기가 옮기지는 않겠지만 어쩌다 보니까 굉장히 많은 병균을 옮기고 있고 이쪽은 식물을 먹는 그룹인데 식물을 옮기고 있고 이쪽은 이제 뭐 동물들 죽은 사체 같은 걸 먹잖아요. 애벌레 때. 뭐 그런 식으로 어떤 연관성이 있지 않을까라는 부분에 관해서 한번 생각을 해보게 되었습니다. 그래서 이 부분까지 인트로덕션을 마치고 이제 제가 개통 연구를 한 부분을 한번 보여드릴게요. 저희는 일단은 그 앞에서 그 많은 그 좋은 발표에서 저희가 그 보여드렸던 이 원카이트라고 하는 그룹과 협력을 해서 여기 보시는 시퀀스들을 저희가 시퀀스들을 시퀀싱을 하였고요. 시퀀싱은 그 베이징에 있는 베이직 지노믹 인스티튜트라는 곳에서 트랜스크립톰 시퀀싱을 진행을 했고, 그리고 저희가 직접 시퀀싱을 한 거는 하이브리드 엔리치먼트 데이터. 그래서 여기 앵커드 파일로지노믹스 센터라는 것이 있는데 여기에서 프로브나 이런 것을 디자인을 해서 저희가 그 굉장히 초기 단계에 곤충 쪽에서는 이 파리 쪽에 앵컬 하이브리드 엔리치먼트 그 시퀀싱을 직접 랩 세팅을 해서 진행을 하게 되었습니다. 그래서 여기서 트랜스크립톰 그리고 하이브리드 엔리치먼트를 통해서 좀 더 빠져있는 정도를 채워놓고, 그리고 NCBI에서 찾을 수 있는 그런 데이터들을 함께 활용을 함으로써 컴바인된 데이터를 이용한 파일로제네틱 트리를 그려보게 되었습니다. 여기서 사용을 한 어떤 방법들은 일단은 그 계통학 부분에는 원카이트 파이프라인, 앞에서도 많이 소개가 됐던 그런 파이프라인을 이용을 해서 어셈블된 그 데이터들을 가지고 우리가 흔히 아는 그 마프트 같은 방식으로 얼라이먼트를 해서 여기서 필요 없는 유전자들을 솔팅 아웃하는 그런 식으로 해서 그 필요한 유전자들을 찾아서 분석을 했고요. 그리고 컴패러티브 지노믹스 개념으로는 저희가 이볼루션에 대한 것을 지노믹 수준에서 접근을 하기 위해서 그 PPAM 스캔이라고 하는 방식으로 프로테인 도메인 서치를 하는 그 방법을 사용을 했는데, 이것은 그 맨 앞에 그 드웨인 메카나 박사님이 설명을 해주셨던 그 플랜트 셀러 디그레이딩 엔자임을 솔팅하는 것과 같은 방식을 사용했다고 보시면 됩니다. 이런 것들은 사실 연구를 하기에 어떤 랩에서의 세팅도 중요하지만 굉장히 많은 컴퓨터 리소스가 필요하기 때문에 저희는 클러스터를 이용을 해서 분석을 했습니다. 네, 그래서 요거는 이제 개통 결과고요. 다른 개통 트리들과 마찬가지로 하이브리드 엔리치먼트 데이터가 섞여 있긴 하지만 굉장히 로버스트한 결과를 전반적으로 보여주고 있어요. 그래도 비교적 그 약한 부분들은 주로 하이브리드 엔리치먼트 데이터가 섞여 있는 부분들에서 아무래도 미싱 데이터가 좀 많기 때문에 그런 부분들이 있다고 생각을 하시면 됩니다. 개통 관계에서 가장 중점적으로 보여드릴 것 중에 하나는 아까 아쿠아틱 그룹으로 설명을 드렸던 악시마이데 같은 경우는 지금 여기 보시면은 이 네오딥테라라고 하는 아래쪽에 브라키세라가 몇 개 들어있고, 그 다음에 이쪽 까만색 이 부분부터가 이 비비오노파인데, 비비오노파랑 브라키세라의 시스터 그룹으로 악시마이데가 펄미스마티데와 함께 붙어 있는 것을 보실 수가 있어요. 그래서 이 그룹에 대해서는 이제 뒤에서 설명을 드릴 텐데 어떤 식으로 서제스천을 해야 되는지 좀 더 많은 샘플링이 필요한 상황입니다. 그리고 또 한 가지 중요한 거는 여기 보시는 사이얼로이데아라고 하는 그룹이 두 개로 나눠지게 돼요. 사이얼로이디아는 예전에 단계통으로 해서 여기 이제 혹파리, 세시도미데하고 디토마이데가 원래는 이쪽에 들어가 있었는데, 지금 이 중간에 비비오니데가 들어가면서 사이얼로이디아가 단계통이 아니게 되면서 어떤 그 진화적인 해석에 대해서 큰 차이를 가져올 수 있게 되었습니다. 일단은 이 엑스마이데하고 펄미스마티드 관계를 설명을 드리면 얘들은 어떤 공통적인 형질로 R2 베인이랑 R3 베인이 나눠져 있는 것을 볼 수가 있어요. 그리고 지금 현재 저희 트리의 모든 이 비비오노파에 속하는 그룹들은 R2랑 R3가 컴바인된, 위에서 보여드리는 이런 형태의 윙 베인을 가지고 있어요. 근데 한 가지 문제는 원래 파키나라고 하는 쪽이 여기에 그 펄미스마티데하고 이렇게 섞여 있는 이쪽으로 가야 되는 그룹인데 저희가 이 종을 샘플링을 하지 못했어요. 근데 이 종의 경우는 지금 여기처럼 R2랑 R3 베인이 갈라져 있는 모습을 볼 수 있는데, 저희가 샘플링한 이 속과 근연 그 속으로 알려진 이 크램토노미아의 경우는 지금 비비오니데의 시스터 그룹으로 되어 있어요. 근데 윙 베인 자체가 이렇게 되어 있기 때문에 저희 현재 트리상에서는 윙 베인의 R2와 R3가 합쳐진 게 그냥 하나의 모노파일리, 그리고 이쪽으로 엑스마이데랑 펄미스마티데가 간 것이 R2, R3 베인이 포크드된 그런 그룹으로 되어 있는데, 사실상 샘플링이 부족하기 때문에 이것은 이제 차후에 좀 더 많은 샘플링을 확보를 해서 확인을 해야 될 부분이라고 생각을 하고 있습니다. 네, 여기까지 개통 결과이고요. 그리고 퍼더 스터디 부분에서는 제가 지금 아까 이 그림은 나왔기 때문에 따로 설명을 드릴 필요 없을 것 같은데 네, 이런 식으로 저희가 이 연구를 했던 것들을 어떻게 보면 적용하는 개념이라고 보시면 돼요. 그래서 저희가 가설을 세운 것은 일단 세 가지인데 우선 셀룰레이즈가 호리젠탈 진 트랜스퍼가 일어났을까? 그리고 자일레네이즈가 아마도 호리젠탈 진 트랜스퍼가 어느 시점에서 일어나지 않았을까? 그리고 디톡시피케이션 진들이 어떤 식으로 여기에서 나타나는가? 그 세 가지를 보기 위해서 연구를 진행을 했어요. 그래서 좀 빨리 넘어가면은 이 자일레네이즈 같은 경우는 전반적으로 여기는 비비노파 포함해서 다른 그 식물 섭식을 할 만한 그룹들을 선정을 해가지고 서치를 한 건데 이 자일레네이즈는 대부분의 파리류에서 나타나는 걸 볼 수가 있었고 여기 보시는 사이알리드 한 종이 이 플랜트 셀월 디그레이딩 엔자임을 굉장히 많이 가지고 있고 그 다음에 여기 그 캐로플라티데 같은 경우 좀 많이 가지고 있고 뭐 그런 것들을 볼 수가 있었어요. 근데 사실상 어떤 그 딱정벌레에서 본 것처럼 뚜렷한 패턴이 나타나지는 않았습니다. 그리고 그거를 이제 그 여기 선충 쪽에서 이쪽에 있는 게 선충 데이터들인데 선충 쪽에서 연구한 것들과 비교를 했을 때 어떤 그 비율 플랜트 셀월을 먹는 그 플랜트 피딩 네마토드랑 그 다음에 누트낫 그러니까 그 혹을 만드는 그런 선충류 그다음 박테리아를 먹는 선충류 그거를 비교를 했을 때 스트럭처상으로 대부분 이제 박테리아를 먹는 그룹에 가깝다라는 것을 볼 수가 있었고 그나마 여기 사이알리드 하나가 오돈토사이아라고 하는 종류가 플랜트 피딩 네마토드랑 비교적 그 비슷한 어떤 비율을 가지고 있었다는 걸 볼 수가 있었어요. 그리고 디톡시케이션 진은 세시도미드하고 사이알리드에서 볼 수가 있었고 이런 것들을 가지고 이제 최종적으로 비비노파의 이볼루션의 어떤 가설을 한번 세워서 이제 나중에 연구를 하고자 하는 부분인데요. 이 비비노파에서 일단 아까 처음에 보여드렸던 그 결과 부분에 세시도미드가 다른 사이알리드랑 전혀 다른 그룹으로 나타났잖아요. 그래서 세시도미드와 얘들이 그 식물 섭식성에 대해서 두 번의 완전히 별도로 된 진화를 이루었는데 아마도 이 세시도미드는 일단은 셀룰레이즈 관련된 유전자들을 찾을 수가 없었기 때문에 아마도 이 호스트에 대한 어떤 서프레서 개념으로 해서 이 식물 속에서 이 유충 단계에서 먹을 수 있는 그런 것들을 공생유생과 이용을 하면서 같이 아마 진화를 했을 거고 그 다음에 사이알리드가 지금 이 비비노파에서 대표적으로 플랜트 피딩을 하는 일부가 플랜트 피딩을 하는 그룹인데 이 그룹의 경우는 아마도 라이브 플랜트를 직접 소화를 하기 위해서 이런 유전자를 가져갔거나 아니면 공생 유전자를 이용하고 있을 거다라는 식으로 생각을 했습니다. 그래서 이거를 우리가 자연에서 흔히 볼 수 있는 그 나무하고 이제 개통도를 옆에다 붙여보면은 얘들은 일단 물에 살다가 로버디테라 쪽에 그 모노팔리는 아니지만 물에 살던 애들이 여기서 자일레네즈를 포함하는 어떤 그 플랜트 셀럴 디그레이딩 엔자임들을 아마도 얻게 되면서 썩은 걸 잘 먹게 돼서 이제 사프로파거스나 이런 쪽으로 진화하다가 점점 이제 위로 올라가면서 이제 그 식물들이 있으니까 루트 피딩을 같이 하게 돼요. 그러다가 이 중간에 있는 그룹들은 프레데터도 있고 그리고 그 곰팡이나 아니면 버섯을 먹는 그룹들도 생기고 마지막으로 이제 이파리 쪽으로 가는 세시도미드가 이렇게 별도의 라인으로 나가게 되고 그리고 그 사이알리드나 이런 쪽들이 그 다양한 헤비테트를 가지지만 그 중에 일부가 식물을 먹는 것이 아닌가라고 생각을 합니다. 네, 제 발표는 여기까지고요. 이 연구에 도움을 주신 많은 분들께 감사를 드리고 예, 이상으로 마치도록 하겠습니다.
네, 우리 신 교수님 수고 많으셨어요. 시간이 거의 다 됐지만 한두 분 짧게 질문을 받아볼게요. 질문하실 분 부탁합니다. 질문이 없으신가요? 우리 학생분들도 질문 아까 좀 궁금한 점이 있는데 시간이 없어서 질문들을 못한 것 같은데 앞부분에서는 오히려 짧게 그러면 내가 하나 아까 펑거스 포로 그룹들이 별도로 하나의 모노파일리를 보이는 것 같은데 걔네들이 그 형태적으로 보이는 예를 들어서 지난번에 여기서 잠깐 언급을 했지만 펑거스 포자 스포 캐링 스트럭처의 어떤 시나포모픽 형질들도 좀 보여요?
예, 그것도 이제 콤브라이크 스트럭처라고 그 어떤 키 캐릭터로 사용하는 것 중에 하나거든요. 그게 그것을 포함하는 어떤 서브 패밀리의 형질이라고 보시면 될 것 같아요.
앞으로 뭐 할 일들이 더 많고 좋은 결과가 많이 나올 것 같습니다. 자, 그러면 우리 신 교수님 수고하셨고, 다음 마지막 연사분을 소개해 드릴게요. 그 마지막 연사분은 송정훈 박사님이시고요. 지금 현재 농촌진흥청에 근무를 하고 계시고, 오늘 발표해 주실 내용은 Phylogeny and Evolution of Coastal Low Beetle, 그러니까 반날개에 대한 조간대 반날개에 대한 파일로제니에 대해서 발표를 해주시겠습니다. 바로 준비를 해주시죠. 네, 송 박사님 시작하시면 됩니다.
네, 들리시나요?
네, 잘 들려요.
네, 안녕하세요. 국립농업과학원 곤충산업과의 송정호입니다. 오늘 여러분들께 발표할 내용은요, 바닷가 반날개의 개통 연구와 진화 연구에 대해서 말씀드리고자 합니다. 앞에 연사분들은 주로 이제 과나 목준이의 좀 큰 개통의 연구들에 대해서 말씀을 드렸는데요. 저는 속준이 위주로 해가지고 이제 좀 더 깊이 있게 보고자 합니다. 먼저 목차입니다. 먼저 배경에 대해서 말씀을 드리고요. 연구 내용은 크게 세 가지에 대해서 말씀드리겠습니다. 바닷가 서식처 진화에 대한 기원에 대해서 살펴볼 것이고요. 두 번째에서는 종 동정이 어려운 종 복합체의 종 한 개를 어떻게 나누는지에 대한 연구, 그리고 그 안에서 나타난 미토콘드리아와 핵 유전자 트리드 간의 불일치 부분들, 그리고 마지막으로 세 번째 파트에서는 종 개통수 구축과 그리고 생물질의 연구들에 대해서 소개하고자 합니다. 먼저 반날개 딱정벌레를 좀 살펴보겠습니다. 이들은 곤충강 딱정벌레목에 속해 있는 그룹으로서 다들 아시는 것처럼 딱정벌레목이 곤충강에서 가장 큽니다. 그리고 이 반날개과는 또 딱정벌레목에서 가장 큰 과입니다. 전 세계 약 6만 5천 종 이상이 기록되어 있는데요. 오른쪽에 보이시는 파이 차트를 보시면은 이 반날개과의 종수가 다른 척추동물문의 종수랑 거의 유사한 것을 볼 수가 있었습니다. 제가 가입되어 있는 반날개과의 소셜 네트워크를 봐도 매주 한 종 이상의 신종들이 계속해서 보고되고 있습니다. 동물계에서 가장 큰 과로서요. 다른 딱정벌레와 달리 딱지날개가 짧게 축소되어 있습니다. 이러한 혁신적인 발휘품은 일단은 노출된 배마디 위쪽 등면 부분이 경화가 되어 있고요. 그리고 배마디는 움직임이 가능합니다. 그리고 넣다 뺐다 할 수 있는 그런 구조로 되어 있기 때문에 좁은 서식처에서도 들어갈 수가 있고요. 또한 그 복부마디에는 방어샘이 잘 발달되어 있어서 작은 사이즈에도 불구하고 이 디펜스에 있어서 탁월한 모습을 보입니다. 이러한 종 다양성과 함께 이들은 역시나 다양한 서식처에서 성공적으로 적응하고 있습니다. 그래서 오늘 제가 주목하고 싶은 서식처는 이 바닷가라는 서식처입니다. 다들 잘 아시는 것처럼 바다 환경에는 곤충이 별로 없습니다. 실질적으로 이 대양에서 서식하고 있는 것은 시스케이터 같은 종류만이 극히 드물게 보고가 되어 있습니다. 왜 바다에는 소수의 곤충이 사는가라는 부분들을 생각해 볼 때 먼저 생리적인 문제들을 볼 수가 있습니다. 바닷물이다 보니까 호흡에 문제가 있고요. 그리고 삼투압 부분들을 해결해야 됩니다. 더불어서 바닷가는 바람이 강하게 불고 있고 파도도 계속 치고 있습니다. 그리고 물에 대한 어떤 저항성도 부분들도 있어야 되고요. 그리고 더불어서 진화사적으로 봤을 때 곤충이 다시 바닷가로 진출할 때 보면은 먼저 갑각류들이 서식을 하고 우점하고 있었기 때문에 그들과의 경쟁에 있어서 굉장히 어려운 부분도 있었을 것입니다. 그럼에도 불구하고 방금 소개해드렸던 반날개과 무리들은 성공적으로 좀 적응하고 있는 편입니다. 그들은 이 바닷가에서 약 400여 종이 이 바닷가라는 서식처에 한정되어 서식을 하고 있습니다. 저는 이러한 바닷가 반날개 중에서도 바닷가에 서식하는 다소형 반날개 속그룹들에 대해서 집중하고자 했습니다. 작년과 재작년 같은 경우는 피서지에 보시는 것처럼 홍딱지 바소형 반날개가 대발생하였습니다. 그러다 보니까 피서객들이 피서객들을 깨물고 그리고 거기에 계시는 상인들에게 좀 피해를 줬습니다. 올해는 아직까지 보고가 되지 않았습니다. 그래서 좀 유명해졌고요. 또 제가 이 그룹에 관심을 가졌던 이유 중에 하나는 재미있는 생활서입니다. 이들은 파리의 외부 기생자로서 파리 번데기에 유충 1년기 때 파고 들어가서 성충이 되고자 하는 파리 번데기를 그 안에서 잡아먹곤 합니다. 그리고 그 안에서 나머지 발생 과정을 거치죠. 얘네들의 생존에는 파리가 꼭 필수적인 것입니다. 실질적으로 이들의 암컷은 파리 애벌레의 흔적, 즉 분변 같은 흔적을 찾아서 그곳에 산란하는 것으로 알려져 있습니다. 오른쪽에 보시는 이 사진을 보면은 파리 번데기인데 나오는 걸 보면은 이제 바수형 반날개 무리들이 나온다는 거죠. 그래서 주요 연구 내용으로 첫 번째는 이들의 바닷가 서식처 진화의 기원에 대해서 저는 궁금해서 연구를 진행했습니다. 일부 종들은 바닷가에 성공적으로 정착했습니다. 알레오카라 속 자체는 약 500여 종이 기록되어 있는데요. 그 중에서 약 22종 정도가 이 바닷가에 한정되어서 서식을 하고 있습니다. 해조류라든가 혹은 이 썩은 사체에서 서식을 하고 있습니다. 그래서 저는 먼저 2001년도에 독일의 마우스라는 학자에 의해서 바수형 만나기 속에 분자계통 수가 그려졌는데요. 저는 이 데이터에다가 바닷가에 서식하고 있는 종들을 샘플링을 해서 추가를 하여 분석을 해보았습니다. 그 결과 왼쪽에 보시는 것처럼 파란색에 매핑되어 있는 것처럼 이들은 총 4개의 기원을 보였습니다. 그러나 최근에 그 호주 태즈메니아에 있는 한 노학자에 의해서 마찬가지로 파리 번데기를 채집을 했는데 그 안에 바수형 반나개류들이 나오다 보니까 저희들한테 이제 컨택을 했습니다. 그래서 그분이 주신 샘플을 추가해서 보았던 1개의 기원을 더 찾았습니다. 그래서 5개의 독립적인 바닷가로 서식처 진화의 기원을 확인할 수가 있었습니다. 이 바닷가 서식처 진화 외에 일부 종은 미토콘드리아 유전자 트리에서는 나뉘어지지 않는 것을 볼 수가 있었습니다. 왼쪽에 보시는 것처럼 박스 안에 있는 것처럼 일부 종들은 서로 구분이 되지 않았습니다. 알레오카라 세그레가타 푸에치아는 나뉘어지지 않았고요. 그 밑에도 마찬가지입니다. 그래서 샘플을 더 추가를 해서 같은 유전자로 분석을 해보았지만 역시나 나뉘어지지 않았습니다. 저는 그래서 이러한 점에 좀 끌려서 좀 얘네들을 좀 핵 유전자도 넣고 그리고 샘 같은 것들을 찍어서 마이크로 스트럭처까지 보면서 통합적 분류를 한번 해보아야겠다라는 생각을 하게 되었습니다. 그래서 두 번째 파트를 진행을 했고요. 그 중에서도 해변 바수형 반나개 종복합체라고 해서 이들은 형태적으로 매우 구분이 어렵습니다. 배마디 마지막마디를 떼서 생식을 해부를 해야지 정확하게 그 종이라고 동종을 할 수도 있습니다. 그래서 저는 이들을 정확하게 종의 바운더리를 설정하기 위해서 통합적인 분류 방법을 적용했습니다. 일단은 유전자 차이를 비교를 해보았습니다. 그래서 그 결과 알레오카라 푸에치아이와 세그레가타 이 두 종은 미토콘드리아 유전자 간의 차이에서 알레오카라 푸에치아의 종내 차이와 거의 유사한 수준을 보여주면서 이들은 종을 나눠주지 못했습니다. 다음으로는 미세고조 형질 차이점들에 대해서 분석을 해보았습니다. 그 결과 이 두 종에 대한 차이점들을 확인을 할 수가 있었습니다. 대표적으로서 더듬이 마지막 마디에 알레오카라 푸에치아의 종은 이 감각 구멍들이 나와 있었고요. 그리고 알레오카라 푸에치아이가 세그레가타보다 프로노톰이라든가 이 헤드에 있는 그 마이크로 레티큘레이션, 즉 그 주름 구조들이 굉장히 더 촘촘하다는 것을 확인할 수가 있었고요. 생식기의 구조를 샘으로 찍어서 더 자세하게 보았더니 확실히 차이점들을 발견할 수가 있었습니다. 더불어서 핵 유전자와 함께 유합된 다계통수를 통해서 각각의 종들은 뚜렷한 리니지들을 형성하는 것을 확인할 수가 있었습니다. 그러나 제가 재밌었던 부분들은 미토콘드리아 트리와 핵 유전자 트리 간에 불일치되는 부분들을 확인했다는 것입니다. 알레오카라 푸에치아이와 세그레가타는 알레오카라 푸에치아의 일부가 세그레가타 안으로 들어가면서 종을 구분해주지 못했습니다. 그러나 핵 유전자 트리에서는 이런 현상들이 해결되는 것을 볼 수가 있었습니다. 저는 이러한 점에 매력에 끌려서 이 연구를 좀 더 확장해 봐야겠다. 그래서 더 많은 지역을 가서 샘플링을 하고 더 많은 유전자들을 추가를 해서 계통을 보았습니다. 그래서 종계통수 스피시스 트리라는 그 방법들을 이용을 하였고 더불어 생률지리 연구까지 수행을 하였습니다. 보시는 것처럼 임플레노타와 트리오카라 아속은 대표적인 바닷가 바수연 반나기 속입니다. 이들은 종수 측면에서 봤을 때 바닷가 바수연 반나기 무리 중에서 가장 성공적으로 진화한 그룹들입니다. 이들은 함께 바닷가로 흩어져 진화한 것으로 확인이 되었습니다. 그래서 세 번째 파트의 목적은 먼저 계통수를 구축하는 것입니다. 방법론적으로는 여러 유전자들의 염기 서열을 하나의 형질 자료로 합쳐서 분석하는 전통적인 콘케이트네이티드 된 방법과 지난 파트에서 발견한 미토콘드리아와 핵 유전자 일일성을 고려해서 모델링을 할 수 있는 멀티스피시스 코알러스 베이스된 스페시스트리 이스티메이션을 이용을 해서 좋은 계통수를 구축을 했습니다. 그렇게 만들어진 건전한 계통수를 가지고 그 다음에 목적으로서 이들이 언제 기원하고 어떻게 지금의 분포 양상을 띠게 되었는지 이해하기 위해서 분기 연대를 측정하고 생물 지리를 분석했습니다. 먼저 마찬가지로 미토콘드리아 DNA 트리와 뉴클리어 DNA 트리 간의 차이점들에 대해서 보도록 하겠습니다. 여전히 이 부분들은 해결되지가 않았습니다. 여전히 차이점이 나타났고요. FHI와 세그레가타는 여전히 나눠지지 않았습니다. 단 흥미로운 것은 FHI는 두 개의 그룹으로 나뉘었는데요. A그룹과 B그룹으로 나뉘었습니다. 이 A그룹은 세그레가타와 하나로 묶여지는 것을 볼 수가 있었습니다. 특히 A그룹에 있는 그 샘플들은 알레오카라 FHI와 세그레가타가 함께 공존하고 있는 이들의 분포가 겹쳐지는 부분들에 있는 샘플들이 주로 그려있습니다. 이 홋카이도 남부뿐만 아니라 이 왼쪽에 있는 러시아의 같은 유도성에 있는 샘플들도 마찬가지의 현상들을 볼 수가 있었습니다. 그래서 이들은 아직까지도 분포가 겹치는 지역에서는 교잡 현상이 일어나는 것으로 보이고, 그리고 미토콘드리아의 유입이 한쪽 방향으로 좀 쏠리는 현상들이 있는 것으로 보입니다. 이거에 대한 추가 연구도 필요한 상황입니다. 더불어서 이제는 모든 유전자를 유합해서 콘케이트네이트된 방법을 이용해서 구축한 계통수에서는 각각의 종들이 뚜렷한 리니지를 형성한 것을 볼 수가 있었습니다. 단 임플레노타 아속의 유럽에 서식하고 있는 알레오카라 업스큘레라 종만이 임플레노타 아속의 바깥쪽으로 나오는 것을 볼 수가 있었습니다. 이들은 형태적으로 임플레노타 아속에 굉장히 가까웠기 때문에 좀 의구심이 들었었습니다. 그러나 스피시스 트리 방법을 이용을 해서 분석해 보아니 임플레노타 알레오카라 업스큘레라는 임플레노타 아속을 묶이면서 각각의 단계통을 형성하는 것을 확인할 수가 있었습니다. 그런 다음 이제는 이들이 언제 기원했는지를, 그리고 이들이 어떠한 과정을 통해서 현재 분포 양상을 떼게 됐는지를 측정하기 위해서 먼저 분기 연대를 측정했습니다. 이티클레이드 같은 경우는 파슬이 없기 때문에 일단 바수연 반나기의 가장 오래된 화석을 이용을 해서 바수연 반나기 속의 데이터를 이용하여 분기 연대를 측정을 하였고요. 거기에서 이티클레이드의 분기 연대 시점을 칼리브레이션 포인트로 잡아서 보정을 해서 본 스페시스 트리에다가 연구에 분석에 이용을 하였습니다. 그런 다음 이티클레이드의 생물 지리와 조상의 분포 지역을 분석하기 위해서 파시모니 기반인 SD바 분석과 보다 최신에 개발된 라이클리우드 기반인 DEC 기반의 방법을 이용을 하였습니다. 이들의 분포 지역은 위의 지도와 같이 크게 4개의 생물 지리구를 나누어서 웨스턴 팔레아틱, 이스턴 팔레아틱, 웨스턴 니얼틱, 이스턴 니얼틱으로 코딩하였고, 각종의 이름 앞에 있는 원의 색깔들은 위쪽 지도상에 표시한 현재 이들이 분포하고 있는 지역의 색을 나타내 줍니다. 더불어서 이 가로 안에 있는 이 알파벳도 위에 있는 지도와 상응합니다. 각 노드에 있는 이 파이 차트들은 이들의 조상 지역을 분석한 확률값의 분포입니다. 일부 노드에 있는 이 알파벳 ABC도 마찬가지로 ABC도 위쪽 지도에 보이시는 생물지리구 코드를 표기하는 것입니다. 예를 들어서 1번의 노드에서 ABC는 그 시점의 조상들이 ABC 지역에서 모두 분포하고 있는 것을 말합니다. 노드 왼쪽에 있는 파이 차트는 SD바 결과이고 오른쪽은 DC입니다. 조상 지역을 분석한 결과 현재 분포 양상을 보이기까지 총 한 번의 분산 과정과 3번의 분단 과정이 요구되었습니다. 이제 이들의 기원과 분화와 관련된 지질학적 사건들에 대해서 논의해 보겠습니다. 분석 결과 SD바와 DC는 동일한 조상 지역을 지지해 줬습니다. 리트클레이드의 조상은 초기 미오세 시기에 분기하였고 이스턴 니얼틱을 제외한 북반구 전역에 넓게 분포한 것으로 보입니다. 다음은 2번 노드를 보겠습니다. 인플레노타 아숙의 조상은 약 1800년 전에 동일한 지역에서 기원을 한 것을 보이고 이후 웨스턴 팔리아틱 지역과 이스턴 팔리아틱 그리고 웨스턴 니얼틱은 분단 현상을 통해서 분리된 것으로 분석이 되었습니다. 이 시점은 지중해와 인도양을 연결해주는 테티스해가 마지막으로 닫히는 시점과 거의 일치하는 것을 볼 수가 있습니다. 유전자의 흐름이 중단되면서 고유의 유전자 풀들을 형성하여 종이 형성된 것으로 보입니다. 다음은 3번 노드를 보겠습니다. 동아시아와 북아메리카의 인플랜도타의 조상은 미오세 중계의 이스턴 팔리아틱과 웨스턴 니얼틱에 기원하였습니다. 이들은 분단 사건을 통해서 동아시아와 북아메리카 클레이드로 분리된 것으로 분석되었습니다. 이들의 미오세 기후 전환으로 인해 급격하게 지구의 온도가 내려가고 해류의 순환이 변화되는 시점과 거의 일치합니다. 그래서 이들의 종분화와 연관성이 있지 않을까 조심스럽게 생각해 봤습니다. 다음은 5번 노드의 북아메리카 클레이드입니다. 컬티덴스와 리토랄리스라는 종의 조상으로부터 웨스턴 니얼틱의 일부 집단이 이스턴 니얼틱으로 확장되는 것을 지지해 줬습니다. 이 시점은 파나마 스웨이가 열리는 시점으로서 태평양과 대서양의 바닷물의 교환이 가능했습니다. 이러한 분포의 확장은 바닷가에 서식하여 해조류를 타고서 래프팅해서 이동하는 것과 관련성이 있을 것으로 사료가 되어집니다. 또한 리토랄리스 종은 분포가 재미있습니다. 북아메리카 서쪽과 동쪽 해안에 모두 분포하고 있는 특이한 양상을 보입니다. 이 두 집단의 분기 시점은 약 500만 전으로 추정되었고요. 이 결과는 오랫동안 학계에서 받아들였던 파나마 해협이 닫히는 시간보다 더 오래된 것으로 분석이 되었습니다. 그 전까지는 집단끼리 교류가 있었으나 이후에 교류가 끊기고 각 지역에 적응한 것으로 사료됩니다. 자, 그런데 말입니다. 근데 이 리토랄리스라는 종은 일단 태평양 대서양 이소적인 분포 패턴을 보이고요. 그리고 북미 서부와 동부 집단 간에 파나마 해로가 마지막으로 막힌 후에 오래전 동안 생식적으로 격리되어 있습니다. 또한 북미 서부와 동부 집단은 약 500만 전에 나뉘어서 저는 이 연구가 끝나고 나서 좀 의심스러웠습니다. 그래서 이들을 좀 더 깊이 있게 살펴보고자 분자와 형태 형질들을 이용해서 보았습니다. 간단하게 C1 NJ트리를 그려서 북미 서부와 북미 동부 간의 차이점을 보았는데요. 약 5% 이상의 차이가 나왔습니다. 더불어서 좀 더 디테일하게 그 생식계 형질들을 보았습니다. 그 결과 일관성 있게 수컷 생식계의 레터럴 부분들이 보시는 것처럼 이 레트로알리스는 학기체 모양인데요. 그러나 이 북미 동부에 서식하고 있는 집단들에서는 이 곤봉 모양이 형성되는 것을 볼 수가 있었습니다. 더불어서 이 배쪽마디에서도 배면에서도 보면은 이 앞쪽의 돌기 부분들이 길고 좀 더 뾰족한 것들을 확인할 수가 있었습니다. 그래서 이제 드디어 신종을 또 하나 찾았다라고 생각을 했는데 문헌조사를 통해서 의문점이 하나 발생이 됐습니다. 동종인명 처리된 인플레노타 마리티마라는 이들이 있었는데요. 보시는 것처럼 이들은 흥미롭게도 북미 동부에 서식을 하고 있었습니다. 이들에 대한 선정 기준 표본에 대한 확인이 필요할 것을 보여서 스미소니안에 연락을 해서 표본을 대여를 했습니다. 그 타입을 비교해 보았더니 아니나 다를까 북미 동부에 서식하고 있던 리토랄리스라는 종은 35년 전에 신원임 됐던 알레오카라 마리티마로서 판결이 됐습니다. 마지막으로 동아시아 지역의 종들을 보겠습니다. 7번 노드와 12번 노드처럼 리트클레이드의 대부분의 종분화는 미오세 후기와 초기 플리오세에 일어났습니다. 이것은 아마도 일본과 유라시아 대륙이 반복적으로 붙었다 떨어졌다 하는 현상과 연관된 것으로 보입니다. 이런 사건들은 동해안의 종 다양성을 풍부하게 하는데 공헌했을 것이라 그렇게 생각이 되어집니다. 마지막으로 결론입니다. 바설만나게 속의 바닷가 설저 진화의 기원을 살펴보았더니 다기원인 것을 볼 수가 있었고요. 그리고 이테클레이드 내에서 미토콘드리아와 핵 유전자 트리들 간의 불일치성들을 확인을 했습니다. 그리고 멀티스피시스 코알런시스 베이스된 그런 방법을 이용한 그 종 개통수 구축이 이티클레이드 내에서는 좀 더 레솔루션이 해상력 있는 그런 개통 유형 관계를 얻는 것을 볼 수가 있었고요. 네 번째로는 이티클레이드의 조상은 이스턴 리얼틱을 제외하고 북반구 전역에 그 초기 미오세 시기에 분포한 것으로 확인이 되어졌습니다. 그리고 이들의 생물질의 분석을 결과를 통해서 한 개의 분산과 세 개의 분단 과정을 통해서 이들은 현재의 생물 분포를 띠게 되었고 이들의 현재의 분포 양상을 띠게 된 과정은 메커니즘은 바이캐리언스가 더 많은 영향력을 끼쳤다라고 판단을 내렸습니다. 이 연구를 해서 많은 분들이 도움을 주셨습니다. 먼저 한국의 반날개 개척자이신 안기정 교수님, 제 은사님이시기도 하신데요. 기관에 온 지 한 1년이 됐는데 한두 시간 동안 이제 교수님과 계속 미팅하면서 또 연구했던 그 시절이 또 가끔씩 생각이 들더라고요. 또한 더불어서 이 연구를 해서 많은 분들이 채집을 도와주시고 많은 분들이 샘플들을 보내주셨습니다. 많은 분들께 감사를 드리며 발표를 마치도록 하겠습니다. 경청해 주셔서 감사합니다. 이상입니다.
네, 우리 송정훈 박사님 발표 감사합니다. 시간이 조금 오버를 했습니다. 마지막 연사분이니까 우리 또 궁금한 질문들 좀 받기로 하죠. 네, 질문들 해주시죠.
네, 질문 하나 해도 될까요?
어떤 문제죠?
네.
오신 박사님. 네.
네. 콜레센 트리 같은 경우에 일반적으로 하이어 레벨 바이올로지니에서는 오히려 해상력이 안 나오는 경우도 있는데 이렇게 그 작은 그룹으로 할 때는 더 잘 나오는 경우를 저도 종종 본 경험이 있거든요. 그래서 그런 이유가 어떤 걸지 연구하는 과정에서 혹시 생각하신 게 있으면 얘기를 해주시면 좋을 것 같아요.
일단 제가 선택한 그룹들은 텍슨 샘플링이 거의 완벽하게 되어 있고 그리고 이제 그들 간의 유전자 트리들 간의 좀 이질성들이 좀 많이 고려되어져서 좀 이렇게 나오지 않았나 생각이 듭니다. 일단은 약간 텍선 샘플링이 좀 완전하게 되어 있는 상황이긴 합니다. 예. 저도 이제 이 그룹밖에 안 해봤으니까요. 다른 그룹에서는 또 어떻게 될지 모르겠습니다. 예.
네, 또 다른 질문 한두 개만 더 받을게요. 예, 저 이승현 박사.
그러니까 보니까 컨카티네이티드 방법보다 콜레센트가 좋아가지고 그거를 쓰셨다는 말씀을 하셨잖아요.
예.
그리고 그 트리를 레퍼런스 트리로 써가지고 이제 뒤쪽 분석들을 다 하신 거면은 이거를 썼을 때랑 저거를 썼을 때랑 트리 토폴로지가 다르면은 결국에는 뒤쪽 얘기는 완전히 다 달라지게 되잖아요.
다시 한 번만 말씀해 주시죠.
네?
다시 한 번만 말씀해 주시죠.
그러니까 분석 방법이랑 진셋에 따라서 되게 트리 토폴로지가 달랐던 거를 보여주셨던 것 같은데,
네네.
그게 다르면 뒤쪽 분석들이 다 내용이 달라지잖아요. 그러니까 해석이 뒤쪽에
네네.
바이오지오그래피에 대한 내용들이. 근데 혹시 레퍼런스 트리를 그걸로 이용하신 이유가 혹시 있을까요? 제일 좋다고 판단한 근거나 뭐 이런 것들이.
제가 참고한 관련돼서 참고한 책들이랑 문헌들이 이제 스피시스 트리 구축 방법에 대해서 이제 최근에 분화된 종들 간에 어떻게 하면은 이들의 종을 바운더리 설정해 줄 수 있을까라는 부분들을 좀 제가 보게 되었는데, 특히 지금 같은 경우에는 핵 유전자랑 미토콘드리아 유전자 간의 이질성들이 굉장히 많이 보이거든요. 근데 이 스타비스트 같은 이 멀티스피스 코알란스 방법은 스타비스트 프로그램 같은 경우는 각 유전자 트리들을 먼저 구축을 하고요. 동시에 유전자 트리들이 입력 파일이 되어서 이들 사이 이질성에 대한 정보들을 이제 모델링해서 구하게 됩니다. 저는 이 이질성 부분들에 대해서 발견이 되어졌고 그렇기 때문에 이 방법을 이용을 해서 저는 구축을 한 것이죠. 네. 답변이 됐는지 모르겠습니다. 예.
네네.
그래서 그것을 가지고 스피시 트리 방법을 이용을 하여 마찬가지로 분기 연대를 측정을 했고요. 그리고 그 분기 연대 측정한 그 스피시 트리를 이용을 하여 그걸 프레임으로 가지고 이제 생물지리 분석을 수행을 했습니다. 네. 그래서 제가 이 분류군에서는 제가 신뢰했던 더 긍정적으로 봤던 개통수는 스피시스 트리 방법을 이용한 개통수를 가지고 했다라는 점을 말씀을 드리고 싶습니다. 예.
네, 이걸로 뭐 시간도 다 됐고요. 우리 송 박사님 감사합니다. 나중에 뭐 또 좋은 발표도 부탁드리기로 하고, 우리 송 교수님 아직 안 주무시고 일어나 계시네.
아, 네.
감사합니다. 사실 뭐 이 심포지움을 우리 신 교수하고 주관을 뭐 만들어 보면서 취지가 우리 아까 송호준 교수님이 말씀해 주셨듯이 사실은 어떤 뭐 코로나 때문에 직접 만나지 못하는 이런 부분들도 있기도 하고 또 이게 그러다 보니까 약간 서로 간에 국가 간에 지역 샘플이라든지 아니면 정보 교환이 오히려 조금 더 필요한 그런 시대도 되지 않았느냐. 그러니까 특히 우리 파일로제니 하시는 분들은 지역 간의 샘플들 비교가 굉장히 중요하고 그걸 또 뭐 다 돌아다니면서 할 수도 없는 거고 그런 취지에서 저희가 심포지움 제목도 인터내셔널 코퍼레이션이라는 얘기를 뒤에서 좀 적어봤고요. 그리고 이제 또 우리 미국에 계신 뭐 이번 심포지움에서는 우리 웨인 맥케나 박사하고 그다음에 우리 송호준 교수님이 발표를 해주셨는데 특히 송 교수님은 또 저희하고는 뭐 특별한 관계니까 나중에 우리 학생들이나 또 연구자들하고 이런 기회를 통해서 어떻게 보면은 또 좋은 것 같기도 합니다. 이게 뭐 온라인으로 이렇게 쉽게 만날 수도 있으니까 그런 기회를 자주 가져도 좋지 않겠느냐 하는 생각도 들고 그런 취지에서 오늘 이렇게 발표를 해주셔서 너무 감사드리고 우리 신승관 교수도 이제 뭐 학교로 와서 또 이렇게 스타트하는데 이렇게 같이 또 이렇게 주관을 해줘가지고 너무 고맙고요. 특히 이제 나머지 발표자분들도 감사합니다. 또 발표자분들 중에서 일부는 이미 또 나가신 분들도 계신 것 같은데 뭐 어쨌든 바쁘시니까 전반전에 우리 아침에는 좀 어제 저녁하고 굉장히 좀 당황을 했어요. 시스템 때문에. 근데 오히려 이렇게 자기 자리에서 하니까 오히려 더 편한 것 같습니다. 뭐 괜히 무슨 리조트 빌려가지고 이렇게 가서 하는 것도 의미가 있고 또 거기서 또 전송을 해주니까 유튜브로 전송을 해주니까 좋은 것도 있긴 한데 어쨌든 이런 기회가 꼭 심포지움이 아니더라도 필요에 따라서 자주 이렇게 커넥트 해가지고 협력을 할 수 있는 그런 기회가 있었으면 좋을 것 같습니다. 우리 송 교수님 혹시 끝까지 이렇게 남아주셨는데 좋은 말씀 한번 해주시죠, 마지막으로.
네. 일단 초대해 주셔서 너무 감사하고요. 그 다음에 저는 이제 진화생물학을 하고 또 개통분류를 하는데 미국에서도 이제는 학생분들이 한국 학생분들이 좀 나오셔가지고 몇 분이 계신데 한동안은 아무도 없었어요. 그러다가 이제 학회를 통해서 많이 뵙고 그 다음에 또 같이 공동 연구하시는 분들도 생기고 그래서 참 보기가 좋고 그 다음에 이제 저 같은 경우에는 제가 가지고 있는 자리? 제 위치가 도와줄 수 있는 위치가 되어서 한국에서 만약에 도움이 필요하시다거나 뭐 여러 가지 네트워크를 통해서 그러니까 미국하고 전 세계적으로 네트워크가 있기 때문에 학생분들이 만약에 좀 접근하기 힘든 분들이 계시면은 저를 통해서 제가 좀 연결고리가 되어드릴 수 있지 않을까 그런 생각도 좀 해봅니다. 인터넷으로 이렇게 하는 거 좋은 것 같아요. 그래서 만나가지고 같이 궁금한 거 있으면 얘기도 하고, 지금 아까 여기 그 김도윤 학생 있었는데 그 친구랑 또 예, 저기 또 다른 거기 김건혁 학생인가 또 다른 학생이 있는데 연락 와요. 같이 막 줌하고 얘기하고. 그러니까 학생들도 같이 분류군이 다르더라도 혹시 같이 공유하거나 궁금한 게 있으면은 저야 언제든지 시간을 내서 도와드릴 수 있습니다.
네, 고맙습니다. 하여간 자주 우리 모셔야 될 것 같으니까 그때마다 좀 부탁드리겠습니다.
네, 감사합니다.
우리 신승관 교수 오지 같이 이렇게 오거나이즈하고 하면서 마지막으로 한 말씀 해주시죠.
예, 제가 어떻게 보면은 처음으로 이렇게 학회 오거나이징을 하게 된 건데, 이렇게 흔쾌히 연사로 와주신 분들께 너무 감사드리고, 예, 또 다들 좋은 발표 해주시고, 또 아주 특별한 문제는 없이 몇 번 끊기긴 했지만 매끄럽게 진행돼서 너무 좋았던 것 같아요. 그래서 이렇게 송호준 교수님께서 얘기해 주신 대로 정말 같이 자주 보고 또 학생들과 그리고 또 여러 외국에 계신 연구자들뿐만 아니라 다른 분들과도 연결이 될 수 있는 그런 기회를 자주 만들 수 있으면 좋을 것 같습니다. 감사합니다.
네, 모두 감사드리고요. 그러면 우리 이번 가을 학회 심포지움은 여기서 마치기로 하겠습니다. 너무들 다시 한번 감사드리고 다음 기회에 또 뵙기로 하겠습니다. 자, 수고들 많이 하셨습니다.
네,
감사합니다. 우리 신 교수님 주무셔야죠.
네. 안녕히 계세요. 네.
그래요. 이제 그러면 이거 지금 호스트를 누가 하고 있죠? 지금 신 박사 신 교수가 하고 있죠?
네, 제가 돼 있는 것 같습니다.
그러면 이거를 마지막에
네.
클로징을 할 때 잘해야 될 거예요. 클로징을 정상적으로 해가지고 레코딩이 돼서 학회에다가 이걸 넘겨줘야 될 거예요. 여태까지 한 게.
네네네.
그러니까, 나중에 피니시를 하게 되면 이게 컨버팅이 되잖아?
네네.
컨버팅이 되는데 그거를 중간에서 끊지 말고 이게 지금 한 6시간 가까이가 계속 레코딩이 됐을 테니까.
아니요, 지금 중간에 끊겨가지고 계속 이렇게 됐다 저렇게 됐다 돼가지고 저도 어떻게 되는지 한번 봐봐야 될 것 같아요. 계속 중간에 끊겨가지고.
오케이.
예, 한번 일단 체크를 해보겠습니다.
오케이, 그래요. 우리 송정훈 박사님도 감사합니다.
네, 감사합니다. 초대해 주셔서.
네.
네.
감사합니다, 송 박사님.
네, 고맙습니다.
이거 리코드를 누르긴 했나, 이 박사?
레코드 자동으로 눌려져 있는 것 같습니다. 지금 기록 중이고요. 네, 근데 제 생각에 이따가 통화 한번 하셔야 될 것 같습니다. 네, 아무튼.
오케이. 일 있으면 전화 주세요.
네네. 그러면은 방은 폭파시키도록 하겠습니다.
네.